Good evening, Professor Dan, Professor Carey, Professor Jesse, and my colleagues here. Good morning. I can see most of the speakers here, and we are still waiting few more students to join, but I think we can start the program. I welcome you all again for this Indo-US bilateral workshop funded by Indo-US Science and Technology Forum. As a convener from India, I welcome you all for this workshop. Professor Dan, if you have to say a few words, and then we can start the program.
Well, welcome everyone to the second day of our workshop. I appreciate everyone's participation and look forward to some excellent presentations.
So I now request Ms. Anusha Gauri, our research scholar, to compare the program and introduce the chairperson for this session this morning. Anusha, over to you.
Good morning. I would like to welcome the session chair, Dr. P. Purushothaman, sir. Dr. P. Purushothaman is an associate professor of Department of Civil Engineering and Environmental Engineering in SRM IST. He has completed his postdoctoral fellowship at Research Center for Environmental Changes, Academia, Seneca, Taipan, 2009. His area of research interest focuses on environmental impact assessment, eutrophication of surface water bodies, water quality, and hydrogeochemistry. Over to you, sir.
Thank you, Anusha. Good morning, everyone. I thank the conveners for giving me the opportunity to chair this session. To begin with, today's keynote lecture will be, we have 4 keynote lectures. And to start with, let me introduce the first keynote speaker of this day, Professor Jesse Bell. Professor Jesse Bell is the Claire M. Humboldt Professor of Water, Climate, and Health at the University of Nebraska Medical Center's Department of Environmental, Agricultural, and Occupational Health and the School of Natural Resources at the University of Nebraska Institute Lincoln of Agricultural and Natural Resources. His study and experience are centered on determining the effects of environmental and climate change on natural and human processes. Dr. Bell founded and held the first interagency post between the National Oceanic and Atmospheric Administration and Centers for Disease Control and Prevention before coming to UNMC. I welcome you, sir. Sir, over to you.
Thank you so much. I really appreciate it, and I appreciate you inviting me to be here today. I was wondering if I could share my screen. It says the host participant screen sharing is.
Yes, sir, it's enabled. You can share.
Okay. Doesn't seem to want to let me do that. Oh, there we go. Let's see if I get this to pull up. Okay. Are you guys seeing this okay?
Yes, sir. Yeah.
All right. Well, thank you so much. Like I said, I really appreciate it. I appreciate the opportunity to speak here today. I hope everybody is doing well and staying safe in this obviously difficult time. But what I'm going to talk about today is building the water, climate, and health nexus, and talk a little bit about some of the work that we're doing at the University of Nebraska to try to understand and address some of these challenges facing water, especially water here in the state, region, and the world, and what that means for human health. And so we know we have a number of challenges. We're seeing a changed environment around us. So currently we have a growing population that will continue to grow over the next century. We have increase in waste generation and potentially pollution. And we also know that our climate is changing as well. And so that's where we get at this water, climate, health nexus, because these changes in the environment, the changes in the world that we're seeing around us, that impacts our health, and it impacts our health in a variety of different ways. And I'm going to talk about some of those challenges and what that potentially means for our health, and how we can potentially start to try to address some of these issues as well. And at the center of this is water. Water is kind of the key conduit to a lot of these different issues that we're facing. And when you think about water, you can think of it in a variety of different ways when we come about challenges, especially challenges that can be related back to our health. Sometimes we have too much, which is flooding, too little, which can be drought, and then potentially not clean enough, which can lead to water quality issues that could also lead to human health impacts as well. And so to start this off, I'm going to talk a little bit about some of the water quality issues and talk a little bit about some of the water quality issues that we're facing in Nebraska and how this is a potential challenge for us and also a challenge internationally as well. When we talk about water quality, we talk about nutrients in water, nitrogen, phosphorus, pathogens such as bacteria, geogenic contaminants such as arsenic, uranium, emerging contaminants such as hormones and antibiotics, pesticides, and even lead, which is due to infrastructural issues here in the US. And there's a variety of different ways that those contaminants get into the system. Some of it's through agriculture, some of it's through development, some of it is just naturally occurring. And here in Nebraska, one of the big issues that we face is nutrient contaminant, especially nitrogen in our water in the form of nitrate. A lot of this is due to agriculture in one capacity or another, either from the application of fertilizer on fields or from issues that we face with livestock and contamination getting into the water system that way. This is especially important for smaller rural communities in places like Nebraska and other parts of the United States. That provides a variety of different challenges for some of these communities to try to address and deal with some of these water quality issues. And in trying to deal with some of these water quality issues, a lot of times we think of the health impacts, but whenever there's a health impact, there's either mitigation or lack of mitigation, and there's costs to that on both sides of that spectrum. And with, for example, here in Nebraska, we're seeing increase in nitrate and some other contaminants in our water systems. And so some small communities are now having to invest into solutions for some of this water quality problems. And with these solutions, these mitigation efforts, they would require a financial cost. And so we're seeing small rural communities in Nebraska investing millions of dollars for small populations of people to try to potentially mitigate against some of these contaminants. But why do we do that? Why do we spend so much money and so much investment of time and research and effort around some of these issues? Well, that's because it's linked to our health in a variety of different ways. This is just one example here from Nebraska and from the central part of the United States. And we're talking about that nitrogen or from nitrate in our drinking water. The sources, like I said, is from fertilizers, animal and human waste. Regulatory limits here in the United States is 10 milligrams per liter. Greatest exposure is in agricultural areas with private wells that are not regulated. And private wells due to the fact that it's not regulated under our Clean Drinking Water Act, and also because of sparse measurements, not knowing what are the concentrations in those areas as well. And this gets back to it. When we were talking about nitrate in our drinking water, we know that nitrate has a link to human health outcomes. The that 10 milligrams per liter that is set for the national standard is based off of methemoglobinemia, which is also known as blue baby syndrome. It's not set to other potential human health consequences. And so that is one of the things that we're trying to better understand here in Nebraska and throughout the United States is what are the potential exposure risks at even lower levels here in the US and internationally as well. And so there's been a number of studies that have looked at some of these relationships. There's been a number of studies to understand that you see higher rates of certain human health outcomes associated with nitrate in drinking water, especially. Like I said, that methemoglobinemia, but also colorectal cancers, thyroid disease, neural tube defects, which is birth defects of the spine, brain, and spinal cord. There's been a number of studies looking at that. There's also been work that has shown that it's increased heart rates, nausea, headaches, and abdominal cramps with too high of concentrations. Other cancers have also been shown to have some relationship with nitrate in drinking water. Pediatric brain cancers, and I'll talk about that in a second. Kidney cancer, bladder cancer, non-Hodgkin lymphoma. Here in Nebraska, there's been already one study that's looked at non-Hodgkin lymphoma and found that there was a threefold increase in risk when there was the presence of nitrate and atrazine. So that's another thing that we have to think about in the environment today. We're not just talking about one contaminant in our water systems or air or anything like that. There's multiple potential contaminants and exposures that could potentially lead to human health outcomes. And so trying to understand how those factors or those issues combined together makes it even that much more complex to understand the ramifications. And then in addition, there's been studies looking at Alzheimer's, diabetes, and Parkinson's with high nitrate concentration in drinking water. And as I've mentioned, this has an impact, especially on reproductive health outcome. At the Centers for Disease Control and Prevention, where I used to work previously before coming to UNMC, there was a report that came out from CDC that showed 1996 that there was a cluster of spontaneous abortions or miscarriages in rural Indiana. It's a state in the United States. And what they did was, when they were investigating what was the potential cause of that cluster of spontaneous abortions, they found that there were private wells that had nitrate in their drinking water that exceeded 20 milligram per liter. So it was much higher rates than that 10 milligram that is our national standard. But again, private wells are exempt from the drinking water standard, or the national drinking water standards here in the United States. They switched the individuals to low nitrate water, and soon after they were starting to get healthy births in that region. In addition, there's been work that's done looking at the central nervous system malformations. And so these are birth defects associated with exposure to nitrate in drinking water. 5 out of 6 studies that have been done across the world have found that there's a positive association with nitrate. And 4 of those studies have shown that concentrations less than 10 milligrams per liter below the national standard have actually been associated with some of these malformations. So looking at this from the lens of public health, we really want to be able to try to identify who are those most at risk or vulnerable populations to some of these potential exposures. Obviously, a lot of people are potentially at risk, but there are certain populations that are more at risk than others. This is in particular young children appear to be particularly susceptible, especially when we're talking about the blue baby syndrome. Pregnant women and women that are considering getting pregnant or potentially could get pregnant, especially in that first trimester, are most susceptible. In addition, people with oxygen transport issues, and then also just anybody that has high nitrate concentrations in their drinking well could potentially be at higher risk. It's also been shown that diet plays some role in that, with certain diets potentially lowering risk and other diets potentially increasing the risk. So going back to it, when we're looking at this from the lens of public health, we're really curious about what does this actually mean in terms of human health, especially in the communities that we serve and the communities that we're trying to positively impact. And so in Nebraska, we know that we have very high rates of pediatric cancer. And we actually have one of the highest rates of pediatric cancer in the United States. And this is obviously concerning for a variety of different reasons. And one of the things that we know is that Nebraska is an agricultural state. We have a lot of row crop agriculture, and we have a lot of people in our communities or in our state that are on private wells. So these are wells that where people are pumping water straight out of the ground for consumption. And so based off of the scientific understanding that we have between nitrate exposure and other agricultural chemicals and other chemical exposure and the potential relationship that that has with pediatric cancer, this is obviously an area of major concern for us. And there's been a number of studies trying to look at the rates of cancer across Nebraska and trying to understand if those rates of cancer are potentially linked to certain things. One of the things that we know, especially when we're talking about pediatric cancer, it's not homogeneous in the way that it's spread across the state. There are certain regions that of the state that have higher rates, and especially of certain types of cancer. For example, pediatric brain tumors. We see higher rates of pediatric brain tumors on the eastern part of the state of Nebraska. Some of my colleagues, Alan Kellogg is one of the individuals here, have looked at this, looked at some of these rates and tried to understand what is potentially going on. One of the things that they did was look at it in the terms of watersheds across the state. Again, trying to get at those naturally occurring boundaries and not those geopolitical boundaries that we typically put on a lot of our studies. And one of the things that I found most interesting of this particular analysis that that was performed was the red is an indication of where the pediatric cancer rates are above the national average. Green indicates areas that are below the national average. And as you can see, this is pediatric brain tumors. This is leukemia, and C is lymphoma. And one of the things I wanted to point out here is if you look at the pattern, the general pattern, and this is obviously not looking at it from a statistical standpoint, but just from more of an observational standpoint, if you look at the pattern of where the highest rates of pediatric brain tumors are in Nebraska, and you look at where we have the highest rate or where we have more row crop agriculture, you'll notice that there's a general pattern there that follows a very similar pattern compared to the two. And you don't see that between leukemia and lymphoma. And this is another issue that we're definitely concerned about, which is the birth defects in the state. We know that in the state of Nebraska, in certain regions, we've been seeing higher rates of birth defects and increasing rates of birth defects in certain parts of the state, and trying to understand what are some of those relationships and what are some of the potential outcomes as well. Where we're seeing some of the highest rates is also where we're seeing some of our row crop agriculture. So in summary of this part of this presentation, studies have found various outcomes, health outcomes associated with high concentrations of nitrate in drinking water. There's still a lot to be understood about what are some of these other contaminants that we're potentially being exposed to, and understanding what are those potential risks to human health. And in particular, one of the things I'm most concerned about is the high rates of pediatric cancers that we see in the state, and trying to make sure that we address and understand what those issues are and how we could potentially do efforts to reduce those rates and protect those populations that are most at risk. And then again, birth defects is another area of concern. So that gets to the water quality piece. And now I'm going to talk about water quantity. And this is where we get to that climate part and the potential relationship that that has to human health. We know that we're seeing changes in our climate system. Not only are we seeing changes in temperature, but we're seeing changes in our precipitation patterns. those changes in precipitation patterns can lead to more extreme events. So more flooding, more drought, and those things can lead to human health outcomes. One of my colleagues said that we're living in the new abnormal. The world is no longer normal. It's a changed world, and that changed world is having impacts on us on a regular basis. We know around the globe, this is obviously an example from the United States, but from around the globe, we've seen higher temperatures. We're seeing changes in precipitation patterns. We're seeing more flooding. Hurricane and severe storms are changing, higher sea level rise, more droughts in certain parts of the world, and more rain in other parts of the world. Heat waves are becoming more intense. And then here in the United States, we're seeing an increase in the wildfire season, which is having an impact on air quality and also human livelihoods throughout the western part of the United States. And so when climate is linked to our health, and it's linked to our health in a variety of different ways, climate impacts us on a daily basis. And when you change the climate, you change the environment that you're used to, that could potentially lead to human health consequences. So with increased temperatures, changes in precipitation, changes in extreme weather events and sea level rise, all of these things change the environment around us. And that change in the environment then can lead to human health consequences. And the reason I put this figure up here is because we really, when we think about that pathway, that linear pathway from climate drivers to exposure pathways to human health outcomes, a lot of times we don't really always think about it in the context of the social and behavioral and the environmental and institutional context. And so social and behavioral is identifying, and this is where really that public health piece comes into this, it's identifying what is the community, the individuals that live in the community, what are they experiencing? Who are they? What are the demographics? What are the vulnerabilities? What are the risks that those populations face? And so here in the United States, there's certain factors that are social determinants of health. And so these are things that potentially lead to you being more exposed or more at risk of a variety of different potential health impacts, including age, gender, race, ethnicity, poverty, housing infrastructure, education, discrimination, access to healthcare and access to care, pre-existing health conditions that can lead to human health outcomes. And then the environmental institutional context is really getting at the land, the environment around you, the built infrastructure, and how that could potentially increase or decrease the amount of health outcomes seen when these types of events occur. And so, like I said, we've seen that our climate has changed. We are living in a changed climate, and in the future, we expect our climate will continue to change. Similar to when we're talking about the financial costs of poor water quality, changes in our climate also has a financial cost. And so here in the United States, this is $1 billion disasters. These are disasters that exceed climate and weather related disasters that exceed $1 billion of economic loss. And over the last 40 years, we've been seeing an increase of these types of disasters across the United States. Similar patterns across the globe as well. And so, like I said, climate impacts your health in a variety of different ways. And so when one of these disasters happen, it's not just a financial cost, but there's also a health cost associated with it. And there's the direct. Those are the things that happen immediately, and the impacts can be seen instantaneously, and then could potentially lead to human health outcomes. So that's extreme heat, air pollution, extreme weather. And then there's the indirect. And these are much harder to monitor and understand. And that's the spreading of disease because of a changed climate, destroying and disrupting of food supplies and water supplies, and disruption of well-being as well. And so here in the United States and Nebraska, where we're at, we're definitely aware of these situations, as many other places are aware as well. We're seeing more of these types of events, and the impacts are becoming more severe. Just in 2019, the costliest inland flooding event in US history occurred. And so reported is 3 deaths. I promise you the impacts were much beyond 3 deaths, but that's what was potentially reported because of that direct versus indirect and our difficulties of capturing those indirect health impacts. Hundreds of people were displaced, and it was the costliest inland flooding event, as I said, in US history. And then this is an area that I'm really passionate in trying to better understand: drought. How is drought impacting our health? Internationally, we know that drought has likely caused more deaths than any other climate-related disaster over the last century. Internationally, that's due to famine and malnutrition in a lot of different places. We also here in the United States see the impacts of drought. We see the impacts of drought on a yearly basis. There's actually a large drought impacting the western part of the United States and even here in Nebraska right now. This is work with one of my colleagues here at UNMC, and one of the things that we've done is look at mortality, changes in mortality rates over the last 40 years. And one of the things that we found was in Nebraska, there is an increase in mortality rates in white males and white females between the ages of 45 and 65 years of age during drought events over that 40-year time span. And so we're not just interested in identifying that there's human health consequences. We're also interested in trying to figure out how do we move this forward into making sure that we're addressing these issues as well. How are we actually getting boots on the ground to make sure that, you know, the next drought that we face isn't as costly in the terms of human health? And so one of the things that we're doing is we're going around the country to host workshops to bring together regional experts to understand how drought is impacting human health and how to better prepare for that in the future. And we're actually working with a federal agency and federal institution, NIDIS, the National Integrated Drought Information System, to create a strategy document for them so that they can work on the public health impacts of drought moving forward in the future. So our environment is changing, and that change in environment is costing, impacts are costing, is a cost to our society, but also a cost to our health as well. And so this is where public health really can come in and try to help with addressing some of these issues. And that's through education of the public, enhancing monitoring, better research of some of the challenges that we face, and then contributing to the public dialogue. That's what public health can bring to the table in trying to help with addressing a lot of these issues. And so that's why I think it's important whenever we're talking about looking at some of these environmental changes, environmental impacts, to make sure that public health is contributing in some capacity. And that's what we're doing here at the University of Nebraska. We have the new program and the new initiative around water, climate, and health. This is funded through the Claire M. Hubbard Foundation. It's a local nonprofit philanthropy institution here. And what we're trying to do is build interdisciplinary collaboration across the University of Nebraska to address some of these public health challenges in Nebraska and around the world, in making sure that we're doing better research, education, policy development, and training the next generation so that they can address some of these issues. And that's also where Water for Food comes in. They're a part of this initiative and trying to build around water, climate, and health so that we can address some of these challenges. Because what we want to do is build a safer and healthier state, and also a safer and healthier world, by identifying multidisciplinary solutions, building collaborations, educating and informing people on the issues that we face, providing mentorship, and recruiting experts from around the world to help address some of these very complex and difficult challenges. And so with that, I just wanted to say thank you very much to all of you, and also to some of these people that helped make this possible. And if you have any questions or interests in our program, please don't hesitate to reach out to me or the program manager, Megan Langle. Her information is up here. And I'm happy to take any questions if we have time.
Thank you, sir. Thank you for your wonderful lecture on the impact of not only groundwater or water quality, also the quantity on human health. Now I open the forum for questions. We can accommodate 2 2 or 3 questions. If you have any questions, you can type in the chat box. Okay, so thank you, sir. Thanks for your wonderful lecture, and we can accommodate questions. If you want, you can type in the questions. We can get the response from professor later. So I'll move on to next keynote lecture. Professor Kerry Weber. Professor Kerry Weber, Associate Professor, University of Nebraska, Lincoln. Her research interests focus on the intricate interactions between microorganisms, including viruses, and the environment at the molecular scale, the ecosystem scale, and ultimately the global scale. Her current project focuses on microbially catalyzed mineral dissolution and precipitation, implications to heavy metal and radionuclide mobility and geovirology. I welcome you, ma'am, and you can take over the show.
Well, thank you very much for the introduction, and thank you very much to the conveners for inviting me to give this talk, as well as for putting this together. So this is great, listening to the presentations yesterday, as well as Jesse's introduction, which was great to hear. Whoops, wrong talk. Hold on. Oh, sorry about that. Right. Okay, are you seeing a WaARISH slide? Microbial activity in geogenic groundwater?
Yes, ma'am. Yeah.
Okay, fabulous. Okay, and the appropriate screen or a note screen?
No, appropriate, yes.
Okay, okay, great. Thank you very much. So Jesse had introduced and talked about groundwater contamination, and I'm going to pick up on the water quality theme. And I'm going to talk a little bit about how microorganisms play a role in the mobilization. And we recognize that microorganisms can catalyze the mobilization of arsenic, and that's been a very well studied process over the last few years. And I'm going to take another step and talk a little bit more about the mobilization of uranium, more specifically. And before I begin, I want to acknowledge many different collaborators on this project. And yesterday there was an introduction of the WaARI program. It's in collaboration with the Water for Food Institute. And there's components of this research that a graduate student, Rudra Mohan Pradhan, had collaborated in with Jeff Westrop, as well as Rajesh Singh. And I believe he's going to be giving a talk later today in contributing to the work that I'm going to be presenting. And of course, collaborators at UNL and the Daughtry Water for Food Institute have certainly been influential and helpful in building this research program. So as we begin, the one thing that I just want to remind everybody that the chemistry of groundwater is a reflection on the lithology and the life that water does pass through and around. So microorganisms are living within these pore spaces of the groundwater. They're attached to the mineral surfaces. And of course, they can catalyze many different reactions, which can either potentially create new minerals or perhaps dissolve the minerals. And when we start thinking about the water itself, it's what they dissolve and would mobilize in water. Sometimes that they can do great things and we apply their metabolisms for bioremediation. But on the flip side of things, as they're living and surviving and just getting by in these subsurface environments, there also can potentially create problems. And so I'm going to talk a little bit about that. When we think about nitrate and nitrate contamination in widespread environments, we've heard about that health impact. And you're looking at a interpolated map of nitrate concentrations that have been measured in the High Plains aquifer. And you can see there's many regions that are dark blue, which are above the maximum contaminant level within this aquifer. Many individuals rely on this for groundwater. And yes, nitrate itself does have health effects, but when I start thinking about what nitrate is, nitrate as a chemical, it's also an oxidant. So that makes me begin to wonder, are there consequences of nitrate contamination that can result? So that brings me to thinking about uranium, an unintended consequence of nitrate contamination, or a primary contaminant leading to a geogenic contaminant. So when we look across the United States at uranium concentrations, there are regions that we do see high uranium concentrations. Of course, these levels above 30 ppb have been linked to negative health consequences. Primarily, it is kidney disease. But bone osteotoxicity, as well as a few studies, and I say just a few, have indicated an increased cancer risk as a result to consuming naturally occurring uranium in groundwater. So there is a health consequence. What's interesting about Nebraska with respect to uranium, we only have one mine located in Nebraska. So this is within the western part of the state, and that's an in situ leaching mine. So over here on the eastern part of the state, we don't have any really anthropogenic inputs to mining. Whereas other parts of the United States, such as Wyoming, Colorado, there are mining and milling waste operations that do contribute to legacy waste within the United States. But within Nebraska specifically, these regions, there isn't a known source of anthropogenic contamination. And prior studies had demonstrated that the isotopic signature most likely is not that of, say, uranium coming from a phosphate fertilizer. So perhaps there is a natural cause for uranium in groundwater. So of course, we have to think about, well, how does uranium get in groundwater? Why is it changing? What happens? Well, we have to start thinking about what's going to be in the water and what's not, especially as when it comes to uranium. Uranium in the environment is primarily going to exist in two different redox states. It's going to exist in the hexavalent state as well as the tetravalent state. In the hexavalent state, it is going to be oxidized. Most often, especially in natural groundwaters, it is going to exist as an aqueous ion, often complexing with carbonate ligands, which are also very common in natural groundwaters. However, when it is reduced, it tends to precipitate as solid phase uranium-4 bearing minerals, and thus it's immobile. So these oxic versus anoxic conditions can control uranium mobility, but also just even thinking about oxidizing versus reducing conditions can also play controls over uranium mobility. We know that when in oxic environments, that carbonate ligands can play a significant role in the desorption of uranium from the surface of particles. That's been demonstrated experimentally as well as within the field. But additionally, these reduced minerals, even under anoxic conditions, can be liberated through oxidative dissolution. So basically converting that solid phase mineral to an aqueous species that is now mobile in the environment. And nitrate, whether it is an abiotic reaction or even in the presence of cells, can catalyze those reactions above the MCL. So it can play a significant role to uranium mobilization. Now, nitrate reduction specifically, when we start looking at microbial life and how it's going to influence these reactions, as nitrate is reduced to nitrite, that primary first step of nitrate reduction, the production of nitrite can result in a rapid abiotic oxidation reaction. So nitrite serves as the oxidant and oxidizes the reduced uranium to that aqueous species. So there's initially a microbiological reaction and then a subsequent abiotic reaction as a result. There are other mechanisms such as nitrate-dependent iron oxidation, where the microorganisms are breathing the nitrate and eating iron, precipitating out an iron oxide that is reactive, which can also react with the reduced uranium. And then that iron oxide can also reabsorb that oxidized uranium as well. But there's also bacteria that can eat the uranium and breathe the nitrate and also mobilize the uranium through that process. So microorganisms can play multiple roles in the mobilization of uranium. So they potentially can control this. But I would be remiss if I also didn't talk about, there's also bacteria out there that can reduce the uranium as well, coupled to the oxidation of organic carbon. So in an environmental system, you have these controls over oxidation and reduction that can control that mobility. So these organisms can play a very significant role in what is going to be mobile or immobile. So as we start to think about that, we know that nitrate can potentially play a role. We began to start thinking about, broadly speaking, what are the roles in natural systems when we start looking at nitrate concentration, groundwater concentrations, and uranium groundwater concentrations. And we first began this project by simply just exploring, you know, if we look at the co-occurrence of nitrate and uranium, are there areas, do they co-occur? Could nitrate potentially be a driver? So this study began looking at a metadatabase of uranium and nitrate groundwater concentrations in two alluvial, two important aquifers within the United States, which can provide drinking water to over 2 million people. And what we ended up finding, when we look at this, you can see these areas which have the dark blue as well as this dark pink or magenta color that ends up becoming kind of a purple color overall. And so this purple color kind of was suggesting to us that, yep, there's a co-occurrence. So we take it a step further to do a spatial correlation analysis and do find that, yes, when we do have these high concentrations, we also find this high correlation. So there appears to be a correlation between nitrate and uranium in these circumstances. But of course, we know that correlation doesn't mean causation. So continuing to explore this in one of these environments in which we've measured uranium as high as 300 ppb in the groundwater, and down gradient, a well that would be below that 30 ppb of groundwater. So in this system, located in an alluvial aquifer near the Platte River, up gradient, we tend to find a more oxidized system. And down gradient, we find a much more reducing system. And I'm not going to go into the details of the redox states, but what I just want to point out is that this up gradient portion, which is more oxidized, where uranium is at the highest concentration, most of the uranium is actually in this oxidized state. So this is Zane's analysis of the natural sediments. So most of the uranium is uranium-6. But down gradient, where we have more of the reduced uranium, it's about 50% reduced and 50% oxidized in this reducing system. So our hypothesis was, in this where we have these reduced uranium minerals, if nitrate is introduced into the system, we could potentially mobilize that uranium via oxidative dissolution. So we set up a series of experiments, taking these natural sediments that are naturally reduced and setting up upflow column reactors where 50% is the natural sediment, and then another 50% is acid wash combusted sand. And we're replicating these lithologic facies that we would find are in the subsurface themselves. So we do all this work even after collecting the sediments under anoxic conditions, bring them back to the laboratory. Everything is handled in an anaerobic glove bag to make sure oxygen never has a chance to oxidize our reduced minerals. So you're looking at a picture from within that anaerobic glove bag of some of these column experiments. We introduce an influent that contains artificial groundwater that contains bicarbonate. So the carbonate ligand, and we have chloride added as the conservative tracer. Nitrate is then amended into this treatment in the column, and we add bromide as a conservative tracer to follow that nitrate amendment. We have another treatment in which we add sodium azide to inhibit nitrate respiration specifically. So when we begin to look at these results, what I want to point out here is at the beginning of the system, this is all of the 6 treatments. So these are all done in duplicate. So all the 6 different columns, we see with the introduction of the influent, the mobilization of the uranium that would be associated with the introduction of the carbonate ligand. So we have a bicarbonate driven desorption. Once we observe that plateau, the system is then amended with nitrate, at least which treatments, right? So we maintain where we have a bicarbonate only as one with nitrate and then nitrate and azide. And what you end up seeing is that we only see mobilization of the uranium or increase in this uranium in the effluent where we have the nitrate. If we only have bicarbonate, we have no further increase. If we add the azide where we've inhibited microbial activity, we do not have an increase. We see the same response with nitrate reduction. So the azide did indeed inhibit nitrate reduction. We don't see nitrate reduction, only we see nitrate reduction where we have nitrate added. And based on what we're measuring for nitrite appears to be the end product. So we have the same amount of nitrite being produced as we see for nitrate. When we have these spikes, we also see these spikes in uranium that are being produced over time within the system itself. And so this is indicating to us that microbial activity is driving these reactions. Nitrate alone cannot do it, but it's the presence of the microbes that's catalyzing this. If we follow total cell abundance over time, we further see this increase in the number of cells as a result of this reaction. So they're growing, they're making a living. So as we, at the end of the experiment, we disassembled the columns, of course, processing them for solid phase analyses and sequencing of the microbial community. So through shotgun metagenomic sequencing and reassembling the genomes, we can identify those pathways. And just what I want to point out is that the total gene copy numbers that are present for nitrate reduction to nitrite certainly are very abundant. So this appears to be the primary pathway that many of the taxa present in the nitrate addition do have, which coincides and supports what we measure within the experimental system, that nitrate reduction to nitrite is prevailing. But the question is, does nitrite then actually oxidize the reduced uranium? So we conducted a separate column experiment where we add the nitrite and nitrite plus azide and bicarbonate only. And we see that nitrite is having an effect on uranium mobilization. Even when we add the azide, we don't see any difference, which tells us that this is an abiotic reaction. So together, when we start thinking about this, we've got nitrate being reduced to nitrite, and the nitrite is abiotically oxidizing the uranium. And I want to bring up the story of iron because iron becomes important in here as well. Because within the column study, when we have nitrate present, we don't see as much reduced iron, which tells us that iron oxides are being precipitated. And we do measure these at the end of the study within the column itself. So we are precipitating out iron oxides. Iron oxides have the potential to absorb iron. So a separate study where we looked at absorption of uranium to reduced sediments. So we kept conditions anoxic in order to conduct these adsorption experiments. And another series where we conducted adsorption of uranium to oxic sediments or air oxidized sediments. And then in collaboration with a colleague of mine at the United States Geological Survey, and Jeff Westrop and Rajesh Singh also collaborated to help start working on the development of the surface complexation model, develop this surface complexation model to determine how much of the uranium could be accounted for by the precipitation of iron oxides. So if we assume that perihydrate is precipitating, we can account for that difference in the amount of uranium that is being absorbed. So there's likely uranium retention going on within the column because of these precipitation of iron oxides as a result. So our lab results support the link between both bicarbonate and nitrate mediated mobilization, and also indicate that perhaps even under some circumstances where you have iron oxide precipitation, you could retain that uranium. And it tells us a little bit about this system where we have this up gradient and down gradient flow occurring, that perhaps there are redox fronts. And so this is an area to begin to investigate, of starting to consider these redox fronts where these oxidants such as nitrate potentially can move in and start mobilizing those geogenic contaminants as those in these systems. But another question, and that often gets asked is, well, there's many regions where you have high nitrate, but you don't have uranium. Is it that there isn't any uranium, or what could be also happening? And it's complicated, but when we start looking at the metadatabase, what we can see is that there appears to be a tipping point. We could say it's between 8 to 11 milligrams per liter in the High Plains aquifer, maybe in the Central Valley aquifer, maybe 6 to 11 milligrams per liter of nitrate might be that tipping point. So over 11 milligrams per liter, more likely to see uranium mobilized. We were actually curious to begin to test this. And so collecting oxbow lake sediments, which would be from a fluvial system, so you could think of this in geologic time as early, before they're buried in any subsurface system. But we selected this system located near Riverton, Wyoming, to allow us, because it is contaminated with legacy mining waste. So we could go into an environment that is indeed exposed to uranium, so we know we'd find it there. So we simply set up a series of culture conditions in which we have these bottles, half 25% sediment, 75% culture medium. Oops, we either amend with deionized water or we amend with nitrate. And after we do add some uranium-6 to the system, because they are naturally reduced otherwise. So we add uranium-6, and after 24 hours, we introduce the spike. And interestingly, when we add nitrate, these solid symbols here are the nitrate additions. When we add nitrate, uranium-6 concentrations decrease, which is suggestive of reduction. We see the same to be true when we start to follow aqueous iron concentrations are increasing. So there is, and what is suggestive of reducing conditions that are onsetting with the addition of nitrate. And we validated that indeed, when we add that nitrate in the sediments, we're actually catalyzing reduction reactions. Zane's analysis tells us that we are precipitating uranium-4 as a result. So this is telling us that low concentrations of nitrate can stimulate reducing conditions, not necessarily mobilization. And when we start looking at, is microbial activity involved in this process? We used chloramphenicol. It's another antibiotic which inhibits protein synthesis. So we can inhibit microbial activity. These circles here that you're following, these are without the chloramphenicol. So when we add chloramphenicol, what you observe without the chloramphenicol addition, well, without the chloramphenicol, we do see the loss. With the chloramphenicol, we don't see significant loss. So that indicates to us that this reduction activity that we're observing is biological. And due to time constraints, I won't go to the microbial community and all the processes going on here. But what I just want to summarize is that, nitrate concentrations, there is a tipping point. What I didn't go into in the story, in the research before, is that yes, if we add more than this 11 milligrams per liter into that system, we get oxidation as we would expect. But it's these low concentrations of nitrate, which actually can stimulate the uranium reduction. So not all nitrate is bad, but there is a potential tipping point that we do need to consider for what is going to control these reactions. And when it comes to uranium in these alluvial aquifer sediments, we have to take into consideration both the biological as well as the abiotic reactions that can drive that mobility. So with that, I would be happy to take any questions. And just to reiterate, none of this work could have been accomplished without the many students, postdocs, and collaborators within this project. So I'm happy to take any questions.
Thank you, ma'am. Thank you for your wonderful lecture on the influence of this nitrate on uranium mobilization. Before opening this forum for questions, ma'am, I have one small query that when you study about iron oxides influence, what about the manganese oxides? Because manganese also was one of the greatest oxic or mineral compound in the this oxic and anoxic pathways. So how does that manganese can affect the nitrate?
So manganese, manganese and nitrate, the biological reaction between the two, there isn't a clear demonstration of manganese oxidation coupled to nitrate reduction biologically. Now, what I can say though is there are some, when you have reduced manganese, well, different manganese oxides can influence different redox reactions, iron redox reactions. And so it's not that they're not necessarily uncoupled, but it is a little bit more complicated. Within the system that we chose within Nebraska, manganese isn't as significant as a player. Now, I can't say the same to be necessarily true within those Riverton, Wyoming sediments, because there's the potential there to play a role as well. So yes, manganese can play a role. We haven't, in our research specifically, we haven't dove into that, but there are several, abiotic reactions that can result between iron, manganese, uranium, manganese.
Thank you. Now I open the forum for questions. We can have a few questions. Yeah, attendees can post their questions in the question and answer box. Okay, we will take the questions later and we'll try to get the response from Professor Weber. Thank you, ma'am. Let me move on to the next keynote lecture. Professor Alan Kellogg. Professor Alan Kellogg is the director of Idaho Water Resources Research Institute, and served as the founding director of Nebraska Watershed Network at the University of Nebraska in Omaha, as well as the director of University of Nebraska Medical Center Center for Environmental Health and Toxicology. He is a prolific writer who has contributed books such as Modern Poisons, An Introduction to Contemporary Toxicology and Twists, a science-based fiction about an extraordinarily infectious prion, are his most recent books. His third book, generally regarded as Safe, will detail the evolution of US food and drug safety from 1900 until present day. I welcome you, sir, and please take over the forum.
Thank you very much for the very nice introduction. I need to share my screen, and I'm being told right now that I cannot. Oh, here it is. All right, excellent. Can you all, I'm just double checking. Can you all see my slides?
Yes, sir.
Great. Thank you very much. Again, I thank you all for inviting me to give this talk. I'd particularly like to thank Dr. Chakraborty and Dr. Dan Snow for their very gracious introduction or invitation to give this talk tonight. I greatly appreciate being here and finding the talks thus far to be very enlightening and very interesting. I wanted to start off and take a page out of one of my colleagues' books, which is Dr. Tyrone Hayes. And what Tyrone Hayes from the University of California, San Francisco would do is he would do his acknowledgements first. So I'd like to, let me see here. There we go. So before I begin the actual meat of my talk, I'd like to thank both IUSSTF, the WaARI program, and Doherty Water for Food for giving me the opportunity a number of years ago, back in 2017, to invite Dr. Rajiv Singh to come and spend 6 months in my laboratory as a WaARI fellow. He then went down and worked in conjunction with Dr. Shannon Bartelt-Hunt, who we heard from yesterday at the University of Nebraska, and spent 6 months with Shannon. And again, I cannot express my gratitude for having that opportunity. Now, in between Omaha, where the University of Nebraska Omaha, which was my home campus, and the University of Nebraska Lincoln, which was Shannon's home campus, there were a couple of very nice Indian restaurants that Rajiv became very affectionate towards. He really enjoyed their food, and we would go there quite often. Now, the reason I'm telling you that is one of the great opportunities that happens when you're doing international collaboration is you get to sit down with your international colleagues, very often over meals, and talk about science. And one of the conversations that we very frequently had was this: the problem of spatial and temporal variability. And you've seen this in Dr. Weber's talk, Dr. Bell's talk, Dr. Bartelt-Hunt's talk yesterday. I imagine Dr. Snow will talk about this later on today. But if you're starting to look at a large geography in the United States, that could be one of the 50 states within the United States, or a region like the greater Midwest or the Mississippi River floodplain, or in India, if you were looking at something like the Ganges, right? The Ganges watershed, a very massive geography. The problem that we all face is that contaminants in those environments are not only spatially variable, where you can have, as you can see here on this response surface, you have some areas where there may be the darker blue where the contaminant is present, and then some areas, the dark red, where there are hot spots, where there's very hot levels of certain contaminants. It's not just spatially variable, but it's also temporally variable. So as you move from season to season to season, the concentration of the chemicals and even the occurrence of the chemical in the environment changes. Now, in that kind of an environment, it makes risk assessment relative to health, as we are all of us who are watching tonight, we're all painfully aware of this. It makes risk assessment extremely difficult because we're literally trying to hit a moving target as the concentration of contaminant in the environment changes over time and changes over space. And I can remember having many great conversations with Rajiv and Shannon about this very thing. So I find that really, really interesting. So let's just take a step back to yesterday. And one thing that Shannon talked about, and she got a really great response from one of the participants in this workshop, was Shannon talked about the value of using citizen science as a data acquisition tool. And by doing that, what you can do at a relatively low cost is you can really spread out your sampling design and really spread out your overall geography that you can sample. And one of the participants yesterday made the point that this can be really good for identifying hotspots in the environment relatively cheaply. And whoever said that, I'm sorry, I don't remember your name, but you were absolutely right. That's absolutely true. And both Shannon and I and others have done a lot with atrazine, have done a lot with nitrogen and phosphorus and other chemicals, particularly in Nebraska. Back in the days when I lived in Nebraska, we spent a lot of time. We would spend a lot of time here. Now this is a red knot. A red knot is a shorebird, and the only reason I put this slide on here is to remind us all that we're currently living in an environment where the chemical contaminants, like the red knot, and look at this animal's migration from the tip of Tierra del Fuego in South America, all the way up to Hudson Bay and beyond in the very northern latitudes of northern Canada. This animal is global in its migratory patterns. This is also true for many chemicals in our environment, whether we're talking about greenhouse gases in the atmosphere, or whether we're talking about certain types of persistent organic pollutants that can be transported by what's known as the grasshopper effect, where during the summer, they're atmospheric, they move, during the winter, they come back down to land, in the summer, they get up and they move again. So they also migrate. So when we start thinking about the geography of contaminants, it's bigger than Nebraska, it's bigger than Idaho, it's bigger than the United States, it's global. So we're looking at the global community relative to the movement of contaminants, and obviously we're looking at the global community. COVID-19 is a perfect example of this. We're looking at the global community when it comes down to public health, because we are all, as COVID-19 is painfully showing us, we're all interconnected with each other. So this idea of the community writ large, the human community, and the geography in which the human community lives is part and parcel to what we're all interested in. Now, in that environment, what happened in 2017 is that the American Geophysical Union initiated a journal known as GeoHealth. And GeoHealth focuses on exactly what I was just talking about. And it's interesting because if you look at this quote from Environmental Management from 2014, Dr. Bunch said, overall, the integration of watershed management and governance and public health is in its infancy. And I think that's really, in a way, a catchphrase for what we're dealing with in this workshop, what I saw yesterday and what we're seeing today. This interface between large-scale geography and public health, and the way that either contaminants or, as Dr. Bell was talking about, drought or other extreme weather conditions impact the geography and then impact the public health of the people that live there. So I think it's really interesting because I think this is an area where we're all effectively working. Now, what is GeoHealth? And effectively, and I'm going to go through this relatively quickly, but effectively it's this synergy or this Venn diagram of human health, the Earth system, and humans. And where those all intersect is this field of GeoHealth. And again, I think in one way or another, we can put or compartmentalize everything that's been said today and yesterday thus far into this overall construct, which again, I find quite interesting. Now, one other introductory comment that I want to make about GeoHealth came out of this this document that was generated by UNEP, the methodology for integrated environment and health assessment for Latin America and the Caribbean. And what they said is today there's more scientific evidence available than at any other time in history, showing us the different interrelationships between ecosystem integrity, environmental services, and human well-being and the quality of people's lives. So again, geohealth, how we fit into the overall worldview relative to geography, and how humans fit into that, and how that impacts our health. I think that's a really interesting place to inhabit, and I think it really provides fodder for our interaction with WaARI fellows relative to being able to look at what's happening in India and what's happening in the United States, and draw comparisons, draw differences, and have the kind of discussions, again, that Dr. Singh and Dr. Bartaltan and I had numerous times when Rajiv was in our laboratories. Now, I'd like to back up for a second and talk a little bit about water contaminants. And really, water contaminants can run the gamut from plastics and microplastics to microbes to chemicals, all the way to radiation, to climate change. And where I want to focus today is on chemicals. So I'm going to be specifically dealing with anthropogenic chemicals and public health concerns. I apologize if I changed the view on that slide. My apologies. So relative to chemical contaminants, there are a lot of chemical contaminants in the environment, and they run, again, they run the gamut from ions, such as what you would find on the periodic table, to relatively larger organic compounds, all the way up to nanoparticles. Now, the chemicals that I've been interested in now for frankly, the last few decades have been agricultural pesticides. And I'd like to spend the rest of my time today talking about this interface between agricultural pesticides, geography, and human health. Now, one thing is, and if you remember a slide that I showed a few slides earlier, where we were talking about the fact that we're living in a data-rich time in history. Now, this is really interesting because in the United States, there is this website that in which you can acquire data, as it shows here, on the county level for every state in the United States. You can acquire data relative to pesticide use in that county for any given year for more than 470 different pesticides from 1992 to 2017. So there is this enormous wealth of pesticide data that's out there just waiting to be mined, just waiting to be explored. And this is an example. I went into one of these, I went into the database, and they have these maps. This is showing the intensity of glyphosate use per square mile of agricultural land across the entire United States. And not surprisingly, at least to the Americans that are in the audience today, not surprisingly, the Midwest lights up with glyphosate. It's less frequently or less intensively used in the western parts of the country until you get over to California. But there's very clearly a distribution. And again, this really represents data that can be mined. Now, what would we do with that data if we were to mine it? The other piece of information out there that is available to scientists in the United States relative to the kind of work that we're discussing today is that the National Program of Cancer Registries aggregates cancer incidence data, again, across the entire United States. So now we can take potentially this agricultural pesticide load data and interface it with cancer data across, like the red knot, across very enormous geographies. So the migration of this data can be really nationwide. So it's really quite exciting. Now, I wanted to point out a couple of things before I leave this slide. One is, and I believe Dr. Bell talked about this a little bit already earlier this morning, and that's the fact that we've been, myself, the University of Idaho, in conjunction with the University of Nebraska Medical Center and the University of Nebraska-Lincoln, have been interested in pediatric cancer. Part of the interest is because you have a developing fetus within the mother that may be developing right during a time when there can be environmental exposure, and there's a relatively short latency period between that exposure neonatally and the manifestation of the disease, unlike something like Parkinson's, which may take decade after decade to ultimately manifest itself. I also wanted to point out on the slide, the black circle is the state of Nebraska. And again, Dr. Bell talked at length about pediatric cancer in Nebraska and its relationship to watersheds and other things that we've been working with. And I also wanted to just point out relatively where the state of Idaho is. Our levels of pediatric cancer are nowhere near as high as those in Nebraska, but they're still certainly of concern. I wanted to throw this in, this little sidebar that just appeared in that, again, and I think Dr. Weber said this as well, this is a team game. This is a team effort. No one's doing this alone. So again, I'd like to thank my colleagues, Dr. Bartelt Hunt, Dr. Bell, Dr. Weber, and Ellie Rogan, who's kind of the, who has been instrumental in getting us all together and getting us all moving in the same direction in this work. Now, I'd like to real quickly show you some data from the western part of the United States. And one thing that's really interesting, without bringing in the adverse health impact data, is the western United States, not every state uses pesticides in the same way. So on the left-hand side here, you can see the predominance of the 25 most common pesticides used in the United States annually. And you can see that there's a real fundamental shift from one group of states to another. The states on the left-hand side, where there's the large corn yellow columns, the states such as Montana, Wyoming, Utah, Colorado, New Mexico, those states are all herbicide dominated. And over on the map on the right, the herbicide dominated states are those states that are on the right-hand side of the United States, the states that border the Great Plains and border where Nebraska is. In contrast, the states closer to the Pacific Ocean are the states that, if you look again on the left-hand side, the blue bars are fumigant dominated states. They're states where the predominant pesticides used in those states are fumigants. Well, why would there be a geographic shift in the amount of usage of different types of pesticides across the United States? Well, the reason is what's being grown there. In the eastern states, the light pink on the map, the eastern states are predominantly growing grains, and they're predominantly growing animal feed. The states on the western side of the map are predominantly growing fruits and vegetables, so they're growing human food. So there's a very different exposure regime, and there's also a very different land use, and there's also a very different trajectory for where those pesticides are going. So the geography actually tells us a tremendous amount about what's happening in the country. Now, to move forward relative to that, if we look at these 11 states in the Western United States, we also see, interestingly enough, and here I tried to color code the name of the state relative to where it falls on the map. You can see in the Western states, Washington, Oregon, California, Idaho. Unfortunately, Idaho is relatively high, my home state now. These states in the western part of the United States use a much larger degree or are much more predominant upon fumigants than they are herbicides. And they're also the states that have higher incidences of pediatric cancer. Now, looking at the data that I just showed you, one problem that could be associated with that data is a problem of scale. This is also known as the modifiable aerial unit problem. And that's the fact that if you look at a large scale geography, like the Western United States, and you look at it on one geographic scale, looking at it from state to state, you may get one result. But if you zoom in and look at the same geography, but instead of looking at it as 11 states, you look at it as the 459 counties that are in those 11 states, and that's what's shown on the right-hand map here, you may get a very different result. And if that's the case, it's really hard to make any inference because it's not really a strong relationship between the geography and an adverse impact. It's more how you're looking at it, what level of resolution you're using in your analysis. So this is a very real concern. Now, I'm not going to go into any detail on this, but here's on a county level, on the left-hand side is the observed pediatric cancer incidence in all the counties in the Western United States. Now, if we model that using pesticide data, and I'm not going to go into the modeling for the sake of time today, but if we model it using the pesticide data that I've already talked about, these kilograms per county per year, look how close these models are. They're actually very statistically very close to each other. So this is a really satisfying result because it's telling us that the geographic unit that we use to discriminate our analysis is not the driver here. The driver is something probably deeper, and there's a deeper relationship between pesticides and adverse health outcome. Now, before I stop for the night, I wanted to talk about another study that I was involved in. And this is a study that came out of the country of Ecuador in South America. And the issue that I wanted to touch upon is that it's easy, and I know that India falls much more closely to the United States, where you have a lot of strong data, you have a lot of strong computational skills. But if you go to some countries, particularly in Latin America, Africa, parts of Asia, that capacity doesn't exist. They still have the same problems, but they're working on a different level relative to the capacity of the data that they have. So I wanted to talk about what do you do in countries where maybe you don't have the magnitude of data that we're fortunate enough to have in the United States. Now, in Ecuador, as you can see here, the study that we did was focused on this Loja region of Ecuador, which is in the red circle, far to the south in the country. And this is effectively their bread basket. This is where they grow their grains. And the three principal grains that they grow are corn, rice, and soybeans. The large map shown here is showing the areas of concentration of those three different crops: corn, rice, soybeans. And what my Ecuadorian colleagues wanted to do is they wanted to develop an environmental burden index, something to be able to ask the question of, do the communities or do the cantons where the people live, are those communities susceptible to pesticide exposure, and is one crop increasing that risk relative to other crops? So in order to address that, the pesticide exposure index took into account the amount of pesticide sold within each of the neighborhoods, each one of these cantons, the toxicity of each pesticide that was sold, the number of crop rotations per year, the population density of the populations that lived in those environments, as well as other factors. And again, I'm not going to go into too much detail here, but rather than just simply to say, look at these maps, because what they're doing on a regional scale in Ecuador, even without the plethora of data that we have in the United States, is they're developing an index that they can use with these artisanal small acreage farmers to be able to look at parts of this community and say, these cantons might really have issues relative to risk of pesticides, where these other cantons, because of the crop, because of where they are, have a much lower risk. So again, it's geohealth, just done with a different set of tools and on a different kind of geographic scale. Now, I want to leave you with a couple of just kind of informational pieces of information. One of which is, if anything that I've told you today piques your interest, take down this website. We, including Dr. Bell, Dr. Bartelt Hunt, Dr. Weber, we were involved in a chemical toxicants in water workshop that happened last June 14 to 16. All of those, the entire workshop was taped. You are welcome to go to this website. You can pull down the video of the workshop, and you can see what a number of different people have done. You can also listen to the chat lines of what individuals were saying afterwards. It's really fascinating, and I invite you to do that if that's something that you would be interested in. Finally, the take-home message, I think, and again, others have commented on this already. The take-home message here, there are a number. First of all, community engaged research and action really needs to be done in this environment. We're not just simply doing research on communities, which was a real problem in the United States, because historically there have been really egregious examples of where that has occurred, and communities were being used as study animals or subjects, as opposed to actually being participants in science. But rather, we're doing work with and for communities. This gets back to what Dr. Bartelt Hunt was talking about yesterday relative to citizen science and community-based participatory research. And then secondly, training and funding the next generation of scientists. And that certainly is where the WaARI program comes in. And changing the idea and the graduate student model of how this is done. And again, the WaARI program is an exemplary example of that. So with that, I'll stop, and I'll be happy to take any questions.
Thank you, sir. Thanks for a wonderful lecture on the importance of global knowledge, or necessity of global knowledge on this kind of contaminant transport studies, and especially highlighting on the societal importance of health issues. Now I have opened the forum for questions. The attendees can post your questions in the chat box or question answer box. Okay, we still receive the questions, and we will get the response from Professor Alan later. Now I move on to the next keynote lecture. Thank you, sir. Thank you for your nice presentation. Sir, 1 second, 1 minute. We have a question from Dr. Soumik. Dr. Soumik, can you elaborate the question? Dr. Soumik is asking that.
That is it. I see his question, and I'll I'll paraphrase it for everyone. Professor Goswami said, I have a question. It's a wonderful lecture encompassing the pesticides and cancer, but how can we replace pesticides looking at the population that we have to feed? That's a great question. And in the United States in the 1960s, when DDT was prevalently used, not for mosquito control, but on agricultural crops, a seminal book was written by Rachel Carson called Silent Spring. And she argued the point that there was an overemphasis on the use of DDT as an agricultural pesticide. And the reason I'm bringing that up is all of my American colleagues, and perhaps some of you Indian colleagues as well, but certainly all of my American colleagues know very, very well who Rachel Carson is and the book Silent Spring. However, what many people don't recall is she never called for a ban on pesticides. What she said was prudent use and minimization of adverse impact. And again, if you think back to the slide that I showed in Ecuador, of course they're going to need pesticides in Ecuador because Ecuador is a tropical country. There's a large diversity of pest insects, and a pest can wipe out an artisanal farmer in a day, right? And completely devastate the economic future of that family. So of course pesticides are necessary. However, we need to be sober about weighing the relative risk and benefit of pesticide use relative to adverse health outcome. And no one's arguing to not use pesticides, but what people can argue is let's use pesticides in a prudent fashion so they're not impacting public health and developing things such as carcinogenicity. But that is a great question. So thank you very much for that.
Thank you, sir. Now, let's move on to the next keynote lecture. The next keynote lecture is by Professor Daniel Snow. Professor Daniel Snow is presently working as a research associate professor in University of Nebraska. Professor Snow has been an integral part of University of Nebraska's Lincoln Water Science Laboratory since 1990. At present, he oversees all aspects of water science laboratory operations and uses analytical chemistry to help understand how water becomes contaminated and what can we do to prevent it. Professor Snow is also directly involved in the development of new methods to measure and use stable isotopes as tracers to study environmental problems and processes. I welcome you, sir. You can take over the presentation.
Oh, thank you very much for that introduction. Very happy to be here and be presenting this evening. And I thought all of the presentations, it's morning there, but it's evening here, have been excellent. So I'm happy to provide my input on this particularly important topic. Let's see if I can share my screen. Well, it started. Might be locked up. Can you see my slides?
No, sir. You have a message screen share.
Maybe can share it again.
Yeah, I'm going to try to share again.
Yes, we can see your screen.
How's that?
Yeah, it's visible.
All right. It's not sharing the right screen though. Let's try this one more time. How's that?
Yes, sir. Perfect.
Great. So I'd just like to spend the next 20 minutes or so talking about pushing the limits of laboratory analysis in developing countries. I think it's been pretty evident from the earlier presentation that pollution is a serious issue, and especially serious in developing countries like India and China. The study was published in 2019 by the Global Alliance on Health and Pollution, indicating that pollution likely kills around 8.3 million people annually. What's important to note is that we're linking a biological effect from the occurrence of pollutions, whether they be chemical or microbial, to a biological effect. So I think we need to take a step back and learn about how we do environmental testing and why it is especially important to develop these technologies and build capacity in developing countries. I think the talk that Alan gave just before this was a great lead into this particular topic, talking about agrochemicals, the use of those agrochemicals and the human health and those ecological health effects. It's important to realize that just a very, very, very small amount of any of those chemicals ends up in the environment after it's lost, typically on the order of 1/10,000% might end up off of a field, either through air or water pollution. And it's a continuous exposure from that very, very small quantity of a chemical that might lead to a biological effect, an adverse health effect to human beings and to organisms. And that's where environmental laboratory methods really come into play. We have to be able to accurately and precisely measure concentrations of a wide variety of chemicals in a very complex matrix. And if we're not able to do that reliably, then we're really not doing justice to the science. One of the most important questions to ask if you're doing environmental laboratory analysis or testing is, what do you want to measure and why? For example, are there chemicals from runoff that could affect human health or organisms in a river? Are these agrochemicals that are used in this irrigated field, do they have an effect on a fish in a river that may be miles or many, many kilometers away from that irrigated field? And what I'd like you to take home is this particularly important question. What are the consequences of improper measurement? In other words, what happens if we don't actually detect or measure the chemical that was present that maybe was causing harm, or we incorrectly or improperly measure a chemical or compound that wasn't present? So we're really, in my field, having confidence in our measurements and reliability of those chemical measurements is extremely important. So what is unique to environmental testing, environmental methods? Well, typically the detection limits are in the parts per billion to parts per trillion, and sometimes even parts per quadrillion range. There's a wide variety of sample types from air, water, soils, to biosolids tissues. Really, air and water is pretty easy to measure low concentrations in. When you get to soils and biosolids or any kind of biological tissue, they become extremely complex, and it's very difficult to separate what you want to measure from the matrix, the material that the contaminant is contained in. We have to decide a particular type of sample preparation, or the chemicals that we want to measure. Polar, that is, are they water soluble, or are they non-polar, are they very hydrophobic? there's always a high potential for interferences, and how do those interferences interact with what we want to measure? And something that we really need to consider often is what are the instrumental requirements? We almost always need to have high sensitivity and high specificity, but often the laboratories that we work in or that we have access to don't necessarily have the capability for those two requirements. I'm going to spend a couple of slides talking about detection limits. That was in the title of this presentation. Well, analytical chemistry is really just a separation, identification, and quantification of chemical components in natural and artificial materials. So what role does detection play in our environmental testing? In other words, how low can we go and reliably detect and quantify a compound in an environmental sample? And it really boils down to statistics. We're often faced with a question of how low we can go with a particular method that we develop in the laboratory. And we have to be able to demonstrate that we can measure that reproducibly at a level that is almost infinitesimally small. And statistically, we want to show that we can minimize the potential for false positives and false negatives. In other words, we want to have that potential statistically proven to be less than 1% at, say, a method detection limit in a clean matrix. So that's what we really rely on, a statistical test to show how low we can go in our environmental methods. Unfortunately, and especially if you go into the literature, the regulatory literature, there are so many ways to express detection limits. We have instrument detection limits. In other words, what a particular instrument can distinguish from background. Typically, that's used just for selling or buying an instrument. Method detection limits is something that we use quite a bit in environmental testing. In other words, we have a particular way that we can statistically measure what our ability is to quantify and detect chemicals. There are comparable limits to method detection limits, such as a limited detection, a lower limit of detection. And most recently, there's been this method quantitation limit where we can actually prove that we can reliably quantify a chemical and environmental matrix. And then finally, there are reporting limits. Typically, this is set above any method detection limit or quantification limit, where we are reporting lab results to customers from a testing laboratory. Those last two are really have come about because of regulatory requirements and the issues that come about when you report detection when a compound is not actually present in that sample. So we end up being very conservative from a regulatory standpoint when we're reporting detections and quantities of contaminants in environmental samples. If you go to the scientific literature, we tend to go more towards the less conservative lower limit of detection or limit of detection or method detection limit, which is really just where we can reliably detect the compound, but not necessarily quantify it within a specified level of confidence. So often, especially if you're a student, you're not really sure what tools are available to measure the chemicals that you want to investigate, what kind of data is needed as you're formulating a research plan. You have to make sometimes some tough decisions and things that you don't really know in advance. And then something that professors, especially in the United States, we always worry about is how much does it cost? Typically, the more sensitive a method, the more it costs. I run a research core facility at the university, and we spend an awful lot of time trying to minimize method costs so that we can allow students and researchers access to as highly sensitive and high confidence methods as possible. And in my mind, the most important result of all of these efforts is that we provide a set of data with a level of confidence that we can quantify statistically. If you don't work in an environmental analytical laboratory, you don't realize how many choices that we make. One choice is what type of sample preparation to use. We have lots of choices, whether to filter or not to filter. Many contaminants are absorbed to sediments. What do you do if you want to measure the dissolved fraction? Well, you need to filter it, but you might be filtering out what you want to measure. Liquid extraction is something that's there's quite a few methods in the literature on, but it's very time consuming and labor intensive. Solid phase extraction, for the most part, has taken over for most environmental methods. It's faster and uses much less hazardous solvent. There are many phases available, and what we tend to like in environmental testing laboratories is they can be automated. Well, even with solid phase extraction, we have lots of choices on how we go about extracting the compounds that we want to measure. Ideally, we want to have an extraction method that's both selective, that is, it can accurately concentrate and remove the compounds that we want to measure from that sample, and we want it to be able to remove it reproducibly and quantitative. In the early days, we were pretty much using a bonded silica or C18 solid phase extraction, but within the last 2 decades, it's been pretty evident that most environmental testing methods use some sort of a polymeric solid phase extraction because it tends to be pretty reproducible. It works for a fairly wide range of compounds, and it's something that can be reproduced in other laboratories. The newer classes of solid phase extraction may include molecular imprint polymers, where the retention is based on molecular recognition, or immunosorbents, which use bound antibodies for selective retention of contaminants. Those newer phases are really useful if you have a complex matrix and you have a polymer or a specialized phase that's specific to the class of compounds that you want to measure, but they're really only designed for certain groups of compounds, and they can't do multi-residue methods the way that we could with a polymeric or even a bonded silica solid phase extraction. So back to that method detection limit, really it's been well defined in the regulatory literature in the United States. What an MDL is, it's the minimum concentration that can be measured and reported with a 99% confidence level that analyte concentration is greater than 0. Okay, and here's an example of how we would go about doing that in an environmental laboratory. We would spike a known quantity and amount into a reagent water, and then run that set of samples, 8 spike samples, through the entire process, and then analyze those as unknowns, quantify our analyte. In this case, it's ethane sulfonic acid, a metabolite of metolachlor, calculate the standard deviation, and from that we can determine the method detection limit. The important part of this is that we're including all steps that we would use in processing a sample from the field. So this allows us to get a good handle on the variability in the recovery and our sensitivity for our environmental testing method. And here's an example of a method that we developed early on at the Water Sciences Laboratory for trace analysis of 18 different herbicides in Nebraska that we use in Nebraska, including atrazine and 2 atrazine metabolites, DEA and DIA. From a half-liter sample run through a C18 bonded silica solid phase extraction cartridge, we could elute that and concentrate that fairly quickly by evaporation, analyze 1 microliter by gas chromatography mass spectrometry, and that gave us a fantastic 5,000 times concentration factor. So it was amazing. This is something that we've had available to us for quite a while now, but at the time it was amazing how quickly and easily we could selectively concentrate these pesticides using solid phase extraction. What was even more amazing to me was how much confidence using isotope labeled internal standards gave us for this particular method. We contracted with, I believe it was with CDN Isotopes to, actually it was Cambridge Isotope Labs to synthesize atrazine, C13 labeled atrazine with C18 DFLND isopropyl atrazine. And if you can see in that yellow diagram, we had very nice separation between atrazine and the atrazine metabolites, and those stable isotope analogs of the parent compound and the metabolites gave us a very high confidence in detecting all three of those compounds in this particular method. So we had a great method and we analyzed a lot of samples, but it seemed like we might be missing something just by using this one method. And it turned out that we were. Our herbicide screen was great for parent compounds of the triazine herbicides and the chlorocetamide herbicides, as well as the metabolites of atrazine. But it didn't allow us to look at these glutathione conjugates and other metabolites of the chlorocetamide herbicides. And it turns out these metabolites are roughly 100 times more soluble than the parent compounds. So this really led us into the investigations of the polar compounds that have become more or less emerging contaminants these days. Looking at these highly soluble pesticide metabolites with new instrumentation and new extraction methods, it allowed us to measure these in samples in the environment. And we actually find that these metabolites are much more persistent and much more widely prevalent in the environment than the parent compounds. So what are the best ways to measure these emerging contaminants or these polar organic contaminants? Well, LC tandem mass spectrometry, it's still the most sensitive instrumental method available for these highly water-soluble organic compounds. We have quite a few choices in terms of how we ionize compounds by LC-MS. And we also have lots of choices in the mass spectrometry that we use to analyze these molecules. The instrument on the left is an ion trap instrument. That was the first LC-MS system that we had at the Water Sciences Laboratory. The one on the right is our micromass Quattro Micro triple quad. We purchased that in 2002 and it's still running. It's a very rugged, robust triple quad. It has analyzed thousands of samples for the research that we do. These polar organic contaminants, the emerging contaminants is organic wastewater contaminants are water soluble and difficult to separate from water and from interferences. The instrumentation is much more complex and expensive than for non-polar compounds like our parent pesticides. This is our Water's Zevo TQS. Was just installed in 2020. The retail price for this is about $550,000. So they're very expensive. And this is one of the reasons that the methods that are based on LC-MS are typically among the most expensive that we use in environmental testing. Well, this slide shows some of the trade-offs in LC-MS. The top figure, the LC with a single quadrupole. The column on the right shows the identification points. In other words, the confidence that we get from using that particular LC-MS system. Early emerging contaminants methods used an LC with a MS with a single quadrupole. They were not very sensitive and probably gave us the lowest confidence in detection. The next step up from that was a LC ion trap, and then the LC triple quadrupole. The triple quadrupole is still today probably the most sensitive and selective available for targeted compound analysis. But many laboratories are more interested in doing a higher resolution analysis using a time of flight or QTOF or even an Orbitrap. Those instruments are great for doing unknown identification, and they can be used for quantitative methods. But typically the high resolution instruments are not as sensitive as the triple quadrupole. So that's one of the trade-offs that we make when we use high resolution mass spectrometry, is that while we can get accurate mass measurements, we actually have less signal in detecting the compounds. And that becomes very important in an environmental sample. So this just is a slide on method development. A particular method that we developed using an ion trap LCMS. We were measuring high explosive residues in groundwater that was contaminated with munitions residue. It turned out it was not a simple task. We had to set up the mobile phase chemistry so we could make adduct ions. RDX was the one that we were most interested in. And it turned out we could only detect RDX using LCMS if we made a formate adduct. And then we had to tune the needle voltage and source conditions so we could optimize sensitivity. That's what the graph on the right-hand side shows is, well, how can we get all of these to be as sensitive as possible without losing sensitivity on another compound? It actually turns out it's quite difficult to set up your mobile phase chemistry and the source chemistry, and then to get the detection conditions optimized when you do multi-residue methods. And we almost always do multi-residue methods in environmental testing. So these emerging microcontaminants, as many people call them, new is in quotes because they're not really new, because they've always been present. That we just now have the ability to measure these compounds that include pharmaceuticals, steroid hormones, personal care products, plasticizers, flame retardants, metabolites. These are really just chemicals of concern that come about because we have new methods for measuring them. We have new mass spectrometry methods, and really the whole point of this is to understand where they occur, where these chemicals occur, and whether or not they pose any risks either to aquatic life or to human health with respect to water quality. And that's what this timeline shows is really the widespread availability of the technology to measure these contaminants has really only happened in the last 20 years. So that's barely a blip on the spectrum for environmental laboratory testing. Of course, we have multiple inputs of these emerging contaminants into the environment. Drugs are used in both livestock production, veterinary medicine, and for human medicine. Once they're used, they can be excreted either partially or completely metabolized into the environment through farm effluents, manure application, or municipal sewage. And then from there, they end up in our river, streams, and lake, and in some cases, even groundwater. So we don't really know, we've only had the capability to measure these chemicals within the last 20 years, but we've been discharging them into the environment for probably the past 50 to 100 years. What are the consequences of all this, all these compounds going into the environment? Well, one thing that I think is very useful, and something that I've worked on with my colleague, Alan Kellogg, is to try to connect the occurrence of these chemicals to biological effects. This is a study that one of his PhD students, Marlo Sellen, did for her work in Nebraska, where we actually quantified steroid hormones in municipal wastewater effluent, and we exposed fish to that same effluent. And we were able to show that the effluent that had the highest steroid levels also had fish that had the highest differential gene expression. Basically, there was a biological effect that we could connect to the occurrence of these compounds. And this is the type of environmental research that I think is important to do, not just in the United States, but everywhere. This is an example of a paper that Paramita Chakraborty just published this year in Water Research, where she and her students looked at the occurrence of plasticizers, BPA, steroid hormones, and caffeine in wastewater effluent in the Bay of Bengal. And we were able to connect the occurrence of these particular chemicals measured through both GCMS and LCMS methods to a particular biological effect using an e-screen assay with a colleague from the U.S. Department of Agriculture, Agricultural Research Service. So this is just another example of how it's possible to do both a chemical measurement and connect it to a potential biological effect and better assess the risk of the occurrence of these compounds. So in summary, really pushing the limits of environmental lab analysis in developing countries is the same as it is in a developed country. This advanced detection technology helps us provide good defensible data, but it requires a thorough understanding of the limits, the properties of the chemicals that we want to measure, the separation and the detection chemistry, as well as the limits of the technology. And I think it's extremely important to connect detection, that is not just report the concentrations, but connect that detection and occurrence with some biological effects, either through screening assays or risk assessment. It really helps reinforce confidence in our results and demonstrates the importance of these measurements. And then finally, I just want to acknowledge all the people that have been involved in this research. It's been supported by a number of different sources. I'm not going to go through everything here, but it's been a journey and adventure being able to do some of these projects, not just in Nebraska and the United States, but around the world. And with that, I'll be happy to take any questions.
Thank you, sir. Thank you for a wonderful lecture. Actually, this will really help the attendee students, research scholars to know the. So we are not able to hear. I think I'm muted. So unmute yourself. Okay, so actually this lecture enlightens the present research scholars to know the importance of analysis and how to know what they are doing and what they can do with the instruments that they have and actually what they should look for. So I open the forum for questions. We can formulate 1 or 2 questions. The participants can post your questions and question and answer pods. Thank you, sir. We don't have any questions, so thank you. Thanks for the nice presentations. Now I hand over this session to Ms. Anusha. Ms. Anusha?
Yes, sir.
Yeah, please. Thank you. You can take over the session from here.
Yes. So I would like to hand over the session to the session chair, Ms. Sija Arun. So Ms. Sija Arun is an associate professor of the Department of Civil Engineering and Environmental Engineering in SRMIST. She has secured National Gratitude Scholarship GATE in the year 1997 from the Government of India. Her area of research interest includes occurrence of antibiotics in water resources and wastewater treatment plants. I hand it over to Ms. Sija Arun.
Thank you, Anusha. Good morning, all. I welcome you all for the invited talk session of the second day of this workshop. I express my thanks to the conveners for inviting me as a session chair. Move on to the first talk. The first talk is by Dr. Arindam Malekar. Dr. Arindam Malekar is a postdoctoral research associate at the University of Nebraska East Lincoln's campus's Water Science Laboratory. His current research focuses on the creation and migration of natural nanoparticles in the environment. In that context, he is trying to figure out how irrigated water interacts with the root zone soil chemistry. I welcome you, sir, to deliver this talk.
Thank you, Dr. Arun, for this nice introduction. I'll just try to share my screen first. Can you see my screen?
Yes, sir.
Okay. Yeah, thank you for the invitation and this warm introduction. I would really like to thank the convener of this seminar, Professor Snow and Professor Chakraborty, for arranging this and giving me an opportunity to present our ongoing research here. So through this presentation on identifying anoxic microsite at root zone of unsaturated soil, I would like to share some of our recent investigation on critical aspect of root zone processes on plant uptake and mobilization of arsenic and uranium. I would like to thank my previous speakers, Professor Bell, Professor Weber, Professor Kellogg, and Professor Snow for setting the precedent about this contaminants which impact public health and giving a global and regional and localized perspective towards this water quality aspect of it. Now, through this talk, I'll try to scale it down a little bit or zoom into a little bit to more field scale study, which was recently concluded. But before I begin, I would like to thank all of the funding agencies, collaborators, which include University of Nebraska-Lincoln, Nebraska Water Center, Doherty Water for Food Global Institute, Nebraska Center for Medicine, for Materials and Nanosciences, USDA Multi-State Hedge Funds, in Indo-US Science and Technology Forum, IUSSTF. I'm really grateful for the funding that I received during my PhD, as I was one of the first WaARISH interns to get this internship and work here with Professor Snow and Professor Yusang Lee, and all of the collaborators who made this work possible, which include Professor Chitranjan Ray, Professor Snow, Dr. Michael Kaiser, Dr. Harkamal Walia, Dr. Bijesh Mahajan, Dr. Darren Rudnick, Dr. Bonajarani Panda, who was again a WaARISH intern, Dr. Anurag Kumar was a WaARISH intern who worked on this project back in 2017, Jordan Seals, and all of the staff from Water Sciences Lab, Nebraska Water Center, and greenhouse facilities at the University of Nebraska-Lincoln, and NCMN staff. So, as I said, that I'll be zooming in a little bit from global, regional, localized scale to more field scale. So that is where the term anoxic microsites come into play. So what do we mean by anoxic? So anoxic is basically a site which is devoid of oxygen, or the oxygen levels are really low. So it is something which has low amount of oxygen. And if I talk about microsites, as I said, we are zooming in to a micro scale level, to a very small scale, which we are trying to identify these small hotspots within the bulk unsaturated soil in a root zone system of intermittently irrigated field system, or basically sprinkler field system, which is the primary way of irrigation in the United States. Basically, to put it in numbers, 55% of the irrigated area in the United States use sprinkler method to irrigate. So now, why do we want to understand the anoxic microsites at the root zone, and what is the main cause of the problem that we want to realize, and how it affects the public health system? So let's look at the problem first, the big problem. So when you look at the introduction of geogenic contaminants, such as arsenic and uranium, we did talk a little bit about uranium from the talk that Professor Weber presented, that how it can impact human health. Arsenic is quite a bit of a toxic element and causes quite a few different kinds of cancers, and it is one of the primary reason of cancers in Indo-Gangetic plain of India. So there are two ways these geogenic contaminants can get into human. One is by drinking contaminated water, which contains these contaminants, which can be, we can get rid of it pretty easily by doing some filtration method. The other route is, let's say if we are using this water, contaminated water for irrigation. Further, if the soil where we're irrigating or we're growing our crops has elevated amount of arsenic and uranium, what can happen is that root zone processes can mobilize these contaminants and can be taken up by crops. These crops can be used to feed animals, and these contaminants can end up in the animal product like milk or meat, or we can directly eat the grain or the fruit or the vegetable that is being produced using this high elevated content arsenic uranium soil or elevated amount of arsenic uranium present in the groundwater, which is being used for irrigation. Now, what is the common entry point for the crops? It's the roots. This is through which it not only takes essential nutrients, but also non-essential trace elements like arsenic and uranium. So one key thing that I realized from my previous arsenic research during my PhD and postdoc, that water alone is not the entry point of arsenic to human body. Food intake can be a significant contributor, especially in India and adjacent Bangladesh. Rice contaminated with arsenic is one of the major way of chronic exposure to arsenic. So, which has been linked to cancers. So this made me interested to understand the unique situation in Nebraska, where soils contain elevated levels of trace elements, and the irrigation water that is used also contain elevated level of these trace contaminants. We are talking about arsenic and uranium here. Now the problem is, as I said, in the root zone, where we are using sprinkler irrigation, the soil is predominantly unsaturated. And when I talk about arsenic and uranium, they are expected to behave differently in saturated condition. As Professor Weber mentioned, that uranium is more mobile in oxidized form, whereas arsenic is more mobile in its reduced form. So in an unsaturated system, where there's a lot of oxygen per se, we should have more oxidizing conditions, so uranium should be more mobile. Arsenic, not that much, because it will be oxidized, which is known to be less mobile compared to the reduced form. But the thing is, plant devises methods which are limited at the root zone for different uptake mechanism, such as sequestration or complexation. And as I mentioned, root is the main access point. So understanding of the root zone processes is key, as the primary uptake will be via the roots. And anything that is not taken up by the root here will either end up as a surface runoff to the surface water sources, or can go down to the vadose zone, or ultimately reach the groundwater below. So therefore, it is clearly of utmost importance to understand what is happening in the root zone and how arsenic and uranium, if present in elevated levels, are behaving in the root zone. So this is what we will be trying to navigate today through this talk. So let's say why we were so much interested about arsenic and uranium. So the slides that Professor Weber presented was clear that uranium is of high occurrence in the groundwater of Nebraska. Similarly, if we look at the soil of Nebraska, especially where our field study was created or field study was carried out, is known to have high amount of uranium and also measurable quantity of arsenic. So this is basically a research and extension center of the University of Nebraska-Lincoln, located in the western part of Nebraska. This is the state of Nebraska, in the very western part of Nebraska, and is known as Scotts Bluff, Nebraska. So what I'll be showing here is a combined data from the 3-year study. As shown before, there are many areas of the US with elevated concentration naturally occurring geogenic trace elements such as uranium and arsenic. And also, as I mentioned, that the groundwater also has high concentrations of arsenic and uranium. And this is basically the source of irrigation water too. So what we did was we set up a field experiment which was randomly distributed throughout the area, which was primarily irrigated through pivot irrigation or sprinkler irrigation. And we installed these soil solution access tubes. So basically these are installed at a particular depth. In our case, it was at 90 centimeter depth, where you can put vacuum and your soil pore water, that is the water that is infiltrating after irrigation or rain event, will be sucked in due to the vacuum. And then one can just collect it after using a syringe to bring the water out. But generally it takes around 24 hours to get a measurable amount of water to do different analytical measurements. Not only that, what we did in that field site is we studied 2 different crop types, corn and soybean. The reason 2 different crop types were investigated because they differ in the way the root behave. And I'll talk about it elaborately later. So we not only collected pore water samples periodically, we also collected soil cores from the beginning of the seeding to the harvest point, almost 30 days apart. Along with that, we also collected plant specimen whenever there was plant specimen, and we divided the plant to root, shoot, and grains whenever grain was possible. So at both the cropping system location, we collected in total of 24 soil cores, and we measured in situ reduced iron and nitrite wherever possible. So first, let's look at the soil variability of the top 90 centimeter. So again, the data that is being presented here is 3 years worth of data from 2017 to 2018 and 2019, where soil samples were collected for the top 90 centimeter each month. This is the average value with the standard deviation. And in the x-axis, we have each month of collection, beginning from the seeding to the harvest period. The left y-axis talks about the trace element concentration, which is of interest right now, is arsenic and uranium. And on the right y-axis, it is the extractable iron or dithionate citrate bicarbonate extractable iron, which primarily focuses on leachable iron or easily accessible iron to crops, again in microgram per gram, but that has to be multiplied with 1,000. So what we see is that, so one thing that we need to consider is that weekly irrigation, generally in the western part of Nebraska, starts in the month of June and it goes through mid-July, and sometimes even it can go to early August. So what we did see is that we noticed that the highest labile iron concentration measured as dithionate citrate extraction or DCB extractable iron was highest in the soil when irrigation started and started falling back as irrigation ended and went to the baseline value. Again, this is 3 years worth of data. So this is the square is the iron. However, arsenic and uranium, this is again total arsenic and uranium in the soil, followed the opposite trend. What we also noted that your arsenic and uranium concentration in soil decrease when labile iron is highest. The trend was observed irrespective of crop type. So be it soybean or corn or maize, it was observed throughout. So that this is the combined data. And this trend was observed and total iron in the soil. But interestingly, what was thing to see was the total iron, when you measure total iron, not the extractable iron, remained constant throughout the study period for the 3 years of study. So one thing that I would really want to spell out here is that the thing that Professor Kellogg mentioned in his presentation about the spatio-temporal variation and the heterogeneity of soil adds to that complexity. That was one of the reasons why we continued with this experiment for 3 years to understand how temporal and spatial variation can impact these results. And what we found was that this doesn't really vary much. Even we did do this experiment in another field site, which I'm not showing here, we also did find the same result. So irrespective of the location and the crop type or the root zone system, we found that the irrigation water has an impact on the extractable iron. Now let's look at the pore water concentration. So the pore water, again, we use those soil solution suction tubes. We put vacuum, pore water was collected, and we started getting pore water right during when the irrigation started, which is starting in the mid-June. Triplicate pore water samples were collected from each field site, and the averages over the 3-year period is presented here again, along with the standard deviation. So one thing that was very surprising was finding so much iron. So if you look at here, the iron is really high. in the pore water, even while this is an unsaturated and predominantly aerobic system. So as I mentioned, in predominantly aerobic system, there will be high amount of oxygen, and iron will be in the oxidized form. Similarly, uranium will also be in the oxidized form. And generally, if iron is in the oxidized form, it is supposed to be in a mineralized or insoluble form and shouldn't come so easily in the pore water. And specifically, we were filtering the pore water in situ before measuring iron. So changes in pore water iron appear to correlate to rather wide variations of arsenic and uranium, although uranium was negatively correlating with it over the growing season. We have also, what we have shown here is the total dissolved iron. What I mean by that is that we used 0.45 micron pore filter, to collect the samples there. Interestingly, as irrigation ended, we started to see a sharp decline in the amount of iron in the pore water at the very end of the experiment, and this was coherent throughout the 3-year study. So that was another setting to see that means this seasonal variation was not just 1-year data; it was coherently happening every year after year. So now the question is, what happened to the trace elements that we did see in the pore water? Of course, we did see quite a measurable quantity of arsenic, which was surprising because unsaturated system will be oxidized, so arsenic will be oxidized and should be predominantly less mobile compared to reduced arsenic. What we found was, I'm just showing corn here or maize here, is that arsenic is definitely taken up by the crop. And it is even reaching the levels which is above the FDA's recommended level of 100 ppb. So it is going to close to 0.01 microgram per gram in the grain. Definitely the highest amount was observed in the roots, followed by the shoot and the grain. And as we moved from August to September, that is the end of harvest period, we started to see more amount of arsenic ending up into the grain compared to the initial state. Similarly, uranium was also highest in the root compared to shoot and the root, but still there was measurable quantity of uranium ending up in the final produce. So clearly one thing was very much specific, that plant rhizosphere chemistry changes during irrigation, and labile iron transformation, which was found to be varying in the soil and the pore water, appears to mobilize trace elements. And the problem is, in a field condition, you will have so different form of iron that really identifying the iron transformation is going to be a hard task. So that is where it led to a very controlled greenhouse experiment, where we try to understand the impact of iron minerals within a controlled greenhouse experiment, and we try to replicate the trends that we observed in the field. So we wanted to see if we are seeing the same trend in a controlled environment, where we can really locate all the missing iron pieces easily. And that led to this greenhouse study, which was published last year in Science and Technology, and where again we had a WaARISH intern contribute quite a bit in the study. So the greenhouse study, we observed indication of iron nanominerals, key role in nutrient availability and trace element mobilization. The form and bioavailability of the redox-sensitive elements such as arsenic and uranium was likely affected by iron mineral transformation, especially within the anoxic microsites. So although the entire soil was unsaturated and oxic, there were pockets, small locations which were most likely anoxic and creating this reducing condition. But we had to identify that. Is it really happening? And we proposed that the cycle of waiting during irrigation creates this anoxic microsites where iron transformation occurs. Now, one key thing that we did realize from the field experiment in Scottsbluff, Nebraska, is that ferrihydrite is one of the ubiquitous naturally occurring nanomineral of iron. And that was what was realized from the soil samples over there in Scottsbluff. So we thought that might be the critical mineral that is well known to co-precipitate or harbor arsenic and uranium within the mineral site, which might be affecting this arsenic uranium mobilization. So realizing the critical role of nano iron in field studies soils, we look to quantify them in controlled greenhouse experiment. And to do that, we had to really control everything. So we had to focus on understanding the intermittent irrigation effect on trace element mobilization at the root zone, and then characterize the root zone mechanisms and link them with the redox processes where iron is the key aspect. So in the greenhouse experiment also, we did look into two crops, soybean and corn. Now I want to really elaborate on the root zone system of soybean and corn. So soybean is known to take up iron by reducing iron 3+ to iron 2+, which is more soluble and more mobile and easily can be taken up easily. Whereas corn roots generally would chelate the iron 3. It will not reduce iron 3, but it will chelate or make a complex of iron 3 and then take up iron, because iron is an essential mineral, essential element required for both these crop types. Now, to really specify and understand iron mineral, we had to make sure that the iron source that we are supplying is the only iron source available to the crops. So what we did, we caught some very specialized sand. And the reason we use such high amount of sand, almost 70% to create our soil, is that we wanted to make the system more porous. We had to provide a binding form of clay through kaolite and combine these two together. And the iron amount was negligible. We did supply cellulose as the carbon source. And along with that, we added 0.5% weight by weight ferrihydride, which was co-precipitated with arsenic and uranium. So we prepared that in the lab. And as I said, ferrihydride was found to be the ubiquitous nanomineral present in the field site. So we thought that this will replicate the condition where iron source is supplied entirely from ferrihydride, and based on the root zone process where soybean will reduce ferrihydride, we should see reduced iron, and corn will chelate. So we do not see reduced iron, but we should see mobilization of arsenic and uranium, because chelation will lead to this. So this was randomly distributed, and what we did, we collected pore water, and in here the pore water collection was much easier. We could collect within 30 minutes. So that gave us enough time to really quantify iron 2+. So we collected pore water 20 days apart, and then on day 60, the last day of experiment, we did harvest our crops, both corn and soybean, and also we took root zone soil samples, which is basically the soil samples which are root intertwined. So let's first look at the pore water data. So clearly, we started to see the replication of the field site. What we did see is that there was high amount of iron, irrespective of the crop type, soybean or corn. Not only that, as this was collected pretty quickly and we could do in situ measurement, as you can see the student doing here, in situ measurement of iron pretty quickly, we could identify reduced iron 2 in the pore water, which was the interesting observation was soybean reduced iron, we can understand, because soybean, as I mentioned, is known to take up iron after reducing iron 3+ to iron 2+. So this was not something out of ordinary. Which is interesting to see was after a prolonged dry period, if you remember, the gravimetric water content in these crops were maintained below 15% to replicate exactly the water content that was observed in Scottsbluff, Nebraska. We still did see reduced iron. Arsenic followed a similar trend to that of reduced iron, which was found also in the field site. Uranium followed an opposite trend, similar to what was found in the field site. Again, the data is average and the error bar is standard deviation. So this was really interesting to see that corn, which is not supposed to reduce iron, still produced reduced iron in the pore water. Now we really wanted to see what exactly is happening to the pore water. So what we did, we collected major anions and major cations data of the pore water and fed into a thermodynamic model or chemical equilibrium model, especially using Frick-C platform. The one key thing that we really realized was definitely corn and soybean root zone was behaving differently. Looking at the saturation index of ferrihydride or FH, initially the pore water, it was negatively saturated or there was not any ferrihydride coming into the pore water. But at the end of the experiment, we started to see ferrihydride coming into the pore water, or it might indicate fresh ferrihydride production. And we can see the soybean, which really reduces iron, has much higher saturation index compared to maize or corn. But we had to also validate our thermodynamic model. So what we did, we didn't feed the reduced iron data that was collected from the greenhouse into the model. Instead, we used that data to match it with the model generated data. And we found that the data is pretty close, which validated our model. So now let's see what happened to the artificial soil system. We definitely did see that there was a reduction in iron concentration from the beginning to the end in both the crop types, along with there was a reduction in arsenic and uranium. Arsenic reduction was higher because it ended up to be taken up more in the plant, and it was also higher in the pore water. uranium also was taken up by the crops. But unfortunately, as we didn't have grain seers, we had to measure the concentration of arsenic and uranium in the entire crop. Now, all this definitely indicates that there is a reduction process going on in the root zone, but we really had to pinpoint and identify, yes, definitely the root zone system of an unsaturated, primarily oxic soil. If you look at the oxygen reduction potential of the soil, of the bulk soil, is primarily suboxic to oxic, and the pH is also not somewhere where ferrihydride would precipitate out. We had to really pinpoint that, yes, definitely the soil has reducing spots. That is where X-ray photoelectron spectroscopy was came into play, where we took this root zone soil after day 60 of the experiment. We also had pre-soil, that was day 0 of the experiment before anything was planted. And what we found, generally X-ray photoelectron spectroscopy or XPS does is that you shine X-ray on your sample, and it is very element specific. So for example, in here, our element of interest is only iron or Fe, and it can tell based on the position of the binding energy if our iron is reduced or oxidized. So if we look at the pre-experiment result, you see that there is no peak at all in the iron 2 position, rather we have a dip. But after 60 days of experiment and the root zone soil, when it was again ran under XPS, especially the corn sample, because again, I'm focusing on corn because corn is supposed to chelate iron, not reduce iron. We do see signature of reduced iron in the system. So this was kind of a clear indication that reducing microsites or pockets exist within the bulk unsaturated system, which is clearly important when we try to understand the impact on arsenic and uranium uptake on the crops. So to wrap up things, there seems to a lot to learn about iron and trace elements chemistry in irrigated, transiently saturated or unsaturated soil, which is prevalent in the US. And we think this is likely due to its natural transformation between nanomineral forms. So this figure shows our conceptual model of what we think is happening in unsaturated irrigated soil. Yes.
Excuse me, sir. Sir,
Yes.
sorry to interrupt you. Your time limit is over. Could you please kindly wrap up fast?
Yeah, this is the last slide. So
Okay,
nanomineral
thank you, sir.
transformation control trace element bioavailability to crops grown in the irrigated soil, and definitely we have a lot to learn about it. And this work is important to help evaluate human health risk from food crops grown in irrigated soil, as well as potential leaching of trace contaminants below the root zone. And before I end, I would really like to thank my WaARI interns, my WaARI mentors, Professor Yusang Lee and Professor Snow, and the current team with whom I worked. I understand we are almost running out of time, so if there's time to take questions, please let me know, or you can always email me at this email address. Thank you. And Dr. Arun, thank you for reminding me about the time.
No issue, sir. Thank you, sir. Thank you, sir, for enlightening us about the arsenic and uranium pollution in agricultural field, in pore water as well as in crops, and how it relates with iron compounds. Okay, now the session is open for questions. Any questions?
I do see a raised hand by Dr. Kanta.
Can I ask question? I'm Dr. Kanta.
Yes, ma'am. Yes.
Good afternoon, sir. Maybe good evening to you. I'm Dr. Kanta from SRM ISD. I have been working with uranium, mostly on the natural radionuclides. And I just would like to know, what is the source for your uranium? Is it any mines are there or anthropological region? What is the reason for the existence of uranium in your soil and water?
Okay, so if you, I don't know if you were present when Professor Weber presented her.
Can you talk louder, sir? I couldn't hear you.
Can you hear me? Hello?
Yes, sir.
Yeah, so
Yes.
I don't know if you're present when Professor Weber presented her seminar. Actually, in Nebraska, soil and groundwater has uranium, which is geogenic. We really don't have uranium mine. We just have one mine in the western part. And this site where we did our field study is not that far away from the mine, but specifically most of the uranium that you see in the groundwater or in the soil is geogenic.
Okay, because we have a high background radiation area in southeast coast of India. And have you looked at the food material grown in that area and also intake to human intake through ingestion?
We haven't looked into the human intake, but definitely we have looked into the uranium concentrations ending up in the crops grown in these areas, which has high amount of, I would say, elevated amount of uranium, higher than the national average of United States. In Nebraska at two locations at North Platte and Scottsbluff, which is one location is the central Nebraska, another location is in the western Nebraska. And we have seen measurable quantity of uranium close to 0.05 microgram per gram, close to even 0.09 microgram per gram of uranium ending up in the grain of soybean or maize. So that definitely we have observed. And we started looking at irrigation pattern impact. So if we use more amount of water for our irrigation practices, that tends to increase arsenic levels in the crops than uranium, whereas amount of uranium is higher if we use less amount of water like subsurface drip or pivot irrigation. But these are really wonderful questions, Dr. Kanta. I really appreciate this.
Because what happened was we have looked at the uranium intake and cancer risk assessment by collecting the food material by direct food studies, duplicate diet method, as well as market basket method. That is, we looked at the uranium and other radionuclides in the food material grown in that area, also the meat, milk and everything. And I identified the concentration and also related to the cancer risk assessment by comparing with the hospital register, because this is a high background radiation area. But if it is in the mining area, that will be really high, because we do have in Jaduguda and other places, we have a uranium mine. That work also I have done. That's the reason I was asking whether you have related the concentration of uranium in the food material eaten by the people living in that area and also related to your cancer risk assessment.
Dr. Gandha, this is really a wonderful question. Unfortunately, we haven't related it to any human intake. And the problem is the crops that I discussed today, soybean and corn, which are grown in quite large amounts in the state of Nebraska, is generally not used for human consumption. And we have also not looked into any animal who consume these crops and have checked out how uranium ends up, let's say, in the meat or the milk produced by cows feeding on these crops. That is something we have not looked into, but this is wonderful. So are you seeing,
right? Because over
yeah, are you seeing, yeah, so are you seeing high bioaccumulation in the end products in your study?
Yes, sir. Yes, sir.
Okay, okay. Yeah, I'd be really interested to, I would be really interested to see your study.
I can
and
send a couple of my publications in that. So, because we grouped them as 12 groups, food groups, like spices, cereal, like meat, milk, everything. Mostly from the food material grown in that area by two methods. One is the duplicate diet study and another is market basket study. What we do is we recruit the people from that area. We go and stay with them for whole day. In one day, how much food they will eat. And then calculate as per the age group also we have done it, sir. Then it was almost equivalent to the hospital registry studies, the cancer risk assessment. I can send you the publications, no problem.
Yeah, that will be really interesting. Please, email me the publication. I would really like, because we had a hard time looking into uranium from food intake, how it impacts humans. So definitely that will be helpful if you can just share that study that you are mentioning right now.
Of course, the amount of uranium will be very less, but you need a large quantity of food material, including fish, meat, egg.
No, but if it is consumed in a regular basis, even this amount might add up, because uranium, we really don't know how it is excreted from the body. So if there is a chronic exposure to even low doses of uranium, as it has been seen for arsenic, for example, in the Ganga Delta, that low exposure of arsenic through rice eating has led to quite a few number of bladder cancers in that region, a high incidence of bladder cancer in West Bengal and parts of Odisha and Jharkhand. So chronic exposure definitely can have impact even in low doses. So that low dose will add up as you start consuming it every day. So that is something to look into, especially with the public health perspective. And as I was saying that there are two ways that we can really have these contaminants within our bodies: by drinking water, which is one of the primary source, or through food material. So not only we have to produce large amount of food for the growing population, but also we have to make sure that the food being produced is of good quality. So I would really look forward to your paper, and if you have my email address, please let me know. Otherwise, I'll be sharing. I can share it here over in the chat also.
Thank you, sir.
Thank you.
Thank you, sir. Thank you, sir, and thank you, ma'am. We are moving on to the next talk. Next talk is by Dr. Pooja Devi. Dr. Pooja Devi is a principal scientist at CSR CSIO, working in the field of material science and sensor applications. Her research expertise is in the domain of materials engineering, techniques and associated affordable and portable devices development for water pollutants detection, degradation, solar hydrogen production, CO2 conversion, and antimicrobial activity. She has filed one patent and transferred one technology. She has 52 international high-impact publications, edited 4 books and 9 book chapters, and 70 conference papers. And she is delivering talks more than in invited sessions, more than 38 invited sessions. I welcome you, ma'am.
Thank you, Dr. Arun, for nice introduction. Let me first share my screen. I hope my screen is visible.
No, ma'am. Yes.
Yes. So first of all, thanks to the convener of this workshop, Dr. Snow and Dr. Paramita, for organizing such a wonderful event, a very informative event, and also inviting me to share the kind of work we are doing. So basically, I am a material scientist. I am having an expertise in the area of material science. So we work in the area of developing materials, sensor systems, FTKs and devices for water pollutant monitoring. So today in my talk, I'm going to focus towards selenium, which is one of the emerging and one of the pollutant of concern in India as well as in US. So I also had an opportunity to work on this particular element during my WaARISH internship with Professor Snow and Professor Rebecca Lay, which gave me better understanding of this element. And with the experience, I could work onto the material aspect of this in developing the device for selenium detection into water. Now coming to the just a brief introduction part of the selenium, we know more selenium as a micronutrient. It is there in several dietary supplements, including dairy, milk, and several other fooders. But we know that if it is consumed above a particular limit, like for every element, there is a limit defined beyond which it is toxic. So if it is consumed beyond a limit, it is reported to cause several acute and chronic health issues. Selenosis is very common problem which is associated with the selenium. Even the deficiency of selenium also has issues because it is required to regulate metabolic activity and even the oxidative stress, and it is a component of various selenoproteins. But when it is above the level of the permissible limit for consumption, it generally replace sulfur in several enzymes, and it affects activity of several enzymes in our system, and that lead to several diseases. So in terms of the cycle, how the elevation of the selenium, in the previous talk, Dr. Arindam told, like most of these toxic elements, they could be consumed either by water or by consumption of the soil or the fodder which is grown onto the soil, which is irrigated with the contaminated water. So likewise, selenium, the origin of the selenium high level into the water or into the soil is known to be both origin, like geogenic activity because of the microbiological activities, as well as the anthropogenic activities wherein the selenium is produced in industries, like in the semiconductor industries are major user of selenium. Likewise, there are printing industries, or so they contribute to even the mining activities contribute to high level of the selenium into the soil. And due to the microbiological or due to the microflora in the environment, it makes its way to the water, and from water and soil it makes its way to the fooders, and finally to the fooders to the life forms. So it is reported to affect different ecological components of the ecological system, being mammals, reptiles or birds or fishes. So they are found to have consume selenium through these sources, and they are found to have high level of the selenium. And like I said, high level of the selenium consumption is definitely not good for the health. If I talk specifically onto the Indian context, in India also, the region where I'm located, that is Punjab, it is found to have very high level of the selenium, like up to around 200 ppb level it is found, while the permissible limit is 10 ppb and 50 ppb depending upon WHO or US EPA limit. Likewise, Rajasthan and Haryana, these are the some adjacent region with the Punjab, which are reported to have high level of the selenium into the soil and into the water, which is being used for the consumption. So even the water testing labs in this particular region, they have generated the map of distribution of selenium contamination in this particular region by using ICP-MS based technique system for analysis of this soil samples as well as water samples, which shows that this high level of selenium is reported to have effect onto the humans as well as the animals, because there is a very common disease which is known as Dagnala in India, which is associated with the animals. So this is very common in this particular Punjab region. So if you can see the distribution of the selenium into the Punjab region into different cities of it, so you can see more than half of the Punjab is affected with high level of the selenium, particularly high selenium rich soil as well as the water. Now, myself as a material scientist, we work on to the developing the materials. We know that most of these elements, they are required to be analyzed to understand their concentration, to understand their movement, to understand their spatial temporal distributions. So like Professor Daniel mentioned about several techniques, how complex are the processes, method development, and depending upon the sensitivity of the technique, the cost of the instrumentation is also very high. So these are the most of the instrumentation technologies which are used for analysis of these elements, including selenium, arsenic, or anions and cations. One of the limitation with these kind of tools is that these are mainly into the laboratory sophisticated environment, and to use those instruments one has to be a skilled person and should know all the sample preparation, method development procedures, and definitely a lot of knowledge is required while doing the analysis of the sample with these instrumentation solutions. And no doubt these are these are the highly sensitive tools to understand the level of the elements into different kind of matrices. Now, if we talk about other kind of solutions, which has the involvement of the end user. By end user, we mean like if you want to do the test of these contaminants into the field level, testing of these elements into the soil and water. So we have the alternate solution like the portable spectrophotometers or portable field test kits. So we try to develop the systems which can be used into the field for the detection of the selenium into the water with focus towards groundwater. And if we see what is available currently, the spectrophotometer which are available, these are miniaturized spectrophotometer. They are based on a method which uses diaminobenzidine. Diaminobenzidine is a kind of organic molecule which can selectively detect selenium by utilizing its nitrogen-based chemistry. One of the problem with the diaminobenzidine method is that the time of the reaction is quite high, and there is a sensitivity issue, light sensitivity issue of this organic molecule. So when we have seen the kind of solutions which are available, which can be used for the detection of selenium, we found that most of the spectrometer which are available from Hatch company or from other supplier, they are based upon a DAB strategy. They're using diaminobenzidine. So this is to give you an idea of what kind of alternate technologies which are available that can do detection of the selenium into the field. Of course, they are for the primary screening of the water sources. They do not have that much match in terms of sensitivity over the sophisticated technique like ICP-MS or IC, but they can give you a primary idea in terms of understanding, in terms of getting screening of the water resources before their final analysis into the lab environment. So these are some of the technologies which are available. More or less, they are based upon diaminobenzidine method. And as I told that these diaminobenzidine method has certain limitation on using these kind of technologies. So what we have done, we specifically being from the material science background, we have worked upon, we have expertise in the area of developing materials. Some of those materials which we quite use for the development of sensors, as well as some other application here in water splitting based on carbon nanostructures, semiconductor nanomaterials, and 2D materials. So the plus point with these materials is where we can work with the surface chemistry of these materials. We can engineer with their shape and size. We can tune their optical properties, structural properties, and functional properties. So for any sensor development, it's important what kind of techniques we are using around which we want to develop the system. So we have we work on to the exploration of colorimetric techniques, photoluminescence technique, and electrochemistry to design these methods to design the materials and translate these materials and methods into the field usable devices. For the selenium, we have mainly focused upon one of the very unique platform, which is known as carbon dots. So carbon dots, if I give a little bit idea to the audience who is not of this background, this is one of the smallest member of carbon family, and it's a fluorescent material. We know there are different kind of organic fluorophores, and as an evolutionary progress over different materials, as a substitute to organic fluorophores, which generally have issues in terms of photo bleaching over a period of time. The gold nano cluster and semiconductor quantum dots, they come as a substitute to these materials. But while developing any platform, if we plan to use semiconductor quantum dots, their composition itself restrict their uses for the field devices, because they are made up of, again, heavy metal constituent, which make their uses, which restrict their uses. While the carbon dots, it's based on a carbon platform. So this is in a way a very environment friendly material. Another advantage with the carbon based platform, or especially carbon dots, is its surface chemistry can be easily modified. So one can design a sensor platform by playing with the surface chemistry of these materials, and by playing with their size, by playing with the doping, by playing with this number of in terms of the functionality of the surface group, one can tune the fluorescence properties. And so with this advantage, we explored this platform to design, to overcome the issues which are associated with the DAB, and to develop a new platform which can be used for the detection of the selenium in the field. So this is an overview of how the carbon dots has been progressed since 2004 with the discovery of the carbon nanotubes. And now carbon dots have found wide application in all chemical sensor, biosensors, as well as in different imaging applications, as well as in some energy related applications. So what we have done, like if you see that these carbon dots, they are reaching different surface functionality, including hydroxyl group, amino group, or the carboxyl group, which highly depends upon what is the precursor which is used for their synthesis, and the process which is used for the synthesis of these kind of materials. So for the selenium sensing, we developed this platform by using citric acid as a source of carbon, and we optimize the whole process in terms of their synthesis time, in terms of their synthesis temperature, to obtain of highly fluorescent carbon dots with a quantum yield of around 34%. And if you see the absorbance and luminescence characteristic of these materials, so you can see this material is blue luminescent under UV light. And in terms of emission characteristic, it shows excitation independent emission phenomena, which is a characteristic that the materials which we have synthesized is of uniform size distribution. So there is a uniformity into the size of the material, which is highly beneficial for a sensor platform development. So you can see what we have done. We have worked on to the various parameters in terms of the time, in terms of the temperature, how do they affect the absorbance and luminescence properties of my materials. And with optimized conditions, we did characterize this material in terms of their structural properties, like size and their size distribution through different morphological and structural characterization technique. Now to use this platform, which is a fluorescent platform for the detection of the selenium, what we have done, we functionalize this carbon platform, which is rich in carboxyl functionality with the shift base. So this shift base, it is derived from an aldehyde and hydrogen, and this shift base, it was functionalized onto this carbon dots to obtain a sensor which we call it as a functionalized carbon quantum dots. So this sensor platform is, again, you can see it is rich in nitrogen functionality, which is reported to have high binding towards selenium. So this functional platform, we confirm the functionalization by several techniques, including FTIR, which clearly shows the appearance of the formation of peak for the shift base, as well as formation of peak for the amide bond. That confirms that the ligand which we synthesized is functionalized onto this carbon platform. We also confirmed like this platform with MS technique as well as C13 and HNMR to confirm that the platform is now rich in nitrogen. So with all preliminary characterization of the platform, we develop the method, we optimize the method in using this platform for the detection of the selenium. And here you can see very clearly. So when we are adding selenium to this sensor platform, we see a quick quenching of the fluorescence of this material. And this is highly selective only towards selenium. And we could see that this is a very fashioned behavior observed while we are using this material. And we could detect the selenium in a wide concentration range. And that shows that utility of this fluorescent platform, which is modified in terms of surface chemistry, shows selective behavior for the detection of the selenium into the water. We did also some optimization in terms of effect of pH, effect of ionic strength, and other parameters which can destabilize the sensor platform. And we identified that in a pH of 6 to 7, the performance of the sensor is quite good, quite stable. So this is the pH environment the sensor is able to perform. And we found a good correlation behavior between the quenching behavior of the sensor platform with respect to the concentration of the selenium. The idea behind developing this material was to use this material to develop a field test kit or field test system. So we studied the material performance towards various other metal ions which could be present into the water. Our focus had been into the groundwater. So we did the effect of various anions. We did studied the effect of various cations and anions, and we also did the competitive ion study where we studied the effect where the other ions are present along with the selenium into the water sources. And we found that the results are quite satisfactory, and the selenium, this probe retains the sensitivity towards the selenium, even the presence of other possible metal ions or cations. And we also tried to study the behavior of this sensor platform in the presence of multi-ion mixture, where we have a mixture of different anions, including sulfate, chloride, bromide, phosphate, nitrate, and several other cations. And we found the satisfactory performance of this platform for selenium detection. To see how is the performance of the material, comparative performance of the material, we studied the behavior, we studied the performance of this material with ion chromatography, and the results were quite good in terms of, so that shows the material has a capability to be used into the field for initial screening of the water resources for the selenium. So with all the promising results which we obtained in the initial results we obtained in terms of the material sensitivity towards selenium, we did work on to the translation of this material into a field device. So where we have worked on to the software aspect of it and mechanical aspect of this device and the design of the device. So this is an architecture of the device which we have developed, and we have already filed a patent for this device. So this device is based upon, it uses a spectral sensor system, and it uses high power LED. Along with it, it has a certain arrangement of optical components, including mirror and lenses, and also heat dissipation system, so that the device at least could match over to the performance of a lab-based photoluminescence spectrophotometer. So this is an overall architecture of the device which we have developed, and you can see the device performance. So the device, it follows the trend which we observed in terms of the PL spectrophotometer, where we have intensity of the sensor intensity in terms of the counts, and here we are getting in terms of the frequency. And you can see that device shows a change over the change in the concentration of the selenium, and then the change is very linear. And so you can see the small prototypes, the small device which we have designed is able to replicate the result which we observed with the standard photoluminescence spectrophotometer for the material which we have developed. So this is the exploded view of the main whole device. So you can see this is the complete design of this particular device. And these all are, I'm not more going into detail of the device design. So this is overall layout of the design where we have worked upon overall device assembly, onto the design of the cuvette, onto the design of heat sink, as well as GUI interface for the user to use this device into the field condition. And this is an overall view of this particular device for the user point of view, where the user can first do the calibration of this device by using reference, and then do the sample analysis by using the measure. And currently this device has been programmed in such a way that it can tell whether the water is having a selenium beyond a permissible limit or not. And we are in process to work on to the adding algorithm into the device in terms of the quantitative measurements. So you can see this is the overall look of the device. It's a very small handy device and of the size of less than your mobile phone. And this device can be easily taken into field and can be used for the screening of the resources, especially for the selenium. And however, it's a modular device. In a sense, the sensor, the spectral sensor which we have used, it is of wide range. So by changing the source or by adding another source, we can extend the device towards detection of other water pollutant as well. So if I tell you in terms of the characteristic of the platform which we have developed, so this platform, it could detect selenium, like one batch of this platform at least can perform 20 tests, and it is very stable in terms of performance. It's highly reproducible, and we have worked mainly onto the reproducibility part of this material. And another aspect of this material is that it can be used and it can be discarded because of the no environmental hazard associated with this carbon-based platform, because it is highly biocompatible and it's an environment-friendly material. So that way this material, it overcomes the disadvantages which are associated with the organic fluorophore or the DAP kind of sensor platform which are there into the market as of now. Another thing I would like to add, like during my WaARISH internship, we also try to develop some platform based on electrochemistry as well as a peptide-based sensing element for the detection of selenium, where we have tried to modify the different kind of nucleotides for the selective detection of selenium, but the results, we couldn't get the very much progressive results into the selectivity part into this peptide. So there is a still scope available on developing the electrochemical platform for the detection of this particular element. To give you just as an information, like we not only working onto the selenium, we have also tried to develop this fluorescent platform for the detection of the arsenic, and we have developed the material and method. We have not yet developed the device around it. Likewise, we have worked into the detection of the into the fluoride detection for the field level detection based on, again, all the platform, they are based on carbon-based carbon dots and modification of their carbon chemistry. Lately, our group is also focusing towards developing the color-based platform, because the color-based platform has a wide acceptability at the user end. So here we are focused has been on to developing the platform which can shows the instant change in color in the presence of the analyte. And we came up with this platform for the detection of arsenic and for mercury and the copper. And this you can see, I'm not going more into detail of it. So these platform.
So I have another connection to the hospital.
Hello. I think there is some background noise.
Is having Sita India, so he's the CEO for.
Hello.
So somebody's mic is.
So that's
Yeah.
Please mute yourself. Others, please mute. Others, please mute yourself.
We initially had tried to start a MBA program as well as.
I think the noise is still there. So I'll continue anyway. So,
I would request Yeah, please mute yourself. Yeah.
So,
Dr. Pooja, please continue.
yeah.
Sorry.
Thank you, Dr. Pal. So like I said, we lately are more moving towards color-based platform, as well as developing the device around this color-based platform, which can read the color, it can digitize the color, and it removes the subjectivity into the interpretation of the color at the end users. And we also modified the process, which is a iodometry-based process, by adding certain combination of the indicators and process optimization for the color-based detection of the selenium. And we are optimizing this whole process to come up with the field test kit for the color-based detection of the selenium. Just now, to summarize, we as a material scientist people, we work more onto the material aspect, onto developing the sensor platform, and also we work on the translation of these materials into field devices, into the field kits, which can be used for the field testing of these elements into the water resources. And we have specifically worked on heavy metals and metalloids, and also we extended it to residual chlorine, which is one of the important criteria for the water treatment, to ensure the treatment of the water before its distribution into the urban or rural network. And I discuss more towards selenium today. So if anyone is interested to discuss about other elements and to collaborate with us, I'll be happy to discuss further. So this is some of the books our group has edited with focus towards the material aspect of the sensors. And also as a collaboration of WaARISH internship, we also touched upon the different aspect of the selenium contamination in water, including its removal, its distribution, its evolution over a period of time, on different treatment technologies, different sensor technologies. So this book gives an overall holistic view of this particular element into the water bodies, as well as it has a little bit touch upon onto the soil aspect of it. So with this, I would like to thank all the organizers, convener, for giving me an opportunity. Now I can take some if there are any questions.
Thank you, ma'am. Thank you for your wonderful talk. And it actually enlightened us about the steps in the development and design of a sensor to detect selenium in environmental samples. Now the session is open for questions. Any questions? I think there are no questions. So thank you, ma'am, once again. And we are moving to the next talk. Next talk is by Dr. Richa Kothari. Dr. Richa Kothari is working as an associate professor in the Department of Environmental Sciences, Central University of Jammu. She worked as a fellow in Robert P. Dr. Water Food Institute, University of Nebraska. She is a member in Professional Societies of Solar Energy Society of India and People for Animals. Dr. Kothari has also worked on research project like study of policies and mechanisms that could facilitate delivery of STA output for social inclusion in energy section funded by the DST Government of India. I welcome you, ma'am.
Thank you. There is one, I'm getting the difficulty regarding the sharing the screen. It's showing that other participant is sharing.
Yeah, ma'am, now you try, ma'am.
Yeah, it's okay. It's visible?
Yes, ma'am. Yes, ma'am.
Okay. And just also confirm me when I'm moving the next slide, it is it's opening, it's on the, you are also visible to see the next slide?
Yeah. Yes, ma'am. Yes, it's fine.
It's okay now? It's okay,
Yes.
fine?
Yes. It's okay.
Okay. We are just facing from the JNK site this type of problem on the normal part.
Yeah.
First of all, I thanks for inviting and giving me this opportunity to share my views on the low-cost bioabsorbent biomaterials and industrial wastewater for the healthy ecosystem. To the organizing team members, Dr. Paramita Chakravarti and Dr. Daniel Snow and other organizing team members. And thanks for organizing team members of the WaARISH 2021 and the organizing this conference in the virtual mode. So now come to my topic. My topic is low-cost adsorbent biomaterials for industrial wastewater integration with healthy ecosystem. As we are aware that water is in the present scenario, water crisis and energy crisis. These two crises are the gaining the attention of the researchers and the academic world because it is decreasing. Fresh water is decreasing day by day, and energy is on the part of as the industrialization is increasing, population is increasing. We are on the search of alternative energy sources, how we can reduce the energy crisis problem and how to we can overcome the problem of wastewater. As we are know that in the 50s and 60s, we go on for the industrial revolution. And after that, the problem of water crisis increasing and the wastewater is on the part of everywhere that wastewater is increasing day by day due to the discharges from the various sources, industrial sources, agricultural runoff sources, and by the domestic uses of the water is increasing. So now the time has to come, we have to move towards the resource recovery from these waste resources, or how we can alternate our potable water sources or increase the potable water sources during this water crisis. So here we are in this my presentation, I am focusing on the how we can reduce, what are the different options available on the low cost part to remove the contaminants from the wastewater and to increase the and to decrease the problem of human population by the availability of this potable water. As this is the normal scenario at global scale and the Indian part also on the national and international part, these are the normal scenic view what we are observing nowadays, and it is due to our anthropogenic activities which is main contributor for this type of scenic views and our surroundings or in our environment. Although in the recent lockdown period, one positive thing is seen and observed by various researchers and cited in various literature also, that this type of scenic views has been reduced. But how much reduced, that is a part of the study. So it is also one of the drawback of the industrial revolution part, that we are using the fresh water, and but after discharging we are getting unhealthy ecosystem, unhealthy environment, unhygienic conditions to the population or the social part. As per literature, there are various wastewater treatment options available on the part of conventional methods. Normally we are using coagulation, precipitation, biodegradation, sand filtration, activated carbon, mostly used on the part of adsorption part. So, other treatment options are, or recovery methods are, incineration, solvent extraction, electrochemical treatment, ion exchange method, membrane filtration. and some recent or emerging technologies which we are focusing, or the scientific world is focusing nowadays: nanofiltration, advanced oxidation, biosorption. As just my previous presentation, Dr. Pooja also mentioned about that, how we can reduce with the help of metal contaminants, with the help of different devices and other parts, materials she has developed. That is one of the important things that are the emerging technologies. We should focus on these type of emerging technologies to reduce the wastewater discharges. So as per the conventional and whatever the literature cited, that coagulation, flocculation, these all have some advantages and disadvantages. We can say that coagulation, it is a simple and economic technique, but disadvantage, highly sludge generation is which is creating the problem somehow in the environment. With the help of the waste is generated, it is lying on the surrounding environment or the on-site sites or the off-site sites. So somehow unhygienic conditions will be developed, which ultimately affecting our ecosystem. Membrane separation, one of the other wastewater treatment method, ideal for specific for chemical dye separation methods, but is expensive in nature. Oxidation, it is a fast process but require high energy and production of large proportion of byproducts. Ozone treatment is there, no toxic metabolites are produced but expensive in nature. Ion exchange method is there but easy to operate but not suitable for all type of organic or inorganic pollutants which we are finding in the wastewater. Then the photochemical treatment advantage is also there, but some secondary pollutants are generated, which again problem or the source of problem for the researchers and the for the surroundings. Adsorption on synthetic materials, highly efficient but economically not well suited. A poor recovery of adsorbent is reported by the researchers. So on the part of wastewater treatment, there are some terms are roaming our surrounding, that is sorption, absorption, adsorption, biosorption. Although not scientifically, these words sometimes create confusion, but there are little bit differences in all the words we are, I am choosing here. Sorption, it is the absorbing or adsorbing either the surface phenomena or the chemical physical phenomena or chemical phenomena which we are using for the adsorbed or absorbed the contaminants from the wastewater. Absorption, it is a physical process. Absorption is the chemical process. Adsorption is the physical process. Then biosorption, use of biomaterials, particularly fungi, algae, yeast, etc. This type of biomaterials to absorb the heavy metals or the other contaminants from the wastewater. So it is, in brief, we can say it refers to dissolution of molecules with a phase, means solid and liquid phase combined together to treat the wastewater. Here two phenomena is worked here. One is three molecules are there: adsorbate, material to be adsorbed, adsorbent, material adsorbing the adsorbate, and process is known as adsorption. According to the processing, there are two types of absorption methods are on their working of the mode: physical adsorption and chemical adsorption. Physical adsorption is a type of adsorption in which adsorbate adheres to the surface of the adsorbent through weak van der Waals forces. These are weak intermolecular attractions. Where on the part of chemical absorption or chemisorption is a type of adsorption in which adsorbed molecules are held together with adsorbent through strong chemical bonds. So there is a little bit difference between physical, not little bit, it a very much difference between physical adsorption and chemical adsorption. Physical adsorption is a reversible process, where the chemical adsorption is an irreversible process. Physical adsorption can be multi-layered. Chemical adsorption, it is a single layered process, means what the material we are using, it is adsorbed on the surface, inside the surface, and chemical changes are observed. Physical adsorption decreases with rise in temperature, whereas temperature is a supporting factor in the chemical adsorption. Physical adsorption increase with rise in pressure, but on the part of chemical adsorption, not significant changes with the pressure variance. So on the part of the, there are different type of adsorbents has been noticed, or as in the conventional part, or in the last 20-30 years back, if we go for that scientific citations or the scientific researches, we have found that there are conventional adsorbents and sometimes there are non-conventional adsorbents. What we are using or what we are focusing on the emerging alternatives on the part of conventional adsorbents. Commercial activated carbons from the biomasses, wood, peat, coconut shell, coals. These are the commercial activated carbons which we are using as an adsorbent on the conventional part. Inorganic materials are also utilized by the some researchers. Activated alumina, silica gel, zeolites, molecular sieves as conventional adsorbents. Then the ion exchange resins are also there, which we are used by the various researchers on the part of conventional adsorbents. These are the polymeric organic resins, non-porous resins, porous cross-linked polymers. So these are the conventional adsorbents which we are using from the historic times. But as the need of the hour or the emerging technologies are there, non-conventional adsorbents or water treatment options to clean the wastewater. So in general, we can say that adsorbents can be classified in 5 categories. Number 1, natural materials such as sawdust, wood, fuller's earth, or bauxite. Second is natural materials treated to develop their structures and properties such as activated carbons, activated alumina, or silica gel. Third is manufactured materials, synthetically materials prepared for as an adsorbents to treat the wastewater options like polymeric resins, zeolites, or aluminosilicates. Other one, as I discussed in non-conventional parts, that it is the agriculture solid waste and industrial byproducts can also be utilized for by various researchers to treat the wastewater, in which we are using fly ash, red mud as an low-cost adsorbents. And fifth is the adsorbent classification defined that biosorbents such as chitosan, fungi, or bacterial biomasses, which we can use as an alternative for the on the part of conventional adsorbents. As I discussed that biochar, that this is one of the type of low-cost adsorbent. It is produced by thermochemical conversion of biomass, and conversion processes include pyrolysis, torrefication, gasification, and hydrothermal processes. Means high temperature is required to convert any material to the biochar or carbon part. It has attracted massive attention in recent years because due to its potential efficiency in the field of agriculture, carbon sequestration, bioenergy production, and environmental remediation. That are the indirect impacts we can elaborate from the biochar part. Biochar has an excellent ability to immobilize organic and inorganic pollutants in water. So it is emerging participant in the community of the low-cost bioadsorbents. Then biosorption. It is a metabolically passive physicochemical phenomena that involves binding of specific molecules on a particular bio-based substances. Biological materials used as I discussed earlier as an adsorbents. Examples are straw, bagasse, fruit peels, pine shells of nuts, fruit seeds. These are the and various researchers working on these type of biomaterials to treat the wastewater, in particular to the dye wastewater and the textile and tannery wastewater is treated from these type of biosorbents. And presently, recent researches are going on the dead or alive algal biomasses, lignocellulosic biomasses to treat the wastewater. And what are the different waste materials we have, which we can use as in low-cost adsorbents are the animal solid waste, food and meat processing waste, crop residues, forest waste materials, wood waste, and algal waste. On the part of algal waste, recent studies we have done, and we found that we didn't get the efficiency of the system not more than 50%, but somehow it is a good option to treat the wastewater. Then forest waste materials can be also used as the bioadsorbents. High metal ion adsorption potential through ion exchange or chelation has been used by the various researchers. Forest waste is an ideal method for the preparation of biochar also. These are the waste: rubber wood, sawdust, pine bark, wood and leaf, acacia waste woods. Various researchers worked on these type of adsorbent materials, low-cost adsorbent materials to treat the wastewater and remove the heavy metals like chromium, then other heavy metals are also lead. These are also removed by these type of low-cost adsorbents. Agriculture waste materials I discussed, and removal for a potential detected by the metals like cadmium, chromium, mercury, lead, nickel. And it has been also noticed that adsorbents derived from agriculture waste can be easily modified by different chemical pretreatments to increase surface functional groups. This leads to enhancement in adsorption capacity. There are different factors of favor the utilization of lignocellulosic biomasses from the agriculture waste, because it is easy to develop this agriculture type waste materials into the biochar. These are the carbonaceous materials. And one important thing also related with this biochar, that is that it has high surface area, increased pore volume and pore distribution, which provide the surface to grab the metallic concentrations in an easy way. Then fruit and vegetable waste materials also can be used for low-cost adsorbent materials by converting it into biochar. There are the various examples cited by the researchers: banana peel, pomegranate peel, and seed, cabbage waste, cauliflower waste, tomato waste, etc. are used by the various researchers to treat the wastewater from the dye industry, textile industry, then highly organic pollutant loading industries, pollutant load industries, wastewater treated. I am also discussed that Adsorption, there are two processes of the adsorption has been cited. One is the batch type and another one is the continuous mode type. Means what is the biomaterial when it contacts with the wastewater? That contacting time is one of the important factor to treating the wastewater from the different sources. So on the part of the batch process, it normally used in the lab scale or the small scale part of the wastewater treatment, and continuous process normally used in the industrial scale processing. In the industrial scale process, continuous process is commonly used in industrial world, while batch methods are prepared on the laboratory scale because apart from their simplicity and ease of operation, they are limited to treatment of small volumes of solution, can be used only batch process. Although if we compare batch process and continuous process, that batch process is most easily to operate for the wastewater treatment, whereas the continuous process industrial scale processes needs a large number of wastewater requirement is there. So for the high volumes of wastewater, we go for the continuous process. As we discussed that one adsorbent, adsorbate, and the process of the adsorption take place in this phenomena. The biomaterials as potential adsorbents has been, this is the phenomena when biomaterials is utilized or the low cost waste materials has been converted into biomaterials. Then when we treat the wastewater and removal of mainly utilized for the heavy metal removal part. And affecting factors are adsorbent concentration, pH, temperature, contact time. These are the 4 important factors which affects the processing efficiency. So inorganic and organic, both type of pollutants can be removed or reduced with the help of biochar based, low cost biochar based materials. If we go for inorganic pollutant removal, then the processing involves ion exchange, complexation, co-precipitation, and physical adsorption. And with the help of organic, when we remove the organic pollutants, we go for hydrophobic adsorption by EDA interactions and pores filling. This is the
Ma'am, sorry to interrupt you.
Yes.
We are running out of time. Can you please kindly wrap up fast?
Okay.
Sorry, ma'am. Sorry to interrupt you.
I will try. So these are the metals, basic processing step which we are utilized by the biochar to reduce the metal removal part. As I discussed, physical absorption, ion exchange, electrostatic for the inorganic complexation and precipitation is there. Then the organic pollutants are utilized by the various, these are the organic pollutants. Removal also has been noticed by the low cost adsorbents that are with the help of processing involves hydrophobic absorption, pores filling, and electronic donor and acceptor interactions. So on an overview, we can say that heavy metals, nitrogen and phosphorus, nitrate or phosphate with the help to treat the loads of nitrate and phosphate, we can use biochar. Dyes has been removed or reduced in the wastewater treatment, and other pollutants, surfactants, pharmaceuticals and pesticides also reported by the various researchers for the low cost with the help of low cost biochar materials. These are the some studies has been cited in the literature that different dyes has been removed with the help of different biomass materials, peanut shells, switchgrass. These are the biomass materials that woody forest biomass materials has been used. Phenols are also that one of the major priority pollutant has been noticed in various wastewaters. Also found to be removed by the help of low cost adsorbents. Type of biomass materials has been used for the phenolic compound removal. Herbicides and pesticides are also removed with the help of low cost bioabsorbents. Paper mill sludge, then what we have told, the non-conventional adsorbents are there. Recently on the part of researchers using paper mill sludge, mixture of different forest biomass materials, fly ashes and ground coffee residues has been utilized by the researchers to remove the herbicides and pesticides. Metallic removals are also noticed. So in the concluding remark, I wish to share that if we go for the bioadsorbent, we have the potential to come out from the chemical world of the materials. Although efficiency part, one drawback is there, that efficiency part is not we get in higher part, but it has the alternative. We have for the green and sustainable world. So adsorption is a very effective separation technique for the water treatment in regard to cost efficiency, eco-friendly, high efficiency is available with some materials only, but simplicity in that design of the experiment is easy. Then ease of operation is there. And insensitivity of toxic substances can also be reduced with the help of low cost adsorbents if we go for the low cost waste materials as an adsorbents. So it is one of the recent field we have to work on this type of materials for our green and sustainable world. So this is all about my presentation, and I want to acknowledge my research team, research students, research collaborators. And I am working in the area of low cost wastewater treatment options and bioenergy production, particular to algal biofuels. I am also worked as WaARISH fellow with the Professor Paul N. Black at Beadle Center, University of Nebraska. So this is all about my presentation. Now the platform is open for question and answer session. Thank you. Any question? One question is asked by.
The question is there in the chat box, ma'am.
Yes. I just seen that if animal solid waste and meat processing wastes are used for water treatment, would it not interfere with the dissolved organic matter of water? That might deplete. That is a good question, but if we go for the animal solid waste and meat processing wastes for the water treatment, somehow we can't say that that water is used for the animal feed. or we can go for that water for the any particular type of potable water use. Although it is interfere, but with the help of in recent experiment, we have seen that we have used slaughterhouse wastewater and treat it with the algal biomass. And we get in good results that we have removed the color has been removed of the slaughter wastewater has been removed. Then organic load is also reduced. Nitrate and phosphate concentration has been removed with the help of algal biomass concentration. And we have used wild algal biomass, that the algal mats we have normally seen in the pond water or the which are creating for the eutrophication problem.
Thank you, ma'am. Thank you very much, ma'am, for your wonderful presentation. So we are moving to the next talk. So next talk is by Dr. Manish Kumar. Dr. Manish Kumar is working as an assistant professor in IIT Gandhinagar, Ahmedabad, with over 20 years of experience in research and teaching. Dr. Manish has an H index of 30 with over 3,200 total citations. He is a member of global collaboration of 51 institutes on wastewater-based epidemiology of COVID-19. He is also an executive committee member of International Water Association India chapter and represents South Asia in IWA specialist group for metal and related substances. Dr. Manish has also been appointed as a judge for IWA Young Scientist Award 2020. I welcome you, sir.
Thank you. I missed your name, but yeah, thank you very much for this invitation, and congratulations for organizing this conference. I don't know that how I will share. Open system preference. Oh man. What should I do? Oh. It says unlock. Okay. Will not be able to record the content of your screen until it is quit. Just a minute. Okay, now everything is audible and visible, right?
Yes, sir. Yes, sir.
Okay, so thank you very much once again. And so today, of course, being at the last, I would like to be the summing up what has been presented so far. And I would definitely finish in 25 minutes and then try to have some questions if there's anything there. So today I'm going to talk about black carbon, perchlorate, and arsenic. Maybe black carbon and perchlorate a little bit new. And so the entire concept of this paper is, or this work is that I wanted to summarize that whatever I worked at UNL. That's why I bring this one. And also is the fact that water is now having the lot of problems. I mean, earlier it was like we could have do the donation that part of country is arsenic and affected, and some part of the country is fluoride or so and so forth. but now it is multi-contamination is happening. Before I forget to acknowledge, several times I used to keep it at the last, but these are the work summarized with the several funding agency. Of course, IUSSTF and Nebraska is there. I would start like this one, is that you can keep black carbon. Of course, I don't know that whether many of you or us is thinking that black carbon is a aquatic pollutant or not, but certainly black carbon has been a air pollutant. And here you can see microplastic in the Sabarmati, because the WaARISH has river in their word WaARISH. That's why I bring this one, that there has been microplastic found in the river. There is a heavy metal may soon hit your plate. These all newspaper cuttings are from our work, which we published. And Sants River needs Satyagraha against superbug. So antibiotic resistance is also coming up in a big way. And of course, the problem starts from the sewage management. And not only that, the many times the even treatment is not the solution of every problem, because here you can see this is also based on our work, which was funded by Asia Pacific Network, that says that treated wastewater turns bacteria drug resistant. So treatment cannot only remove maybe chemical removal or biological removal, but whatever left, and it says that something which do not kill me will make me stronger. So, and that's happening. That happens very evidently, where we found that influent had the less percentage of the resistance bacteria, but effluent had more. Arsenic, the Professor Carey has presented arsenic and uranium. I think our group was the first who were the coined that or told that this is arsenic and fluoride co-contamination. We talked about that. When I was in Nebraska and talking with Professor Carey, and then we realized that uranium will behave certainly the same. And we, just after that talk, I called my PhD student at Assam, Tezpur University, and I asked him to analyze 5 samples, and all 5 samples showed uranium. So arsenic, fluoride, uranium coexistence or co-contamination has also been highlighted in many newspapers. And black carbon is, of course, the biomass burning is the soot particles of black carbon can be different of the different origin, but there is a there is a lot of emissions happening in the Indian subplate. The subcontinent, and black carbon, of course, commonly known as the soot particles. So we are talking about the nano-size particles, which has been being emitted after the burning. And when we know that there is a lot of agricultural land is there, and a lot of agricultural waste are being burnt, and that is contributing black carbon. And you can see the US is a big emitter. I mean, certainly 200 teragram per year. And South Asia, of course, India is not there in this diagram, but you can see the Southern Asia is one of the 4 biggest emitter. Of course, COVID must have put the brake on this black carbon emission by aircraft globally. But I miss that. Had it not been COVID, I would have met all my WaARI fellows, and also the Nebraska collaborators. But anyway, from the black carbon the perspective, it is a little bit useful. Every coin has 2 sides, and maybe this is a brighter side, but that negative side is too scary. Black carbon has, being the lifetime in days, impact regionally more. Globally, it may not be, but so which means that if biomass burning happens in the Gangetic Plain, probably the Gangetic Plain is going to bear the burn. And this is a life cycle, and this is a cycle, a black carbon cycle, published in Science in 1992. And this red one is given by me, where we thought that, okay, black carbon will be emitted, but anything that is in the sky has to come down at one point of time. And so our question was, how much there will be a wet precipitation or dry precipitation, how much will go fall in the river, how much will go to the infiltrate to the groundwater. And that was the question, research question, with which I got the I got the WaARISH fellowship and in the first batch. Black carbon is a nanoparticle, but here just let's have a look on the Indian subcontinent. This is published work from my paper, where 2015, 16, and 17, you see that how many surface mass concentration per kilogram per meter square it is. So but these data are available, and you can do with the so much the black carbon emission are increasing and effective. Some of the things I have kept for the information, because if organizer want, I would make it PDF and send to all the people. But the point is that some of the things are for information, so I may skip or not highlight, but of course you can ask me during the question time. Then coming to the perchlorate, how black carbon and perchlorates are related, because both are the genesis of the both is burning. Rocket fuels are known for perchlorate. The crackers are known for the perchlorate generation. And why it is dangerous? Because it's highly reactive, and then it goes to, of course, it can impact IQs of children, and especially if the mother gets infected with the perchlorate during pregnancy, the IQ level or those things has affected too much. So sometimes we don't know that why a child is born such, but maybe such contaminants are responsible. And it also can affect thyroid, and it is so highly reactive that it reacts with the endocrine, the hormones, and it is a endocrine disruptors. Here also, I would acknowledge the idea that was given to me by Dan, and that's why I kept him also as the author of this paper or abstract. And also, Carrie I had a lot of good discussion with her on the perchlorate. She also, has worked on these issues. And India, the problem is more vital, because 166 million housings are dependent on solid mass biomass burning. So straight away, the black carbon is generating, straight away perchlorates are generating, and straight away receivers are there, and the human receivers. So there are a lot of knowledge gap still is there. And so I would, of course, encourage a lot of audience to start the research on this line. But whatever the little we could do, I would just say. So the pertinent questions are that how much cogeneration happens if by burning, whether a burning process really generate heavy metals like arsenic or metalloid or the perchlorate or so and so. Whether what is their dissipation signature? How, if it generates, how does it dissipates? What is the relationship with the natural organic matter? What is their kinetics, breaking kinetics or depositional kinetics? What is the microbial degradation of perchlorates? And of course, you can always model it out. This is our responsibility also, because black carbon is found worldwide, but of course, we are the biggest contributor of it. So basically, I am going to explain perchlorate and black carbon interaction based on these two published work. So you can always download this work, or you can ask me, I can provide this. So you can see that the first work is the perchlorate behavior in the context of black carbon and metal cogeneration following fireworks emission at Oak Lake, Lincoln, Nebraska, USA. So there during my stay at Nebraska, I did that one. And any work that we do as under some supervision is not complete unless we replicate it. So when I returned, I did work in Ahmedabad, and that published this single author paper, runoff from firework manufacturing as major perchlorate source in the surface water around Diwali. So there we did the monitoring of the fireworks, Uncle Sam Jam Day. It means Independence Day celebration of the US, 4th July. And of course, this is in Ahmedabad, in India, where logically Diwali becomes the event before and after which I wanted to monitor. So this is the point of this Nebraska lake. Most of you have already visited this beautiful town. Or the state, of course, but Lincoln as far. So this is a Lincoln, this is Oak Lake. And what we did is that there is a creek going along. So we collected the sample here and after, and then also there were several sides of the lake. So there is the east side, west side, and of course this one far west. And so fireworks location was here near the west side, and the east side was opposite one. And then this one. So this location may be important for us, but what happened is that fireworks was, I told you that this was on 3rd July. So 2nd July, we found perchlorate is like below 1 microgram per liter. But 3rd July, there was incident, fireworks has happened, and suddenly 4th July, the 5th July, we took the sample and it is suited up. And then, of course, dissipation has happened. We did the sampling 7 days, so that until 11th July. About the rain, we also recorded because if there is a rain, so 5th July, there was little rain, so maybe a little bit more dilution or something, but of course, that is not also shown. And the point is that we have, this is the data in 2015, and this is the data in 2016. So 2015, this is the data, and then 2016, the peak has achieved just next day. The 3rd, the fireworks happened, and 4th, there was a peak day. And but east side, the opposite side in both cases has shown peak first. It means the opposite side of the fireworks has shown the, so it was very natural though. And then there was a circulation or mixing within the lake, and the difference between the different sampling point diminished with the time. Yeah, the same sample, we collected the sediment and we analyzed it at EDS and SEM. You can see that apart from black carbon, there were heavy metal, zinc, of course, aluminum. Apart from that, the barium, calcium, potassium, which are used in the fireworks for the different colors, has been generated. So this was already established through the microscope, also the elemental analysis. We did carbon-13 analysis, and that also shows its nature, not of petrol or coal or thermogenic or biogenic or atmospheric, but it showed the entirely different signature. So that way also it proved that those thing is generated from the wastewater. Then we did this multivariate analysis, and you can see that this is a group of the perchlorate. And the immediate connection of that is with cadmium and total organic carbon, pH, copper. So which means that the burning of this firecracker do generate metals. Now this is the Indian case, and Ahmedabad, we did, there was a city, there is a city where we collected from the river and the lake. What we have seen is very, very interesting. I mean, that is something that if you monitor, you can be 7th November 2018 was Diwali, and the 6th November I took the sample. In the lake, there was nothing detected in 6th November, and after 7th was Diwali, so I let it 1 day pass because if to precipitate finally from the air. And 9th November, if I collected, and it was 0.975 microgram per liter, which increased another day. And to the 1.03. So eventually the imprints of Diwali has been clearly visible here. But the most surprising thing was noticed in river. River, of course, the 1 day before the sample, I never had 65 microgram per liter of perchlorate. You can see this is 1 microgram and this is 65 microgram, and Diwali has not happened. Firecrackers had not been burned, but the river has shown this much. That is a typical case of how runoff, how people, the runoff can affect water quality. Not the actual burning, but I feel that the supply of the firecrackers arrived from the industry to the dealer and dealer to the retailer, and during that, so much the things or the has been sprayed and it comes into the river with runoff. Next day, after 2 days, it detected 1.14, but after just after 10 days, then on the 19th, there is no detection. So of course, it is a flowing system, so it can get flowed, and also there are a lot of effluents or those things are coming into the river. So of course, dilution happens fast. But in the lake, the dilution didn't happen fast. So that was the finding of the in the lake in Ahmedabad, and it is published. Of course, I have already, this is a graphical abstract of that paper, so you can see. So we had the perchlorate. I already told you that this red color, how it can produce is the salt, calcium salt produce, orange color, yellow can produced by sodium salts. So similarly, these colors have been produced by certain metal burning. And that's what happened is the river, they're pre-Diwali high and then post low. So river can give you something. Actually, if we keep monitoring our river, we can know. Regulators must not only the major or those things industrial effluent, but also the rivers are the system that can tell whether the regulations are being maintained or not, whether irresponsible behaviors are going or not. This is a dissipation kinetics we already did. Generally, it followed pseudo first order kinetics, but somehow if there is some microbial activities or some other co-generated things, it may go to the pseudo second order. So those things is there. Of course, I have a lot to tell. So I will now. So what does, one thing I have established till now is that black carbon and perchlorates can be co-generated, right? Now I want to tell about real interaction. What we took is that we took black carbon in the, and then put the different concentration of perchlorate. And we took the deionized water. I will take the laser pointer, So this, you can see, this is the DI water. The mobility distance, of course, you can go and read about the methodology. I can, that paper, because it is very brief time to tell you everything about that. But if black carbon would have been mixed with only deionized water, it would have traveled only 20 meters down the lane in the groundwater. This is the model for the porous medium. And this is the 3 ppm if it will be 40. And 50 ppm, the 5 ppm would have increased the mobility distance. Capability to being mobile will be 2.5 times more. So it means the perchlorate enhances the black carbon ability to get into the water system or the groundwater. Of course, with the time, we have also seen that until 168 hours, but it seems like the stability achieves in 3 days itself. So now I need to talk about a little bit about arsenic and remediation, because I promised to that. I already have, so at that time, I didn't thought about arsenic, but so I analyzed zinc and copper and like that. But certainly this idea, why I mixed arsenic with this black carbon and perchlorate business is because of the two things. One is that I already explained, the river is something in WaARISH already integrated. But we all know that arsenic is getting into the rice, and the rice husks are burnt in our fields. Whether that burning will release arsenic, because that the biomass has arsenic, because it can take in Indo-Gangetic plain, there are a lot of the irrigation happens from the groundwater that has arsenic, high arsenic, and that arsenic is very likely to be taken by the rice thing. And then, of course, if we burn, whether perchlorate, I already proved that black carbon is there, but how about arsenic? It can also produce arsenic by the burning of that. So that is why I brought this item. And of course, I will our group will be doing on work on that. But here I am just giving you a complexion of the Gangetic floodplain and Brahmaputra floodplain, a kind of contrast. So here I have done this mid-Gangetic plain, and here Brahmaputra plain. Those people from the US, of course, they can know these things, but This is also I am explaining in terms of the 2 publications that is already there in public domain. In one, NPJ Clean Water, which talk about natural recharge transcends anthropogenic forcing that influences arsenic vulnerability in the mid-Gangetic plain, and the another is in geochemistry, where we talked about the Brahmaputra floodplains. What we know, so in one case, there is a little bit saturated urban growth. Another case, there is a rapid urban growth is happening. We did the seasonal sampling. So in Gangetic floodplain, it seems like transient shift is happening. There are 3 clear season impact or the differences can be found. In Brahmaputra floodplain, it seems like rapid shift is happening, a way that there is only 2 switch, post-monsoon and pre-monsoon. And it is very likely because that place has a lot of rain and only 2 predominant seasons are there, with and without water. And this is the Gangetic floodplain. Because of this seasonality, there has been found a shift in weatherability, because we all know weathering impact on the arsenic and their mobilization. But in Brahmaputra, it remains, it doesn't change much with the season. What we found that in mid-Gangetic plain, there has been an impact of recharge, and Brahmaputra floodplain, there is a impact of iron hydroxide. So the iron oxyhydroxide breaks down and releases the arsenic. Mid-Gangetic plain seems like hottest spot and consistently. And but Brahmaputra floodplain seems like temporal shifting because of the groundwater withdrawal, some pesticides and something. So it seems like the arsenic spots where we found more arsenic this time we didn't find. So here we are reporting you maybe 3 years data and like that. So it seems like shifting the high arsenic places, but mid-Gangetic plain consistently at one location we found high arsenic. But of course, the level of the highest is the difference. We did do some probability distribution or prediction, and we found that in the Gangetic plain is likely to see more change in process or something has happened between the season and between the years. And Brahmaputra floodplain is still going through a gradual change, but the speed and those things, of course, we can do with the decadal samples or the data. Now I have 2 minutes left. This is the mid-Gangetic plain. A lot of impact of ORP we have seen that, and saturation index changing and those things. Of course, and iron has a good correlation in Brahmaputra. So if we will talk about that, okay, tell me one person or one thing that is responsible for that, I would say that ORP pH dominance is a regulatory factor in mid-Gangetic plain, and while urbanization and those related things is more governing in Brahmaputra floodplains. We have given some proposed models that, okay, for different places, but of course it is not right time to understand it. But the point is that we did felt that natural recharge led decoupling of the pH and ORP, causing the arsenic enrichment in the Gangetic plain. Whereas of course, river morphology, we already talked about that river morphology or soil feature enhances the enrichment process. But other than that, because of in Brahmaputra floodplains, there are a lot of sedimentation happens. A lot of one side sedimentation in the next year has put at the different side. So here still dissolution or the iron hydroxide dissolutions and happens, but it is going to impact us in the lot of different ways in toxicity thing. So we have to, even if concentration is high or low, but toxicity might be completely different. So I would suggest that, okay, the maybe health implications are also nicely likely to be talked. I have just put these 2 slides about the pilot scale because people were talking about the remediation and biochar and all that stuff. My 25 minutes please. So I need to stop it. So this is pilot scale. We have done arsenic fluoride removal. If there is a problem of arsenic fluoride coexistence, I thought it is my responsibility to find a solution for that. And now we have created a win-win composite membrane, and which is under patent. It is application is under patent. We had the idea we created two-phase genus particle, which has the core of the iron nanoparticle. And so that one side will remove arsenic and another side will remove fluoride. Of course, we have a lot of things, but I would leave you with this video. Maybe this is just 3 minutes I will operate, and then maybe we can, but I need to find if it is, it can work. Okay. Is this, can you hear the sound? Sound is audible there or not?
Sound is not there, sir.
Okay, so then what I will do.
Yes, sir.
Okay, so I think I need to remove my earbud. Okay. Oh, if there is no sound, but no, we have just tried to create that. How can we do the if every hand pump can be the made. So this was the entire concept. Okay. The entire concept, I would just show you the 1 second and then. So main objective of this was that, of course, the genus-like particle, which is the two-phased one. And if that bead will be incorporated into this seat, and that seat can be incorporated with the hand pump. And so each hand pump can have the system installed in that, which can remove the arsenic fluoride. Okay, that is all. Of course, this sound one I would have checked before, but that's all. This is my favorite picture of the WaARISH one. But of course, these are the different ventures or the landmarks. Thank you very much.
Thank you, sir. Thank you, sir, for your wonderful talk. Now the session is open for questions. Any questions? The one question is there in the chat box.
Yeah, Somik is really active, and I like that. Yeah, how to remove perchlorate from natural water bodies? Can we rethink about the constituents of the firecrackers? Perchlorate is almost entirely eliminated from the human body 6 to 8 hours. Then how will it so adversely affect thyroid? We can manage thyroid disinfection with supplements and also. So there is two parts of this question. One is that how to remove it. I already the cyanide or the perchlorate are so reactive, I would say that because he himself has written that in 6 to 8 hours it goes out of, which is correct. But it is exposure. Once we get exposure with the carbon monoxide or perchlorates, its damage is done. We cannot, damage will be done once we are exposed. So saying that the weather constituents of the firecracker, of course, it is getting more and more regulations are coming, but it is not only firecrackers, but rocket fuels and those things. So where there will be instant energy has to be required, perchlorate is bound to generate, in fact. I don't know that we can change the burning process, our biomass burning process. So it is little bit far cry, and firecrackers are getting more greener and greener in terms of course, the more regulations are coming. But I showed you that even I wrote in that paper that whether manufacturing is a problem or burning is a problem, because in the river Sabarmati, it was shown that before burning, it was so high elevated before Diwali itself. So even if the warfares, lot of things we are building. What do you think? I didn't have that sample and all that, but that is going to bound to have those things. So it is little bit complex aspect, but what I would say is that we need to keep monitoring, and so that people should be aware of it. Maybe the mask can be good while burning the firecrackers. But then the second part is the thyroid. I already told you that the reactivity is very high. So probably that is the one. Of course, we can correct it through the supplement or through the medication.
Thank you, sir.
Yeah, so if presentation was nice, there should be more questions. Otherwise, it was a okay average presentation or something like that.
Okay, sir. Thank you very much for your wonderful talk and presentation. So we wind up this session now. So lunch break is there next, and we will meet again at 1 o'clock for afternoon session. Thank you all. Thank you all.
Good afternoon for the evening session. So I would like to welcome the session chair, Dr. P. Muthamil Chelvi. Dr. Muthamil Chelvi is an assistant professor of Department of Chemical Engineering in SRM IST. Her area of research interest includes development of integrated water purification hybrid system, and her current research work in progress includes design and scale-up of adsorption column for cations removal. I would like to invite you for the session, ma'am.
Thank you. My voice is audible or not? My voice is audible or not? Hello. Good afternoon to one and all present here. My voice is audible or not? Sharad?
Yes, madam, you are audible.
Yes, thank you, sir. So I thank both the conveners from India as well as US who gave the platform to me to share this session in the Indo-US bilateral workshop. So in this event, I welcome Rajesh Singh, a scientist at the National Institute of Hydrology, Roorkee, and he is an awardee of the prestigious Water Advanced Research and Innovation Fellow by Indo-US Science and Technology Forum and Government of India. Dr. Singh is a member of PS Public Drinking Water Supply Service Sectional Committee, SSD 14, and has served as a member for various committees constituted by Honorable NGT, MOWR, etc. So he is actively involved in research, training, and consultancy activities related to water quality and pollution abatement in the National Institute of Hydrology, Roorkee since 2011. Before joining National Institute of Hydrology, he was a technical manager at Ion Exchange Limited, Mumbai. He was involved in designing water and wastewater treatment system based on IX RO, membrane bioreactor, etc. His current research focus on water quality monitoring, pollution abatement, nutrient remediation, and microbial induced contaminant mobilization. He has published more than 45 papers in peer-reviewed journal, conference, and 1 patent. He has 247 citations to his work and has an H-index of 8. With this credit, I invite Rajesh Singh sir to deliver the talk. Over to you, sir. Sir?
Am I audible?
Yes, sir. Yes, sir, you are audible.
Okay, so thanks for the nice introduction, but I am not able to share my screen.
Please wait for a few minutes, sir. I will help you. Sharad? Sharad? Sharad?
Yes, now I think you can share.
Yes. Sir, kindly. Yes. Sir, kindly you please check now, sir. Yes, sir.
Yeah, I think now I can share.
Yes, sir. Your screen is visible to everyone, sir.
Okay, so thank you very much, madam. And first of all, I would like to thank the organizers of this Indo-US bilateral workshop on water air research initiatives for societal health. And so I am going to take you to the wastewater that is emanating from our households, and I am also going to take you to the rural India where wastewater now is a critical problem. Now, the motive for the study which I am going to present, it came from the ponds, and when I was a child, I used to go to the ponds in my village, swim there. Apart from that, we were getting very tasty fish from the ponds. But nowadays, if you will see the right-hand side of my screen, then almost all the ponds, they are covered with the aquatic weeds like water hyacinth, duckweeds, lemna. We surveyed the village ponds in the western UP, 3 districts, and we found that almost all the ponds were hyper eutrophic in nature. Now, what has led to the conversion of this ponds from the aesthetic look to this choke condition, almost dead. And that is because actually I think most of the people says that this is because of the excessive use of fertilizers in agriculture, but agriculture was practiced earlier also. So this change, actually this change has come from we people, from because earlier this was a source of water for the people in villages, but with the advent of hand pumps, with the advent of bore wells, and then the government of India, it came up with the ambitious program of providing piped drinking water supply. All these are very good, because now the people are getting very good quality water. The diseases related to water, drinking water, they had reduced a lot. But because of this, we had started neglecting the ponds in the villages, which were lifeline for the village. They were ensuring the availability of water throughout the year. And apart from that, actually, if we go into the hydrology of the system, then these are actually the shock absorbers during the monsoon period. So whenever the high rains are there, these water bodies actually, they take part of the rainwater and reduces the floods in the rivers. Now, when these ponds are eutrophied, then slowly the capacity of the ponds also reduces, and because of that, the water retaining capacity decreases. Apart from that, they act as a recharge structure, and if the water is contaminated, then what they are going to recharge? So they are going to recharge the contaminated water, and this contaminated water will appear in our drinking water. So that's why we thought that this is a big problem, and this we need to find out a solution and showcase it to the government, so that that can be adopted by the government along with the other along with other schemes. And while surveying the villages, we found that that the Swachh Bharat mission, which is again a flagship ambitious program of government of India, making India the open defecation free. That is also contributing to the plight of this ponds. This program has given us very good results. Now almost 97% of people in rural India, they are using toilets. But actually this program came with twin pit toilet designs for the rural India. But our rural India is also heavily populated, and because of the lack of space, the people are generally not going for the twin pit toilet designs, but going for the septic tanks, and the septic from the septic tank, it goes into the drains. Now the outlet from the septic tanks, that is 30 to 40% treated. So it is having all the organics, or I can say the BOD. So BOD is reduced to 30 to 40%, and it is having all nutrients. Now these nutrients as well as organics, they go to the pond and contaminate it. So what we thought that we need to treat it. And now there were several treatment options which are visible on this slide, and these are very good technologies. But from the Ganga Action Plan, we learned that these technologies, although they are very good, they are able to give very good quality treated water. But in our conditions, these technologies do not operate as per for what they are designed, and that is because of the lack of uninterrupted power supply. Apart from that, the lack of maintenance and the lack of manpower. So that's why we pointed out that what the technology for rural India should be having what, and so these are some of the points. And it was that it should not require mechanical equipment, it should not require power supply, it should not require trained operators, it should utilize natural methods, because if we are dosing chemicals and blah blah, the people in the village, they may get affected. And the byproduct of the treatment that can be utilized by farmers, or at least it is not harmful to them. So we zero down on the root zone wastewater treatment technologies, and there are several variants, although I had put on the slide the three most prominent, and then other variants are the change in these three variants. So here in this village where we wanted to so-called this treatment technology, we went for the horizontal flow, subsurface flow constricted wetland. And as you can see the picture on the right hand side of the slide, so this is the so this is the pond, and this is the constructed wetland. So very aesthetic look, and the villagers were also quite happy with the treatment system. And you can see the birds. So a lot of birds started coming to the pond as well as the treatment system, because the life has come. So actually, it is having so it is having all forms of life except fishes, and I will discuss about that and we will go at the end. So what happens in the constructed wetlands? So here the plants play a very important role, as well as the microorganisms or bacteria, they also play an important role. So the plants actually, they release oxygen from the roots, and near their roots aerobic, anoxic and anaerobic condition exists. So as we go near the roots, then it is aerobic. Slightly away from the roots, it is anoxic, and then anaerobic. So we can say that there are millions of reactors with this different oxic conditions in the reactor, and there are millions of the microbes or bacteria which are there to degrade the organics that are present. So because of the change in the oxygen level concentrations in the constructed wetland, we are able to go for nitrification as well as denitrification, and because of that, the removal of nitrogen takes place from the wastewater. The microbes, they oxidize the carbon that is present in the water, and at the same time the plants uptake the nutrients from the water. So in this way we are able to reduce the nutrients as well as organics from the wastewater, and these are some of the mechanisms which is taking place for the removal of pollutants from the wastewater that is going into the constructed wetland. So coming to the rejuvenation of pond in Ibrahimpur Masai village of Haridwar district. So this is the study area. We are also in Haridwar district, and therefore we selected a village which is around 15 to 20 kilometers away from our institute. And here you can say, so this is the Ibrahimpur pond where we constructed the constructed wetland. And this is the Masahi pond, which we also monitored as a control pond. So to understand that exactly what is happening, how much improvement is because of the constructed wetland that is installed. So these are the salient features of the two ponds. This is the reference or control pond, and this is our experimental pond. This is the area of the pond. So there we selected the ponds which were almost equal in size, and they were receiving similar type of water, and they were in same geographical locations as well as weather conditions. So here we rejuvenated the pond, and for that we did desilting and other activities. So, so earlier the pond depth was 2.3, and we increased the pond depth to 3.4. So you can see the pond is deeper than the previous one, and the reference pond is having depth of 1.5 meter. Although this is 1.5 meter, but still I will be showing the results, and we were getting lesser DO in the Masahikala pond, and then there was 2000 meter cube, almost 2000 meter cube increase in the capacity of the pond. So these are the activities that were carried out. So we did some household surveys also, and this is the status of the pond before the rejuvenation activities. So we removed the weeds, we did desilting activities, then we constructed the constructed wetland, and then we also did the performance evaluation. So we monitored and operated this treatment system for almost 3 years, and I am going to present the results for 1.5 years. So this is the schematic of installed treatment unit. So the grid chamber, which was having three numbers, so 3 baffles were there. The length is 2.5 meter, width 1.4 meter, and the depth was 1.3 meter. So actually 1 meter is the actual depth, so 0.3 meter is the freeboard. And this is the root zone based wastewater treatment. So here, this is the side view and top view. So this again, this land for the constructed wetland we expected from the pond because no land was available for this activity. So this is the pond. This is the constructed wetland in the initial phase, and before I showed you along with all the flowers and the plants. Here we used Canna indica and Phragmites australis for the treatment. This is the diurnal variation in the wastewater of the ineffluent to pond, and here you can see that the fluctuations in the discharge that is coming from the village, and it was very high in the morning hours, and then it reduces, and then in the evening it again it increases. But most of the effluent was coming in the morning hours, and this is the BOD fluctuation, and you can see the BOD has gone up to something around 20 to 30 milligrams per liter, indicating very high strength domestic wastewater, and that is because of the washings of the cattle rearing sets and the food waste that is coming in the drains. And one interesting thing we observed that the electrical conductivity, BOD, as well as nutrients, they were following the pattern of the wastewater flow. So when the wastewater flow was high, the BOD was more, the conductivity was more, and the nutrients are also more. So that is because of the input from the activities that the people were doing at that time, and the flow was increased, and then the sweeping of the drains. So whatever has been settled in the drains, that is also coming along with the wastewater. Now coming to the results. So here on the left hand side, I had shown the the parameter value for the inlet to the treatment system, and the pond water quality. And here the comparison of the Ibrahimpur pond is the IBR pond is Ibrahimpur pond water quality, that is our experimental site, and this is the control pond where no interventions are there, Masahi. So as far as the pH is concerned, you can see very good variation in pH is in the ineffluent, and same is reflected in the pond water of Ibrahim Masahi. But after treatment, the Ibrahimpur pond has pH is in the narrow window, and this is actually what is required for the organisms to survive. So if a lot of fluctuations in the pH or any other contaminants are there, then the life actually it will not sustain. And this is the BOD levels. So BOD levels as it has gone up to, as I told, that it has very high BOD levels up to 350 in the ineffluent. And however, the BOD reduced quite a lot, and the average BOD was around 41 milligrams per liter. So almost what we found that almost 83% reduction in the BOD was observed. And if we compare the control pond with the Ibrahimpur pond, then we see that almost 60% less BOD was there in the Ibrahimpur pond. And these are the values for the nitrate and ammonia. And here again, we found very low concentrations of ammonia in the pond, as well as a nitrate concentration also reduced. So this is initial period when we were establishing the pond, but after that you will see that the system has stabilized, and we are getting very good results. The average value is slightly higher because of the disturbances in the initial period. And then coming to phosphate, again, phosphate was also very low. And because of this decrease in the nutrient concentration, we found that the reoccurrence of eutrophication was not there, and we did not observe the growth of water hyacinth or duckweeds again in the pond. Then coming to the E. coli, which is an indicator of pathogenic contamination, we found 3 log reduction in the pathogenic counts, and almost 3 log, the E. coli counts in the Ibrahimpur pond were almost 3 log less than what was observed in the Masahi pond. Then coming to DO, which is a very important parameter for the aquatic species, and here we found so we found a good increase in the dissolved oxygen of Ibrahimpur pond, and as you can see in comparison to Masahi, the DO was quite high in Ibrahimpur, and the average DO that was average annual DO that was observed was around 5 milligrams per liter. But and then we thought that okay, now the pond is suitable for the pisciculture, because we wanted that the system should be sustainable, and if we can grow fishes in the pond, then it will be self-sustainable, and the village can take operation and maintenance of the treatment system as well as other associated activities. So because of that, we went for we monitored for the diurnal variation in the DO concentrations. So there was a good variation in DO, and that was because of still the organics were going in the pond, and then there were good amount of algae. So the algae, it was generating a lot of DO due to photosynthesis in the daytime, and then in the nighttime, because of respiration, the DO was reducing. So we talked with the fisheries department of Uttarakhand, and they said that yes, we can go for the fisheries, and this will also reduce the algal content in the pond, and this decrease in DO after that will not be so much. But unfortunately, after that, the Corona episode came, and because of that, we were not able to introduce the fishes in the pond. So yeah, so we got, and here the overall result I am presenting here. So these are the pictures actually show the different stages of the pond, and this is before rejuvenation, and this is after rejuvenation. So you can see DO has increased, and BOD as well as other nutrients has reduced, as well as coliforms. And the aesthetic look of the pond also improved. The villagers were very happy, and the kids, they were they really enjoyed the boating when we were collecting the samples from the pond. So we kept our boat there for a few weeks whenever we were going for sampling, so that the kids can also enjoy. So this can be a good point for a good location for recreation activities. Apart from that, we also monitor the GHG emissions along with CEH UK. So along with CEH UK, we monitored the zooplanktons as well as the greenhouse gas emissions, and we found that the almost 10-fold reduction in the methane generation was there in the pond with the treatment system. Introducing the treatment system will also help in environmental mitigation. Apart from this project, we are doing several projects through Ministry of Water Resources, River Development, and Ganga Rejuvenation. And one project we are doing with the DST, whether we are monitoring the treatment efficacy of the floating wetlands as well as different other variants of wetlands. We had also constructed a full-fledged constructed wetland treatment system for our residential premises, and we are monitoring decay rate for this part of different pollutants for this part of the country, and so that a manual for construction of optimized construction of these treatment systems can be developed, as well as we are also trying to improve the performance so that the footprint can be reduced. We have also introduced the online monitoring system so that we are getting continuous data from the treatment system. So, so with this, I would like to thank my research team who are working with me on this project, on this real-time, real-life problem. I'm also thankful to NIH for the continuous funding, because of which we were able to conduct the experiments in Ibrahimpur. So with this, thank you, and now I am ready to take any questions, if any.
Thank you, sir. Thank you for your wonderful talk. The talk which was delivered by you, it is important as well as need to the current scenario. And in addition to that, we seen the method is developed. It is a natural method, and it will free from chemical free also. Yes. Now it is open for questions. Participants, if you have any question, you kindly please raise the question or post it in the chat box. Participants. Have any query, kindly post it in the chat box, or you please unmute yourself and then you can ask directly to the speaker.
It seems all slept because of this post-lunch session.
It is, sir. Sir, I have one question in my mind.
Yep.
So, for example, sir, the growth depends upon any monsoon conditions like that, sir.
Growth of what?
The wetland, the plant, sir.
Yeah, so actually during monsoon and summer season, the growth of plants are very good. But during winter season, if when we are actually the problem is when we are establishing the wetland. So during winter season, when the temperature is very low, and especially in Roorkee, so here the temperature goes to 4 to 5 degrees Celsius, and the maximum temperature during December and January is around 12 to 13 degrees Celsius. So during and it's almost smoky during that time. So because of that, the sunlight is also not there, so the plants does not grow. But once the system has been established, then the plants as well as the microbes keep on doing their jobs. As well as the advantage with the wastewater is the temperature of wastewater is generally higher than the ambient temperatures. And because of that, the system performs during that time also. Only concern is where the freezing environments are there. So we are also planning to set up some experimental sites in the hill regions of Uttarakhand to see whether they can perform in the winter season also, and if they can perform, then that will be a good contribution for the hilly states where again this the mechanized treatment system that is not able to perform during the freezing conditions.
Sure, sir.
Yep.
Thank you, sir. Thank you for your wonderful talk. So we will share your mail ID to the participants. If you have queries, kindly they will contact you, sir. Thank you, sir.
Sure.
Thank you for your wonderful talk. We will next move to the next speaker. Thank you, sir.
Thank you, ma'am.
Thank you, sir. Next speaker, Dibba Sakkar, is working as Assistant Professor, Department of Physics and Nanotechnology, SRM IST, and has been active in the nanostructure and material science field for 20 years. He graduated from Jaipur University, Kolkata with the physics honors. He obtained a PhD in material science and nanotechnology from Jaipur University and then moved to the University of Jerusalem, Israel for his postdoc research. His research focused mainly to design a nanofiber air filters for PM2.5 filtration and understanding the photoreactivity of semiconductor nanomaterial. He successfully completed DST project on the development of nanostructure media for PM2.5 capture and field a pattern. We request you, sir, to deliver the talk. Over to you, sir.
Okay, thank you very much for your nice introduction. Let me share the screen first.
Sure, sir. Yes, sir. Your screen is visible, sir.
Okay.
Please make it slide. Thank you. Yes, sir.
Yeah, hope it is visible. Yes.
Yes, sir.
Okay. 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 Thank you very much. So, so my last speaker, Dr. Rajesh, he discussed in details about the water pollution and its treatment. Yes. So today I am going to discuss is another aspect of this workshop, that is the air pollution, and we are going to discuss in details about our development of this air filter. So before going to discuss this our development, I would like to spend a few slides on the basic understanding and some information. So what is air pollution nowadays? Everyone we know. And yesterday morning, Dr. Tripathi discussed in details how this secondary pollutant sources affects and how this air pollution is coming. So I want to add one more information here. So do you know a person in a day we can take 10 liters of air from this environment per minute, then you can estimate how much air we are taking in a full day. And if this air is polluted, and if there are many harmful substances in this air, so it will affect a lot in our health. So, and air we cannot visible, yes. So that's why it's called this invisible killers. And do you know that every year annually 7 million deaths is due to this air pollution, and that is all over the world. But if you observe, let me, yes. So if you observe, this effect is all over the world, but this region, what is black color showing, like the developed country in Asia, India, China, this effect is it's more. And this due to this air pollution, so it's affecting the lungs, so lungs cancer, strokes, brain cancer, heart. So all are related to this air pollution. So it's so much dangerous. Now, suppose you are staying in your institute or home, how you can understand the what is the air quality there. So there are two websites. So you can also visit these two websites. So where from you will get the real air quality index all over the world. And suppose if we are in India and we want to focus here, we just zoom in. So we are getting different type of dots, yes, with different colors. So each dot is representing a life center where they are monitoring this quality of the airs. And the green dot is good quality air, and if when you go for red dots or deep red dots means it's hazardous. So if you focus any of the Delhi around, so if you will get all this data, and all we know how much air pollution affecting in Delhi. So we are getting 449. So what is this 449? This is just a number. So from this number you can understand this quality of air is not good, and green means good, like moderate, like this. And from this website you will get last 12 months total PM 2.5 percentage, total PM10, oxide, nitrate, all these data you will get. Now the question will come, what is this PM? So PM means basically this particular matters. It's called PM means particular matters. So what is this? It is basically a mixture of the particle, and it mixes with the liquid, and all this liquid and particle, and there are lots of organic carbon, elementary carbon, oxide, nitrate, all are mixed with this particle, and all these things is suspended in the air. And there are, all of you know the name, maybe PM10 or PM2.5, yes, like this. So what is this PM10 and PM2.5? So this is basically, it depends on the diameter of this suspended particle. It's called the aerodynamic diameter. And if some particle diameter is less than 10 micron, it's called PM10. And some particle diameter is less than 2.5 micron, it's called PM2.5. And all we know, this PM2.5 is the more dangerous. And it is not necessary this PM, it will be like a circular shape. It's not necessary. It may be circular, it may be rod-like, it may be any shape, but diameter will be less than 2.5, less than 2.5 to any region until the nano level. And there are different chemical composition also there. Now you have to understand that air quality is good or bad. How you can understand? There is some color codes, yes. So like it looks like this, okay. So if less than 50 means green, that means it's good quality of air. Now if yellow means it's moderate. So if it is like reddish means it is unhealthy. Now it is deep maroon means it is hazardous. So from this color code you can understand this quality of the air. Now the question will come, what do this number means? Yes, so that it is very important. Suppose someone is telling here in Chennai PM2.5 is today is 160. So what is 160? So this number basically the concentration of the floating PM2 point, the organic compound, including the inorganic compound, all the mass, and we are calculating the mass per unit volume. So in this case, generally we the unit of mass it measured by micrograms, and suppose we are telling this until 50 is good. What does it mean until 50 is good? That means 50 micrograms of PM2.5 it's allowed per meter cube of air. So if you are taking 1 meter cube of air, on that 1 meter cube of air, the PM2.5 should be 50. That means it's good. If it is more than 50 or 100 to 50, so what is 100 to 50? As I told, it is the micrograms of this particular matters, that should be. And why we're more concentrating everyone is 2.5, because it is found that if the particle size is less than 2.5 micron, then it can easily penetrate to your respiratory system, and it can go throughout your respiratory tract from the airflow when you are inhaling. And what will happen, then it can easily penetrate from here to the alveolus or the blood vessels. That means that if the air is polluted, that pollution or that means that's poisons you are taking directly to your blood. Now you can understand how it is related to your heart, your brain, cancer. So you can understand now. Yes. Now what is the solution then? Yes, there are lots of solutions. So like all government is taking this Clean Air Act, and you can build awareness among us, and you can use more clean energy or renewable energy. Eco-friendly transport, because many pollution is coming from the transport, so you can use the eco-friendly transport, and green buildings. Okay, but so when suppose we are staying inside our home, we are safe? No. So when we are inside, when we are outside, everywhere, if air is polluted, it can go inside your house also, and if you're coming out, that time also air is polluted. So we have to keep ourselves inside our home and outside our home. So there are, what is the solution? Solution is there. Yes, solution is there. So currently this solution is available, like this the protection which is available in the modern buildings. So like any modern building, so you'll get some ventilation systems. But what about the normal building, like normal residents or normal people? So, and this ventilation system is too much expensive. So you cannot install in normal home. You cannot use, all people cannot use, because this is so much sophisticated. There are a lot of segment. It's related to power consumption. You need electricity. There are many filters. That means this system you cannot use everyone in our home. Yes. And moreover, the things, whatever the system, air filtration system is available, they are very good to filter PM10, but they are failed to filter perfectly PM2.5, which is dangerous. Now, if when we are talking about outdoor, yes, you can wear a mask. So nowadays, last 2 years, all we are habituated to wear a mask. Yes, and suppose you are using good mask, like very good sophisticated mask. If you are using very good PM2.5 mask, is called. Actually, it is every mask is not 2.5. It's just written. Okay. Now, if you use a good mask, you will feel that if you wear it like maybe 2 hours, 3 hours, you are going to feel some breathing problem. Yes. Similarly, when the ventilation system which is commercially available, there are also some airflow. So what is the breathing problem? The breathing problem means air from outside is not coming properly inside it. So you have to take, you have to give more force. You have to give more force to take the air. Yes. So what is this? This is called pressure difference. Okay. So why it is coming? Because all this commercial filter or this mask, so they are using this micron fiber in this system. So what is happening? So they are the system, like the filter, whatever using in this conventional air purified system or this air filter system or mask, it's stopping, that is blocking this airflow. Okay, so how it is? So it's made by micron fiber. It is stuck type of structure. Okay, it's stuck structure. So, and as I told, this is not successful when it comes to filter PM2.5 because it has large pore. So what is happening? It is blocking the big particle 10 micron particle. But when it comes less than 2.5 micron, even nanometer scale, it is easily penetrating. Okay, it is easily penetrating. Now the solution to finding this solution, last 10-15 years, lots of research going on throughout the world, even more in US, even in India also. And if you notice, the number of publication is increasing slowly. And this field, you cannot say this field is for the material scientists. You cannot say this field chemist. You cannot say this field is for environmental scientists. So it is totally interdisciplinary field. So everyone is using, everyone is working now. Okay, and is developing. So what is the development? So everyone is trying to search some new materials. Okay, that is helpful to filter this PM2.5. That means we have to search a new material. Okay, from this, the conventional technology, it is not working because Until PM10, it was okay, but now last couple of years in India, we are focusing a lot of PM2.5, and thanks to nanomaterial and nanotechnology. So that nanomaterial nanotechnology is creating a new opportunity, new direction in this field. So how we can use the nanomaterial? Suppose this conventional, what is the scaffold structure of microfiber? So if we somehow create the nano net type of structure, so like there will be scaffold like layer by layer by layer and all interconnected together. If we able to prepare this net type of structure in the nanoscale, so then what will happen? The pore will also reduce, and it will effectively can filter this PM2.5. Okay, this is the concept. That means if air will come with the polluted particle and it can able to filtrate. Okay, and there are lots of other benefits also in the nanoscale. What are the benefits? So if when we will go in nanoscale region, what will happen? We are going to get a large surface area. Okay, and that means we are going to get large aspect ratio, and these are very good for mechanical property if you go in nanoscale. And there are lots of tuning in your hand, like you can tune the surface chemistry on the surface of the individual fiber, you can tune, which was not available in the traditional filter. Okay, now you can incorporate the multifunctional properties. So instead of one property, you can add 2-3 properties also in the nanoscale region. And still now, what are the development we found? So the question will come, okay, we understand nanoscale it is possible, then what type of material we should use for this type of development? So still now we found that polymer material is very good to capture this PM2.5. So this is from Stanford Professor Cow paper. So what they found here, that this result is shown. What are the results? The polymer they can capture efficiently, and this PM2.5 capture efficiency, this is removal efficiency, which is increasing and related to the dipole moment of this polymer. So like if you consider this is 0.6, 1.2 is the dipole moment of a single unit of the polymer, because they found this capture of PM2.5 is related to dipole-dipole interaction or induced dipole interaction. So when we increase the dipole moment of the polymer unit, so that is more capable to filtrate PM2.5. And one second more observation we found, what is found, that this what we are talking about, this nanoscale fiber or nanoscale net. So the diameter of the net, it is also related to air filtration. If you have low diameter, so you will get more surface area, and more surface area means more chemical, you can more binding energy, more dipole-dipole interaction, more induced dipole interaction you will get. So there is two things. One is, so we are going to keep in mind that there is two things. One is dipole moment, and second is this diameter of the this net structure. Now, what is our plan? If now, so okay, what was the problem? They was found. So they are able to capture PM2.5 very efficiently, but there was the problem of this air blocking. So air blocking, because we want any ideal air filter, it should be very thin, so that when we are taking the air, though if it is mask or it's individual or home, it should be very thin, so air can pass easily. Our purpose to block the PM2.5. We should get a clean air, but our purpose is not to block the air, so air should flow. So that was the problem. So by keeping it in mind, so in our group, what we plan, okay, first we will prepare a scaffold structure, net like a structure, this type of structure. First we will prepare, and then we will focus how we should increase the binding affinity of PM2.5. Binding affinity, how we can bind this 2.5 in this individual fiber. That was our plan. So that means first we have to synthesis this net type of structure. Yes. So how we can synthesis? There are different methods of synthesis of this nanomaterial, all of you know, but there is only one method is there where you can synthesis this net type of structure. And our purpose is not in the laboratory scale, like a small piece or small dimension sample we are synthesizing. No, our purpose to commercialize this one. That means we need a big scale, nanoscale, but in big. Okay, so large nanoscale. So there is only one technique that is called electrospinning. Okay, this is very well known and well established techniques. So what is electrospinning? I am not going to discuss. This is another field. But here we applied a high voltage. High means 10 kV to 30 or 40 kV in between. So this is the polymer solution. and this is electrode, means this is metal. So in the polymer solution here, this needle, this is also one metal. In between this metal and this metal, we are considering two electrode. In between, we are applying kilovolt order of electric voltage. So what is happening? A huge electric field is creating here. And due to this electric field, it is pulling this polymer solution towards the collector. Okay, so we are going to employ this electrospinning to prepare this net type of structure. This is our plan. And second, as I told, that we have to consider like how we can increase the binding affinity for PM 2.5. This is not a particle. The lots of ions, lots of chemical is surrounded on a particle. So what we want, suppose this is a PM particles and there are lots of ions is there. So we want to create a attractive force towards our filter. So that point we are considering. We did some theoretical calculation. This is from the density functional theory calculation. So our collaborator Dr. Thapa did this one. So what we found that we tried with different type of cationic head and polaric tail. We tried different type of chemical. So then we found there is one chemical is called CTAP, which is very commonly used for surfactant. And when we are adding CTAP pan this one, so we tried with different type of polymer and we added this one. What we found when we added CTAP in pan with some certain percentage of ratio, we are getting more dipole moment. So that we want, because at the end we want more dipole moments, because we want more dipole interaction for the PM particle and with the nanofiber, this scaffold structure. So okay, now theory we develop, and now we have to synthesize this one. So with the electrospinning technique. So first we prepare the solution. Okay, so we have taken 6 to 8% of pan and with 1%, 1.5 and 2.8% of CTAP. Okay, and we prepare the solution and put this solution in the syringe and we apply the electric field. And there are different type of process parameter is there in experimental. So all the parameter we have to standardize one by one. So we have to tailor the diameter and porosity by standardize one by one parameter to get a good nano net type of structure. So we change all the process parameter, and at the end, whatever the sample we prepare, as it is nanoscale, we have to understand what is the dimension. So we did FECM analysis, field electron scanning electron microscope, to understand the diameter of the sample. So next slide I am going to discuss. This is the diameter of the sample, or this is the morphology. So this is very nicely prepared. It is very nicely prepared. This nano fiber type net type of structure, you can get easily that it can whatever we want we are getting here. And what is this 1, 2, 3, 4? This is pure pan one polymer, and this is adding the surfactant 1%, 1.5 and 2%. Now what are the fiber diameter? So what we did for each sample, we have taken 10 different place of the same. And from each sample, we have calculated 100 diameters. So we did a distribution curve from the statistics, and this is the distribution curve of the diameter. So like it is the diameter in nanometer scale, and this is the distribution curve. And so similarly, this is pure, and this is by adding the CTAP with different percentage. Now, as I told you here, the pore is important because the PM particle, it will capture on the pore. So we have to calculate the pore. That means the gap between this we have to calculate. So we calculated this pore also by distribution. So this is the distribution of the pore. Okay, this is the distribution of pore. Okay, now what is the summary? Okay, summary is when we are increasing this CTAP weight percentage, we are getting the lower diameter. Suppose this is the diameter distribution curve around 300. So this is 200. That means diameter is decreasing. Similarly, pore size, if you notice the pore is distribution. Distribution means all distribution pore is there, nano to micron. So distribution curve is 3, but here we are decreasing the distribution of pore. So pore also decreasing, and also diameter also decreasing by adding the CTAP. So this is one point which we started from this scientific report paper. The diameter is important. That point we are getting, that we are getting lower diameter by adding the CTAPs. Now, why this diameter is decreasing? There are lots of signs. This is basic the electrospinning characteristics. So we measure the concentration wise, conductivity of the solution, and the viscosity. And if you understand that when you're adding conductivity is increasing due to in the CTAP, there is a lot of ions is the bromine ion is there. And if you want to apply it for the real application, so it should have the mechanical strength. Yes. So what we did, we cut a small piece of fiber, and we hang different type of weight. Okay, so this is the weight what we are hanging here. So we have taken only 30 mm, that means 3 centimeter by 1 centimeter, very small, and the thickness is 20 micron only. And these small things, it can hold 80 gram anti. So that means it is very good mechanical, mechanically very good, strong. And what we did now, this diameter distribution, you can understand this is the top view. Yes, we want the side view, how it looks. So what we did, we side view FSM we study. Then we found there are lots of scaffold like net, so the defined layer. So it nanoscale, it looks thick. Okay, and the top view, that is the, so we got the scaffold and nano net structure, very good nano net structure, and we can understand it's not easy to pass this PM 2.5 from here. Now we have to, our sample is ready, that means our filter is ready. Now how we can measure this? Yes, it is working. So we designed a setup. Okay, so our basic purpose, so we'll give a airflow through this line. Okay, and we'll clamp our filter in between, and from this left side we'll give a polluted air, and if it is working, it will filter here, and at the right side we will get the filtered fresh air. That means we need some sensor, it's called particle counters. So we need 2 particle counter. One will be left side where polluted air is there, one will be right side where filtered air is there. So we are going to measure this pollution here and pollution after the filtration. So you can understand if something is blocking. So if something is blocking, like okay, polluted air is going and it is blocking. So this side also we will not get. Okay, so we have to prove yes, it is not blocking. So for this we change the airflow this direction, and we are going to measure the pressure before this filter, and we are going to measure the pressure after, air pressure. So if there is huge air pressure difference between the right side and the left side, that means air is blocking, and air is flowing properly means we will not get any air pressure. Yes, because air is flowing. So we are measuring this pressure difference. So once you have all this theory, you can get the filtration efficiency, which is in minus out by in divided 100. That is the percentage. And major things, a filter is main property is quality factor. What is the quality factor? Quality factor, we define that efficiency also pressure drop, and both are very conflict relationship. You can get if you want good filtration efficiency, you can make thick fiber. Okay, thick filter. And in this show, what are you going to do? You are going to lose the airflow pressure difference. You are going to lose. Maybe both are conflict, but we want both good filtration efficiency. So that's why we want thin fiber, and we want to get the air is flowing. And this is the details of our experimental setup. So this is 2 counter, one right side, one left side, 2 PM sensor. And here air is flowing or polluted air, and we are controlling the airflow through a fan with the control system, and all the data is coming to our computer. And this is the pressure difference. We are measuring pressure difference before and after the filter. And we are artificially, we have created the polluted air. So how? So agarbatti, burning incense. So we use the agarbatti. So because it is already proved last 2 years, there are a lot of pollution in this agarbatti. And now the point will come to so what we did, so we measured the filtration, we clamped our filter in between this setup, and from the left side and right side, and we measure all this PM count. Okay, and we left side PM 2.5, we keep more than 500, like 600-700. Okay, so we have input. And after filtration, we want very less. Yes, that is our purpose. That should be less than 50. Yes, and we plotted all this filtration data. So we measure with every minute, like 10 minutes, after 20 minutes, after 30 minutes, and we calculated the efficiency. We are almost getting 99% efficiency. And parallelly, we measure the pressure drop also, because we want air should flow. So all the pressure drop is under 30 Pascal. That means almost 0. 30 Pascal pressure means nothing. difference of 30 Pascal. And if you notice, this is a different type of filter, and all filter is showing very good. That means what is the effect of CTAP? We did not notice here. Okay, without CTAP all are showing very good 90% efficiency. Okay, and if we zoom in more, so we will get this data like this. Only without the CTAP we are getting 99.75, and with the CTAP 99.95. That's been the nothing. All are showing very good result. Then what we did, we found okay. We prepared a fiber which is 20 micron. So we started to synthesize fiber with a very low diameter. Okay, like thin, very thin, very thin. And then we surprisingly found the result is changing. So what we found here, so we changes this synthesis time. That is thick to, we want very thin fiber. So we want very less synthesis time. And we found that pure sample without CTAP is 92% efficiency, but here it's showing almost 99 efficiency. That means for the thin fiber, our field adding of the CTAP, this cation, it's working very nicely. And we are getting very good quality factor with the, because of very low pressure drop. Okay, that means okay, we reach at the end a very good filtered materials. Now, if I am telling it's very good, it's not enough. So what we did, we purchased different type of face mask and HEPA filter from the market, which is available from the well-known company. So I don't want to give the name of the companies. So some of them is prepared from some IITs also. So what we did here, so we measure from the same like this black color. It's black color is basically the PP. Okay, PP is very well known. So many of you are maybe wearing this PP. Many of you now are using maybe this medical mask. So medical mask is different. PM 2.0 filter is different. Okay. And we are getting, this is very good glass fiber. It's some from 3M company, and this is HEPA filter. Okay, so we cut it the same piece and we clamped in between our setup, and we measure the efficiency. So okay, let's come one by one. This black filter, which is almost 0 for PM 2.5 filtration. Now the medical mask, any type of medical mask, this is only 20% is able to filter. Okay, for COVID it's okay. COVID is something different, but if you concentrate on PM 2.5, it's not good. Now this 3M company is very good mask, white mask. It is very good, more than 80% also efficiency. And this is HEPA filter. Okay, HEPA, most of the cases for AR purifier, very good AR filter, they use HEPA filter. That is also more than 80%. Okay, and our 90%, almost 99%. This is our filter, also 99%. Now the scenario is going to change when you changing the airflow velocity, because suppose you are in your office, there is almost no airflow, like 1 meter per second maybe maximum. But if you are outside, some wind is blowing, that means air velocity is changing. That time what will happen? The performance is changing. So we checked this filtration efficiency with the more airflow. Then what we found, this white filter, which shown above 80%, it certainly dropped to 10%. Okay. Now this HEPA filter, which shown 99%, it's dropped to 70%. But our developed filter is very stable and good with all this air velocity. Okay. So what we did, we patented it. Okay, in our developed filter. Now the question will come, why our sample is showing very good capture, why it's showing very good. So then we tried to find out that the reason why it's showing very good. So what we did, we studied systematically this is called FSM images. So what we did, this is the pure sample. Now we are giving polluted air 1 minute, then we checked again. Then we found here few dots, PM 2.5 particle is attaching here. Then we given polluted air for 5 minutes. Then we checked the efficiency. So number of polluted particle. Now you can see the polluted particle here. It's increasing. Now we did 15 minutes the polluted air. Okay. And what we found, lots of polluted air. Now you see how very beautifully it's attached throughout the fibers. That means this is the pure. This is before giving this polluted air, and this is after 15 minutes. So that means it is very nicely, it can able to capture all the polluted air. So we want to understand what is this polluted particles. So we did EDAX. Okay. So what we found, there are different type of element is there. We found from EDAX the chlorine also, carbons, lots of oxygen, some calcium, okay, potassium, some silicon. Okay, and as the beginning I told that okay, PM means it's not like a solid particle. There are lots of chemical composition. So we want to understand more regarding this organic. Okay, so what we did X-phase analysis. X-phase is photoelectron spectroscopy. So what we found, so this is before smoke on the sample, carbon, nitrogen, oxygen data. And this is the after smoke. So we found basically the oxygen concentration that we calculated the atomic percentage of this carbon, nitrogen, oxygen, and which was the oxygen 2.7. But after filtration, oxygen increasing 2.7 to 14. That means from the X-phase, we found the major of this organic is coming from carbon, oxygen, and nitrogen. Now, what is the mechanism of this PM 2.5 capture or any filtration mechanism? So generally, it's considered there are three major mechanisms to capture or filtrate any particular matters. So one is called impaction, another is called interception, another is called diffusion. So here we added another term extra, which is called electrostatic for our development. Okay, and this electrostatic, because as I told that it's already proved that the filtration is due to the dipole-dipole interaction or induced dipole. So we added extra term which is called electrostatic attraction. And the filtration mechanism to filter or separate this particular is the comprehensive effect. Yes, it is not like due to only impaction. So it is due to the four effect. And if you want to know more details about this impaction, what is impaction? Suppose some particles when is flowing through the air stream whose inertia is more. So if air stream is flowing and some particle is more in inertia, mean mass into momentum, you can say more inertia, it cannot break the streamline. So it will directly go and hit the fiber. So this is directly coming and hit the fiber. This is called impaction. Next is interception. Interception means few particles whose inertia is less. So what will happen? It can divert or it can break away from the streamline. It will go and it will, when it will come close to the surface, it will attach. So this is called impaction. The next is very common. It's called Brownian motion. Okay, next is very called diffusion. So diffusion effect is based on the motion of the randomly motion of the particle, which is very low diameter, because PM 2.5 means less than 2.5. So some particle is suppose 500 nanometer, 400 nanometer, or less than 1 micron. They're very tiny, so they can randomly go. Okay, so this is called diffusion. Okay, so diffusion, and as I told, the PM content lots of elementary carbon, organic carbon, and different type of ions. That means here in this case, is surface property is important for any filter. And for this reason, in our developed filter is working much better than the commercial filter. Why? Because we tuned the surface property of the filters. For individual filter, if you take individual, we tune the surface property. There are lots of ion is there. Okay, that means here the electrostatic force is working and it is playing the major role. And due to this electrostatic force, all this impaction, interception, diffusion effect also more in our case. So if you summary, so it will come like this. Suppose this is our nanofiber filter. So some particle is intercept, some particle is impaction, and it is diffusion, and this is the electrostatic piece. So in our case, the excellent filter efficiency performance, as I told, due to the two reasons. One reason is that in our case we are getting very low diameter, like 200, 100, even 50, 60 nanometer diameter. So due to the less diameter, the airflow around this individual diameter, the behavior of the airflow is changing. And secondly, in our fiber there is lots of ions is there. Okay, lots of surface ions is there due to the CTEP. And that means you can say that lots of charged particle is there individual in the fiber. And due to this charged particle, it is attracting other charged particle which is attached in the PM. So that's why our materials or our developed filtration very good filtration property. And for the proof of concept, we did a density function theory, DFT calculation, theoretical calculation. And what we found, this absorption energy for different type of pollutant is more for this pan CTEP. This is your absorption energy. So this is a commercial filter. It's -1, but its absorption energy increasing. So that's when our filter is working fine. Now, okay, now what we did, as I told that, okay, low fiber diameter is giving low filtration efficiency. So what we did now, so we taken this 6 weight percentage pan, now we changes this weight percentage of pan to getting the more fiber diameter. So this is the proof that, okay, this whatever the theory we developed, that is working or not. So we changes 6, 10, 12, 15 weight percentage of polymer concentration. What we are getting, when we are increasing the concentration, we are getting the more fiber diameter. Yes, the diameter also increasing. So it was 200, now it is 1000, 1 micron. Then we measure the filtration efficiency. What we found that when we increasing the diameter, efficiency is decreasing. That means this is true what we discussed. So similar thing we did with pan setup also. So pan setup we taken different type of diameter, but we found that filtration efficiency is almost constant because this C to the setup we have lots of charge particle on the surface. And this is the instrument which we have used. This is in our lab. And this is our samples, how it's come. Okay, and same sample we have taken. This is the same image. Okay, same sample, similar samples. And what we are getting 99% filtration efficiency. Okay, easy to breathe because a lot of airflow we are getting. And we have measured not only laboratory scale, from the same instrument we have measured in the very large scale, all are nanofiber, very big, all are nanofibers. This is my 2 PhD students who prepared, and we prepared some developed a mask, also prototype mask. And this is our website. You can visit and you can check our development because now we are developing another aspect. It is coming soon. So thanks to my all PhD students and DST and SMIST, and thank you. So now if you have any question, you can ask me.
Thank you, sir. Thank you for your very informative talk. You have started from the basic and finally it ended with the product development. And you have talked about that real-time air quality index also. I hope hereafter everybody will look at which atmosphere we are. Yes, sir. It is a very informative one.
Yes.
So it is open for question. Participants, if you have any question, you please write it in the chat box or you please mute yourself and ask the speaker. Sir, meanwhile, I have one question. That is, yes, sir. Sir, you have told that is you have added that surfactant CTAB. Instead of that, there is any possibility to add the natural material and we can bring it like materials.
Yes, some natural materials you can add, but we have to check that dipole moment is increasing or not. If dipole moment increasing, we can use, because there is 2 major factors. One is the dipole-dipole interaction here. So if dipole moment increasing, yes, it will work.
Sure. Thank you, sir.
Okay, thank you. Thank you.
Participants? Yes, sir. Thank you, sir. Thank you for your wonderful talk. We will move to the next speaker. Thank you, sir. It is my honor to pleasure to introduce the next speaker, Dr. Paramita Chakravarti. Paramita Chakravarti joined SRM Research Institute and Department of Civil Engineering as Assistant Professor in 2012. She has obtained her PhD degree in Natural Science with full scholarship from Chinese Academy of Science. Dr. Chakravarti is leading the Environmental Science and Technology Laboratory in SRM Research Institute. Her special research interests include freight transport and remediation of organic contaminants. Her research expertise is in the domain of multimedia movement of persistent organic pollutant in the hotspot of India cities, long-range atmospheric transport of persistent organic pollutant, quantification and ecological impact assessment of emerging contaminants of concern in riverine and marine environment of India, cost-effective remediation techniques for emerging organic contaminants in soil, sediment, water and wastewater. She is a recipient of Travel Award by the 13th International Conference of the Specific Patient Constituent to deliver talk in Perth, Australia. She has co-authored first runner-up Best Policy Analysis paper for 2011 in the Journal of American Chemical Society, Environmental Science and Technology. With this, I will request Paramita Chakravarti ma'am kindly deliver your talk. Thank you. Over to you.
Thank you, Dr. Muttami Selvi. That was really a very big introduction, possibly not required that much. Well, thank you. So I think my slides are visible.
Yes, ma'am. Yes, ma'am. It is visible.
So we are actually at the end of our Water Air Research Initiative for Societal Health Indo-US bilateral workshop. So at the end, you all have already listened 10 important and interesting talks. So might be a little tired of listening too much. So what I'll do, I'll just take you to a little bit of a story, like what is happening during this pandemic period. And little bit the topic, what I have chosen is like something known, something unknown for this pandemic, and what actually we need to do urgently to understand or to get the preparedness for this pandemic. So it is slightly different, but then there is some connection between water and air research at the end. So the climax is yet to come. Now, we all know this known fact that the mode of transmission of COVID-19 virus. COVID-19 is the coronavirus disease, the name of the disease. It is caused by the virus because it causes severe acute respiratory syndrome. So that's why it's called SARS-CoV-2. These are the known factors. And we also know like how it gets transmitted through contact routes, through transformation through formatives, like with direct contact or indirect contact. If somebody is coughing, that can be spreaded. There can be airborne transmission of this kind of virus from one person to another. So there can be varied modes of transmission. This is known to us. A little bit not known to everyone is like we want to understand what is the persistence level of this virus in different environmental matrices, which is important because then we need to know like how do we handle it. So in hospital wastewater, there are studies, Wang et al. Like earlier days, this is not a new, like not SARS-CoV-2, but other coronavirus family, which showed like hospital wastewater, it can survive for 2 days. And the new one, like SARS-CoV-2, we have seen like there is a publication where it is showing that it can survive for 4 days in diarrheal stool samples. And even given the fact that it's having an alkaline pH, and it can remain infectious in respiratory specimens for 70 days at room temperature, which is why it makes it more dangerous, and which is why the problem comes with the spread of the infection. So because of its persistence in a particular environment. Now, one is its own persistence. Another factor is like if it is getting transmitted or the droplets are falling at a particular surface, will that also cause some kind of transmission? In that case, definitely the persistence of the virus is important, but it's also important on what sort of surface will it be staying for more time. So there is this interesting paper by Dormalin et al. 2020, he published this. Here, if you see, like different surface materials were studied, and interestingly, what was found was matrices like stainless steel and plastic, they had the maximum residence time for the virus. So virus can be on stainless steel for nearly 5.6 hours and on plastic 6.8 hours, which also is important to understand because we might be using a lot of plastic stuffs where nowadays in our daily life, we have too much of plastics around. So definitely that makes it important that if you have, say, for example, a cell phone casing with a plastic and you know that you need to sanitize. So this is an important thing, like we may not be certain factors which we don't consider much, but when you come to the scientific approach, the surface, the persistence on different surfaces that do vary with the virus. And this is what like should be in our mind. Now, certain other factors other than the residence time in different environmental matrices, and also like to what extent it might be staying. The other factors that determines is also like the physicochemical parameters. Like say, for example, when the previous coronavirus family, like it is said, like the medium has to be slightly alkaline for it to stay longer, which means if it is not alkaline, then we can expect that it might be removed faster. But wherein in this case, in the SARS-CoV-2, like it has been found like it is actually sensitive to heat. Shin et al., they have published this work, but again, there is a question. There were a lot of papers published early 2020 talking about like high temperature could have could have given rise to the lesser spread of the pandemic, especially in the hotter regions, but that did not come true. So again, like those papers are again in question, like whether it's really is a concern like high temperature areas, but exactly how much high. Definitely, this paper absolutely talks the correct thing because it goes up to 70 degrees centigrade. So up till there, like the virus can survive. So which means even if we have a very hot summer month, it doesn't mean that the pandemic won't spread. It will spread if it has to. And coming to this, the same paper talked about like the pathogens that are largely affected by the pH of the medium. So it's a wide range. So it's between 3 to 10 means it can be in acidic condition, it can be there in alkaline condition. So this is again another story that we should understand, like why it is so much staying back in the system and not going away so fast. And sunlight, it like SARS-CoV may be less persistent in an environment when it is more exposed to sunlight or temperature. But then again, the temperature should be high enough, and it's not. Again, there were papers like this where certain regions where the temperature is very high, and then people were hypothesizing that it might not spread. In the early 2020, before the outbreak, like spread it everywhere across the globe. And definitely, yes, the ultraviolet radiation could be another important factor, which is what we are using for, in many cases, like to remove the virus from the surfaces. Maybe nowadays, like many places, you have to keep your stuffs in such a UV chamber just to get it cleaned. And even we did it for our experimental purpose. All the used up materials, you do use UV radiation for that, for disinfection. So what is the need of the hour? So we know something is known, something unknown. So this call came, like this paper came 2020 last year. And it actually talked about, like most of the editors of environmental science and technology, they came up like there is a need for research, especially on the role of environmental factors, right? Which is, again, like if we know the factors, the known factors, the unknown factors, so that we understand how to handle the pandemic better. So that was the need that instigated many of us to come to research or take up a newer area, I would say that way. So what happened is, like this early 2020, this one newspaper clip from Chennai Metro Water, they did some samples for state testing for wastewater samples for SARS-CoV-2. And this was the time when Australia, WaARISH and the entire group, they worked, especially Jochen Muller's group, they worked on the wastewater surveillance. Wastewater surveillance, wastewater-based epidemiological survey is a very important and quite widely used procedure to understand the spread of the any epidemic or a pandemic. And that's a very good way to do that because it is cost effective. So this is the first paper that came out and talked about wastewater surveillance. It came from Australia, and they had quite a bit of a sampling, quite high frequency of sampling, I would say, and so that they could put it up in the model and they could get the mean infection levels in the community in the particular catchment. So this was like kind of a model paper for many of us to initiate the work. So what we did, like then we had some idea that we need to come up something, especially in a developing country like ours, where cost is definitely a very big factor. So how do we handle this? And as an environmental person, what do we contribute? In that regard, like I could somehow draft a synopsis kind of a thing and like put it up to a friend, and then it went for a funding. So we are very much thankful to Space Agency for Development and Cooperation for funding this idea, but this came up pretty good. So there is a little bit of difference in the story, but that I'll be talking a bit later. So the outline of this story would include like, how do we do this work, the cost-effective, monitoring of early signaling of SARS-CoV-2 in Chennai city, and what did we do, how did we do, and what are the results that we got, and how do we take it as a way forward, and whether there is something else also coming up in addition to. So what basically is this wastewater surveillance? In a very simple way, what could be said is, if you see that in the wastewater, the raw sewage will have the viral load from the stool sheddings, and the virus has the nucleocapsid. This nucleocapsid, if you see the genome structure, two important genes N1 and N2. These are the ones like which can be quantitated, which is similar to how we quantitate the human samples, like when they take the swab samples, same way reverse transcriptase RNA polymerase chain reaction is done to test the quantitate, like whether the sample is having SARS-CoV-2 or not, positive or negative. So this is the basis that they use in a wastewater-based epidemiology system. So if we know that, for example, a particular catchment, if we know the number of people in that catchment, and we can quantitate like how much amount of raw sewage might be coming from that catchment. So once we know the viral load, we can estimate, we can predict the number of infections. So that is basically what Ahmed et al.'s paper also worked out, similar wastewater-based epidemiological approach. So not only this Ahmed et al., like followed by that, there were a lot of other countries, and including in India, like a lot of researchers started working on this, and there were a lot of stories coming on like how they can predict this kind of infection spread. So IIT Gandhinagar, Dr. Manish Kumar, who gave a talk this morning, and he also did excellent work in IIT Gandhinagar. Then Dr. B. Lal Institute of Biotechnology in Jaipur, they also did quite a good work in India. Then there's another group in Hyderabad, they had been doing good work. And a lot of other countries like France, and there are Dutch scientists who had been working just to understand like what would be the spread of the infection by regular monitoring of wastewater or in WWTPs. So what was the question that we had? One question was like that I was thinking when we were planning for this wastewater surveillance, like is there a chemical marker that can be used like as a tracer for human wastewater? Now, why this reason? Because we are aware of the fact that a lot of sewer outlets are there in the river bodies in our country. So in that case, there could be also viral loading onto the riverine bed. And if we know the chemical marker to what concentration, like which are the areas it is loaded, we possibly can understand like what could be the viral load. Okay, we cannot in that case, because we are not knowing the entire number of population, so we may not be able to estimate the exact number. But then again, we can at least have an understanding like which are the areas we should be careful about. And then the question arises, like we were in lockdown. So once the lockdown gets over, the post-lockdown phase, are we going to see a change in this distribution pattern? If at all, then what we would be seeing and how we should be careful about it. And another important question is like because you see, talk about social distancing. Now, this is again interrelated with the socio-economic status, the health conditions, because say, for example, in the slum dwellers, the community is very closely located. So the social distancing factor in those low-income group communities is really a challenge. So how we sense that, or how do we understand, or how do we help them in order to understand that, like we need to have a better way of handling the problem. So we had seen KFIN, like this morning, Professor Dan has talked about this paper already, like, but I'm taking a different approach. I'm trying to understand, like, we found that caffeine is a potential marker for human waste, then it was there in my mind, can't we use caffeine again and try to see that whether in a very diluted condition also, this caffeine can tell, okay, this is the area where there is too much of human activity. And can we go ahead and try to see the viral load and understand which catchment would be more problematic, apart from checking with the STPs? The river beds can also be checked like this. So, and also another point was also there in our mind, like this could also give us an idea whether the STPs are functioning properly or not. So what was our objective? We wanted to measure SARS-CoV-2 in Arden rivers, in the treatment plants, in the pumping stations, and in SRM hospital wastewater. But there was different planning for this design, and to investigate whether there is any co-occurrence of different sort of chemical markers. So we use different pharmaceutical compounds also we included in the list. In addition, this chemical markers like caffeine and carbamazepine were also there in the list. So how did we basically do the experiments? Let's see. Well, before I explain the experimental design, I would say I have to thank Metro Water because they came up to help us. Otherwise, accessing all the sewage treatment plants or the sewage pumping stations were not possible. So definitely, we are very thankful to Metro Water, and it was a joint initiative to work in those treatment plants and sewage pumping stations. And for us, we had SRM hospital wastewater. So daily basis in different time intervals, we were monitoring SRM hospital wastewater as well. We were in the site. We had a very excellent team, my entire group. I'm so very thankful to them. Like, of course, I have their acknowledgement at the end, but still I would say we analyzed the samples in the same day. So we had gone early morning, we'll go collect samples, and sample collection is also not easy. A lot of protections, a lot of care was taken during the sampling phase. And we also ensured that the wastewater we are collecting, that we analyze it on the same day, so that we don't lose the information that we want. At the same time, that we don't put other scholars who are working in the lab into trouble. So this is the map, Chennai map. So we have Kuam and Adir, two important rivers. And we have the STP's location. You can see in the map, the 4 STPs, Kodangayar STP, Coimbatore, Nesapakkam, and Perangudi. And close to the STPs, we also have pumping stations. So near Kodangayar, we had 2 pumping stations. Rest of the 3 STPs had 1 each. So all these 5 pumping stations and 4 STPs were considered for this project. And in addition to that, we also were monitoring the river samples, river water samples. Interestingly, for river water for Adir and hospital wastewater, we could get samples before the lockdown, during the lockdown and post lockdown. So we could see the changes as well. And those are really, really important messages that we have. So we had been collecting samples from STPs, from the influent, from the primary sludge and the effluent samples for each of the STPs. So basically what we did the sample extraction, which is basically the wastewater extraction, I would say, but to concentrate the RNA of the virus. So we tried to follow 4 different procedures for the same sample, and then subjected to N1 and N2 genes and RP for the human gene, just to have a control sample to check that as a checkpoint for our analysis. This 4 methods, supernatant, composite, sediment, and syringe filter, so each sample was subjected to 4 different methods, and they were also done in duplicates. So the first method, supernatant method, is a bit elaborate, and this method actually is practiced by several other groups. They have followed this method. So this method was completely, we were following whatever was given in other literature. We did not make much modifications here. So this is elaborate because we take the sample, centrifuge it, then we try to sample to lower down, adding a alum to it, and then we use the supernatant, filter it out, and finally we try to concentrate it for the RNA after passing this Amican ultracentrifugal filter. So not only it is a bit time consuming, another thing is each step is very essential. And we had to be careful with the fact that we properly take care of the supernatant, and while we are filtering it, we don't miss anything. So those were the steps, and it's a prolonged step. So manpower was divided. So we did not waste the sampling time, the sample extraction time. It was done within 24 hours. This kind of technique was used. But then there were three important techniques which we also incorporated. One is this composite sample technique, or sediment and syringe filter. These techniques cost between 1 to 2 hours time, and these are faster. So we could also compare whether this faster technique would be more useful. And the previous technique, where this Amican filtering devices, whatever we are using, that is also very expensive to concentrate the wastewater. But in these three techniques, it wasn't that expensive. Apart from, of course, the most cheapest one was the sediment procedure. So a loop of sediment was taken from that 20 to 50 ml of the sample, and then it was subjected to proteinase K for the RNA extraction, the next step. So for the composite and the syringe filter, only for composite we chose 1 ml of sample, whereas for syringe filter we took only 10 ml of the sample, and then the filtered sample was stored in a 15 ml Falcon tube for RNA extraction. And in between, like this composite method, just directly the 1 ml of sample was taken in the centrifugal tube, and then 10 ml of proteinase K was added and placed in a dry bath for 10 minutes at 65 degrees. And then it was subjected to, again, RNA extraction. So for all the 4 methods, once RNA was extracted, it was subjected to RT-PCR analysis, and the counting of the RNA copies was done using the nano drop. So all the instrumentations we have, Raman research block, so it was pretty easy, and my colleagues were really helpful. Like, they gave up their instrument most of the time for this project. Now, what did we see in the result? Let's look at this. Basically, when we get the RT-PCR result, the signals comes like the cycle threshold value. The signals comes like this, and we have something called the CT values. So nowadays, we all know what is actually a CT value is, because whenever we go for a testing, so it will give the CT value. This is actually the cycle threshold value. And the cycle threshold value, they say the lower it is more, it is indicating more of infection load, and higher, but then again, it also has a range. 23 to 40 the range that is considered, and if it is closer to 40 means the infection load is less. Closer to 23, as it goes down up to, it is more infection. Of course, for human samples, it can even go up to 17. In our case, what we saw is in the STP sample. So if I try to explain you that we had what are the categories of sample. One is STP and the pumping station. Then we had the hospital wastewater on different time and everyday basis, weekly basis, what we collected. And we had the other category was the river samples. So these are the data that we got from STP and sewage pumping station. So if you see that more or less, mostly for all the 4 methods, all the samples were found positive, which was one signal that all the STPs in the inlets, it was positive. But outlets, all the outlets were negative for all the methods, all the 4 methods. This was the first information. Second is we found like if the outlet which was not having the load, but we could see in the pumping stations, we could see quite a good amount of viral load. Now, if we compare the 4 techniques, why did we go for the 4 techniques? And definitely We named it in a small way. COM is composite, SED is sediment method, supernatant, SYR is syringe method. We found that definitely supernatant is giving like how everybody else got relatively a good result. But we found like sediment gave very good results, especially for switch samples, the STPs and the switch pumping stations. This we are suggesting that given the fact that SARS-CoV-2 being hydrophobic, these envelope virus might show higher partitioning onto the solid matrix. So sewage or the sludge samples having more solid material, then might be more partitioning onto particles. So which is what we thought, like this is the reason why we were seeing more in the sewage and STPs. And in another way, I can also say if there is a budget constraint, we can go ahead with this kind of with this kind of experimental procedure instead of going for the supernatant basis. But on other condition, like if you look at the hospital wastewater, because hospital wastewater was not having much of the dissolved or suspended solids, so what we saw was like supernatant was much better method. If you look at the CT values for our SRM hospital wastewater, it even for many cases it was less than 30. And there was this one coincidence, like out of the 4 dates of sampling, like every day, like every weekly once we did sampling, 3 weeks during the lockdown and 1 week after the lockdown. And every day we did it for 5 times a day, like 7 a.m., 10 a.m., 1 p.m., 4 p.m., and 7 p.m. What we saw was there was an increase in RNA copies a particular date when the more number of, because we had this information also, like more number of patients were admitted here. So that gave an indication, definitely we will have more viral load. So from wastewater itself, we get that indirect understanding that the viral load is more, means more number of patients might be coming in. So now the factor is, what did we see in the river bed? In the river, COM and RDR, both the cases, during the lockdown, we saw that the levels were relatively low on the river bed, but post-lockdown, the load was increased, especially in certain areas in COM. Of course, COM had more viral load, especially in the slum areas in the northern part of the city, where the congested community is there. There we could see like more viral load on the river, and relatively RDR was less impacted. Now here another interesting thing, because we got the river results before, so we could see that if the 2 markers, caffeine and carbamazepine, if we compare, like we were saying, like okay, caffeine can act as a human-based marker, chemical marker. So in that case, if you see like where the loading is more, the concentration of caffeine is also more. So this is something very important to understand. Like, so if we have monitoring for caffeine, and then we can say like which are the areas where we can expect more community dwellers activities, and then associated viral load. And then looking into the STP samples, what we saw is the inlet all were positive and outlets all were negative. We cross-checked our results with the NABL accredited lab because that was important. We established this lab for the first time, so it was important to check back. What we again saw in the STP samples is in the outlet, 85% of removal, on an average more than 85% actually in certain samples, caffeine was removed, and there was no remnants of the viral load in the effluent, which means that in Chennai, the STPs are functioning very well. They are taking care of the STPs, the functioning, normal functioning, which can treat, which can actually take care of the viral load, and the treated water is clean. Treated water doesn't have the viral load at all. But other than caffeine and carbamazepine, like we also did some of the fluoroquinolones, which were pretty high, and most importantly, we found this azithromycin, because azithrol, it was very, very frequently used drug during this period. So which is why we are seeing that azithromycin may be used. The usage of azithromycin caused high loading of azithrol. Now, how do we understand whether now the connection, like, okay, what we are seeing, can we statistically understand in some way that caffeine can be used as a good marker? Now, what we try to do is we try to use a dynamic range, which is nothing but the ratio between the arithmetic mean of the concentration of this organic tracer and the RNA copies of SARS-CoV-2 that is coming in the influent of the STP. So this is one approach, the dynamic range. This dynamic range again can help us to understand like whether both the viral particles and the marker are staying together or not, or are we seeing the concurrence of this or not. So if you see for all the method, for both N1 and N2, it's more than 90% concurrence with for caffeine. And because the dynamic range is so high, which means caffeine value is like more than 8,500 for in the dynamic range, which indicates that even in a diluted condition, this marker can be used, not just necessarily in a concentrated concentration, which is again true for when we are talking about the river water sample. So if there is a sewer outlet, if there is a community, we can actually understand, if not the correct complete prediction, but some amount where the preparedness is needed before a next hit of the next wave of the pandemic can be understood in this manner. So what we did is, like, if we know, as I was telling you, with all the 4 methods, what we got, the viral load and the catchment population, each of the catchment population, Metro Water gave us all the informations. And then what we tried to understand is whether the number of people affected we can get if, say, for example, the population is 100 units of population is there, and we get a certain amount of viral load. Each person will give out like 2.58 gram, 258 gram of stool, and then we can back calculate, like, if this much of viral load, this much is the population, we can quantitate what would be the predicted number of cases depending on the viral load. So what we saw during that period is that these are the 2 sediment and syringe filtration method. These 2 methods worked very well for STP and pumping stations in our story. And also you see the predicted infected cases where percentage was matching with the number of cases even in that particular catchment. So in an indirect way, this is a very, very good technique, because if a catchment is having a population of, say, 10 lakhs, we don't need to test 10 lakh people, but we can monitor every day this wastewater sample. Say 100 samples may be good enough to make us understand like what a particular catchment would look like whether there will be a viral load, because both symptomatic or asymptomatic patients can shed the viral particles in the stool, and then we can back calculate and predict how much quantity it may be there and what would be the number of infections spread. But for this, a constant monitoring is required. Now, this is not the end. In my group, we mostly work with organic contaminants. So if you're working with organic contaminants, it is also essential for us to understand, like, is it just the water quality when the lockdown time, viral load in the river matrix is going down? But is it also true for a lot of organic contaminants which are endocrine disrupting chemicals mostly, or which may persist for longer phase? Since in my group we work in this, so I had this interest, like, let us work out. Already some of the scholars are working in different areas of such contaminants. So let us see whether this, when the water quality, everybody is telling water quality is improving, air quality is improving during lockdown phase, is it true for this kind of organic contaminants or not? So what did we see? See, the problem is now, like, especially the plastic waste. During this COVID-19 pandemic, we have accelerated the usage of plastic in every sector, including the mask we are using, or even the oxygen mask that are used in the hospital. So this quantum of biomedical waste, mostly the plastic in the biomedical waste, has increased. And the United Nations, they have given a very clear-cut information that especially the South Asia region or Asia Pacific will be extensively loaded with this waste plastic, and which is actually coming to the river stream. Now, why I give this photograph of mine, and there is also this two are the ones which we took in 2020 during our sampling campaign. We'll show you more photographs from our scholars also, like when we are visiting the sites. So this is a situation happening. This is in Daman Ganga in Vapi in Gujarat, and this is a case in Adir Kuam region. So what I mean is that the plastic loading, these did not stop. And the quality, like the plastics were ending. There was already past few years, we are thinking about like plastics are ending into the water bodies. Now it is even more. And which is what UN also has come up in their reports. They have talked about it, like there will be more release of plastics, especially from the biomedical waste. So not only plastic is getting into the water bodies, but they are also burned. The materials waste open burning is not a control factor. See, look at the locations where we had been going during this pandemic period, and fortunately, none of us got infected during that phase. While we are doing sampling, we were careful with all the with the protection and other PPE stuffs. We were maintaining it. But look at the waste that we generate and the burning of the waste. This doesn't stop anywhere, even though whether it's a lockdown or it is not a lockdown. So although the economy can be affected, the other sort of air quality might be affected, but not certain kind of contaminants. So what basically happens is when there is burning of such different sort of plastics or different kinds of plastic waste that is coming to the solid waste stream, that can lead to lot of emerging contaminants, which will have endocrine disruption potential. So we were seeing what we saw in the surface water was like some of these contaminants levels were actually higher during the pandemic phase. This is something possibly unknown to you, so it's kind of a new information. One of the scholars who is working on legacy POPs, Ronnie Rex, he has worked out this before the outbreak of the pandemic. Of course, we did not know that pandemic was about to hit. So September 2019, there were samples, and there were samples from September 2020. What we are seeing is like these are polychlorinated biphenyls we saw, and like increment in certain congeners during the pandemic phase. If you see here, the bottom one is the pandemic phase. So certain congeners which were not visible, especially I would highlight this PCB52. This is a good marker for open burning. We have already found, we have established like PCB52 is a good marker for open burning of solid waste. So which levels, which was much higher than what we were seeing during the pre-pandemic phase. And this was much higher during the pandemic. And not only that, among the organochlorine pesticides, especially DDT and the PPDDT, which was not seen at all during the pre-pandemic phase, but PPDDT was pretty much high in the surface water. We are suspecting possibly because this DDT is used for vector control purposes, which could be a reason. Now, not only this, as I was telling, like this kind of chemicals, open burning of solid waste associated with polychlorinated biphenyls or even dioxin-like polychlorinated biphenyls kind of compounds. We also did another set, which Sarath Chandra, another scholar, he's working on this kind of burning associated with dump waste. And actually, he was there in the previous photograph as well. So we are getting into this waste and trying to understand the problem. So he also, like the phthalic acid esters that analyzed in the river water samples were pretty much high during this phase. So if we say that, yeah, our water is clean in terms of surface water quality, but that is not the case with such organic contaminants. What is the message for you? Now, if the burning activity is happening and we are seeing such things in water, does it mean that we cannot, we can ignore atmosphere? We could see, interestingly, for one of the flame retardants, which are added in most of the, most of the consumer products, especially the electronic gadgets, etc. We could see Chennai city alone in the atmosphere, the BD level, one particular container, the level increased since 2007 to 2020 now. So which means, apart from that, I told you about PCB 52, we are analyzing all these compounds, a cocktail of compounds, trying to understand, like, so the atmospheric emission has also not stopped because of this burning activity, which is not stopping. The amount of quanta of waste we are generating, that is not stopping during this pandemic phase. So, and an important point that a kind of a take-home message is, like, we just recently got a paper in ESNT wherein different cities we've tried to model that to what extent, what would be the residence time for a air parcel. And we see that in the inland cities, the air parcel can stay longer, which means like the inland cities like Bangalore or New Delhi might have a more severe impact of this organic contaminants. And if they are released in this good amount, like if there is an increasing flow. So not only that, it also depends on certain other characteristics, but coastal cities might be a little lesser impacted over the inland cities, but still there can be a short-term impact that we cannot ignore at all. So this is another unknown information, like even though we are talking about SOx, NOx going down in the atmosphere or surface water quality improving, a lot of people came up with that, but it doesn't hold true for a lot of disease, endocrine disrupting chemicals. So a simple take-home message. So what we get is, yes, wastewater surveillance can be a quick, cost-effective method, and we can use CAFIN as a marker, which can help us to understand the functioning of the treatment plants, and also in water bodies, especially in developing countries where the sewer outlets are directly on the riverine bed. We can possibly understand which region of the pandemic which region should be more taken care, or we need a preparedness. If we have a plan, I think we can better face it. This pandemic is like war, so we need to prepare ourselves. So to prepare ourselves, we need to have this kind of surveillance at the background so that we understand which catchment to take care faster and how do we handle that. And of course, one important message is that ongoing entry of this organic contaminants are not going down. So health impact associated with this kind of contaminants, whether it comes to water or air, in both the aspects, it might be of serious concern. Well, now it's time to acknowledge. I hope I stick to my time. And firstly, yes, we had a very good biomedical waste management in SRM IST. I have to thank the scholars who were involved. Yes, Chandra, Ronnie Rex, they are working on this different data set with different EDCs. And Nihal Mohammed, whose face is not visible because we were in the sample collection sites, and he was a RA with us. We also had Dr. Sundar as a postdoc for that project. And we had all Metro Water engineers also helping us during the sample collection. So I'm really thankful to all of them. And the spontaneity the scholars had was amazing. And it was a fabulous teamwork. And I have to especially thank Mrs. Moitri Mukhopadhyay, who's a senior scholar in the group, and she was in the background and helping all of us in several ways. And that was a big contribution from her side in this project. And I'm earnestly thankful to Dr. V. Thirumurugan, our Associate Campus Life Director, because whatever waste we generated during the experiment, he has taken complete care of that. So we were by abiding all the WHO rules and regulations while we were doing this analysis or continuing this project. And nevertheless, all the group members, some might be, are there here, or some have left out, they have moved on to different new places, but they all have contributed a lot in understanding the story that we do with emerging contaminants. So whether they were part of this project or not, but I really thank them for choosing this group and being with me for whatever time frame they were with me. And in this group is theirs. And we really enjoy research here. And only motivation is we want to give some information in terms of societal perspective, which is why I think is also the agenda for this workshop. With this note, I say I thank you all for listening to me, and if you have any question, ask. I'm here for all the questions.
Thank you, ma'am. Thank you for your wonderful talk, as well as a very informative one. So we learned two things from your talk. One is your talk is mainly now, I think now towards it is environmental awareness, and everyone know about that COVID. In addition to that, we know that the lockdown is only taking a period to rest. But based on your presentation, we are able to see that how much the environmentalist is work for the society. Thank you for your work. So now open to the question. Participants, if you have any question, you can post it in the chat box. Yes, ma'am. Thank you, ma'am. There is no more question, so we will close the session, and I will hand over to the session to this person. Thank you.
Thank you. Thank you very much. Thank you.
Thank you all for participating in the two-day workshop. This concludes the series of lectures for the workshop. I invite all of you to join for the validatory session. There is a message from the convener USA, Dr. Daniel Snow of Nebraska, and here is a message from him.
I would like to thank you all for participating in our Indo-US bilateral workshop, especially those of you who submitted posters describing your research. I am constantly impressed with the quality and scope of your work. Keep it up. We owe much thanks to Dr. Paramita, her students and research staff for organizing this event. I also want to personally thank SRM Institute of Science and Technology for hosting, Doherty Water for Food Global Institute for technical assistance, and especially IUSSTF for sponsoring our workshop. Finally, I would like to announce that we received approval for a special issue in Environmental Toxicology and Chemistry, the journal put out by the Society of Environmental Toxicology and Chemistry, featuring research related to this workshop. Dr. Pero and I look forward to seeing your articles in the next few months. We will be sending out an invitation very soon. Thanks once again.
I kindly call upon Convener India, Dr. Paramita Chakrabarti, to announce the list of winners of the e-poster presentation session and share a few words.
Well, I think we are at the end of this session, this workshop, 2 days workshop. I believe all the participants enjoyed each and every talk. All were stalwarts in their fields, and I believe they gave very interesting talks, very important ones as well. We also had early career researchers. They also delivered very, very important and interesting talks. Though due to time limitations, they had a limited time, but I think each and every presentation was excellent. And we all have enjoyed thoroughly each and every presentation. We had also a poster session wherein we had judges from SRM IST, including Dr. Muthumi Selvi, who is here, and Professor Shivakumar, Professor Purushothaman from Civil Engineering Department, Dr. Muthumi Selvi from Chemical Engineering Department. We also had Dr. Girija Bharat from Miyugama Consultants, New Delhi. She's the director of MGC, and she's a sanitation health expert. In addition to this, Professor Daniel Snow has also looked into all these posters and given a score. So we have got a normalized score for all participants. And of course, who did not win doesn't mean that they were less or anything. It's just a beginning. Some people scores a little more, some people scores a little less. That's part of life. But I believe you enjoyed presenting. That's more important than anything else. We will announce the name of the winners, and I would like to mention that we would need you to send your address, physical address, so that we can send you the certificates and other stuffs. And yes, so let me not keep you waiting, and let me announce the results. For the water category, we have two people who won the award, and Vamshi is also a very bright fellow. He's an inspired fellow. Vamshi from the Department of Civil Engineering, Osmania University, Hyderabad. He's one of the winners. And we also have Shyam Babu from Department of Civil Engineering from SRMIST in the water category. They are the winners. From the air category, we have Ms. Kanishta Dubey from Department of Civil Engineering from IIT Kharagpur. Kanishta won prize in air. Along with her, Sarat Chandra from the Department of Civil Engineering, SRMIST, won another body for this category. And in the health category, we have Dr. Shomik Goswami, who's an assistant professor in Department of Zoology in Sunbeam Women's College. He really, his poster was excellent, and his work was really identified by most of the judges. And Christopher Olson was the other one who got from Florida Agricultural and Mechanical University. Christopher also working on this. I think it's something between the health sciences and also community services. So that is another important arena in terms of societal impact. So Christopher Olson also is under health category. I congratulate all the winners, all the participants, and I wish that they all stay safe and enjoy more of this kind of future connections with us, especially with the spatial issue. As Professor Dan said, like, we are coming up with a spatial issue, and we will be announcing those. Yeah. So with this few things, we would like to wind up the session and this valedictory session. And again, once again, I'm thankful to entire SRMIST fraternity, especially our Vice Chancellor, our Registrar Sir, and our Director Sir, our Dean Research, our Head of the Department of Civil Engineering, like for everyone for being there, for the support, all my colleagues, friends in Department of Civil Engineering and SRM Research Institute, and also all my, most importantly, all my research scholars who have taken really enough effort to make this program a success, and also IUSSTF for funding this 2 days workshop. And please remember, we will have this spatial issue. Once again, I'm telling, it's in the Journal of Environmental Toxicology and Chemistry, and we will come back. I hope some of you would be interested to submit a paper. Thank you.
Once again, I thank you all for attending WaARISH 2021. I kindly request everyone to rise for the national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे